I am the president, Davide. Can you believe it? It's incredible. We have today 25 talks, 9 on Zoom, and we have 3 posters. If you're interested in the posters, they're all at the entrance over there, okay?
Now we have 5 posters.
5 posters. Okay, so it's changed recently. If remember to register at the entrance as well. And I'm going to give you also a few tips on how to run this meeting smoothly in a second. Interestingly enough, we need to thank Julieta because she put together a fantastic conference this year. Both in terms of diversity of talks, we have from, I think, all the orders and also some minors group like Collembola, mites, and we range from systematic, taxonomy, ecology, behavior. Then we go to genomics, and we also have some sort of databasing and projects on this sort. Also, the presenters, the diversity of presenters is amazing. Local presenters, North American, but also we have South Americans, Argentina. We have people also from South Korea that join us live and also online, I think.
Yeah.
And then, of course, we've got some Italian folks. So that's pretty interesting.
And Russia and England.
Russia and England, yeah. Fantastic. So thanks, Julieta. Please, again, another round of applause. I will be your moderator for the first half, then Alessandra will switch and moderate the second half. There are some differences allocation for timing, so we are gonna try to moderate that. So essentially what we're gonna give you is 5 minutes prior to your end of your talk. We're gonna announce it for you when you're there, so please look at me. I'm good looking, so you can do it. And then at 1 minute left, I'm gonna also tell you, you got 1 minute, and after that, so you can wrap up your talk, and after that, I will just push you out of the stage, okay? So please, we have a very busy schedule. We have 25 talks, so please try to remain on schedule, especially people that are present live. The pre-recorded one should be relatively good to go. We have 3 coffee breaks, okay? There are over there, and also we have a lunch that is provided by the CSE. So if you're interested, you can stick around. The coffee is located right here, anytime that you want. We're asking you that when people are presenting, please take the big loop outside and go to the back instead of crossing this stage, okay? At the end of the entire program, we have also the business meeting, which is open for anyone. If you want to stick around and understand what we did the last meeting and also what we are going to proposing to do, please stick around. If you're interested, you can also help us maybe in the future. Bathrooms, which are really important, are located down this hall. You go out this way, you turn left, and they are exactly down the hall. All right, I think that's it. If you have any questions, otherwise I think we can start. David will help you out putting on your slides, and I think we can start with the first talk. Do you want to say anything, Julieta?
You can exit to the stairs, the short distance, but you cannot come back into the stairs after 10 o'clock. They'll be locked and unmanned, so you can't come to the front of the museum.
Right. I think we can start.
We can, but since we have a few minutes until we're scheduled to start.
Okay.
I wanted to ask, I don't believe I have the slides of the person presenting at 10:15, before the last talk, before the first break.
He's gonna present live.
Okay, great.
Okay, yeah. Just email me this morning.
Excellent. Just making sure.
Yeah. Right.
So yeah, I think we're ready.
I think we're ready. Perfect. So the first talk is a pre-recorded video by Jackie Miller on the highlights from the Miller Island Lepidoptera archives. It's going to be pre-recorded.
Before I started, can you confirm that it is showing up on your screen? The screen share is working.
Is it dark enough, light enough?
Perfect.
Okay. Should I leave that door closed or open? Open. Open.
I apologize. I'm gonna pause this because apparently we're not sharing audio right now over Zoom. So I'm gonna just take a quick second to try and fix that.
Could it be louder or is it?
Just have sweet time.
After the publication of the West Indies book in 1994, we focused more intensely on the butterflies of the Bahamas and also did some macromoths, collecting usually up to the light sheet wherever we stayed, along with the Arctiinae. We collected Sphingidae, having recorded 35 species for the island thus far. In 2010, we got a little bit more serious about moths, including micros. In 2014, we received a National Geographic grant for our project, the Lepidoptera of the Lucayan Islands. We were able to collect on the 13 major islands. We recorded a total of 1,007 species for the archipelago. There are 54 endemic species in the Bahamas and 14 new species have been described. Andros is the largest island. It includes North Andros and South Andros. Most of our work was done on the North Andros, which had been two noteworthy species, Batis de Villiers and Dionysia carteri. Both species also are included in Cuba, with Dionysia was the only rhyodentid in the West Indies. One of the most vexing butterflies on North Andros is a subspecies of the flasher, Telegonius sagua. Herrick Finch collected one in 1973, and Don Harvey collected some in 1976. We saw them, but often did many trips down this lonely road of tears, as it were, did not result into any species specimens. So in 1977, we published the Telegonius sagua harvey based on the existing material. On South Andros, we were pleased to find Coranthus richmondi. Herrick Finch asked Lee to identify the skipper from the Exumas, which led to the description in 1965, and it was Lee's first adduction to the fauna of the Bahamas. Our first were not only a record for the South Andros, but also the first specimen for all of the Andros. One of the highlights of the Grand Bahamas and Abaco was having Cytheronia supracalis come to our lights. These were the first specimens ever collected on the Bahamas. This specimen and the Io moth are the only ones of the Saturniids that occurred in the Bahamas. Halloween night on Abaco was especially memorable because of the most common moth at the sheet, which was this orange and black species of Pseudocheros. These moths were quite docile, and Mark Simon was able to put more than one on each finger. Another amazing sight on Abaco was hundreds of Atala butterflies, actively nectaring. Atalas were also very common on the Grand Bahama Islands and likewise occur on North Andros. Many of our trips involved overnight stays in Nassau, connecting flights to other islands. We did some collecting with the University of the Bahamas, and we also presented workshops for the students and also provided identifications and teachings collection. A highlight of Eleuthera Island was working with staff at the Levy Preserve. We collected several locations on that island and included the preserve as well. We were also provided identifications of the Lepidoptera for the collections at the preserve. Cat Island has been a favorite over the years. It is known for Mount Alvernia, which is the highest point in the Bahamas at 206 feet. The Tortricid Megalota insularis is described from a single male, which we collected. On Exuma, we were excited to find Callistocyphila. The species was only known from New Providence Island and North Andros. Populations were very localized, and in 2014 we only included 2 individuals. We were fortunate to be on the island after the significant rains. There were lots of other nectar sources available. There were several Lycaenids active, including the subspecies of the Miami blue. One of our stops was also at Dean's Blue Hole, this is the deepest blue hole in the world at 663 feet. On San Salvador Island, we stayed at the Jurassic Research Center. We discovered a new species of hawk moth, which we named in honor of Kathy and Donald Gerace, who were responsible for the development of the center. San Salvador is a very small island. Collecting was very valuable for butterflies. Mark Simon and Gary Goss did most of the field work for Crooked Island. In 2019, all of the scientific research permits were suspended pending a review with respect to the Nagoya Protocol. I'm happy to report that after 3 years, Mark and Gary were able to go back and to obtain permits and visited Acklands and Island and last year. Mark and Gary also worked in Mayaguana and Great Inagua. We described 2 species in their honor. Galchethi simoni is from Mayaguana and Turks and Caicos. Galchethi gossi is from Great Inagua. I hope you enjoyed this quick tour of the island highlights. I'm especially grateful for all of the research opportunities I've had over the years, and to all of the people listed here. I want to give thanks to Gary Goss for live moth images, and to Deborah Matthews for helping with the PowerPoint preserve. I thank you again for honoring me at this year's Centers for Systematic Entomology meeting.
What we saw in this talk was all the life achievement of Jackie Miller, which has been extremely successful in many things, not just as a taxonomist, but also as a biogeographer in her lengthy career. She recently retired from the Allen Curator of Labs at the McGuire Center, and she published 3 books and more than 158 papers, which is outstanding. She's currently recognized and internationally recognized as a systematist, biogeographer, taxonomist of Lepidoptera on the West Indies and the Bahamas overall. She has been also secretary for ESA, vice chair, and also chair for different sections. She's been extremely involved in almost everything that deals with entomology. More recently, she has been awarded as a fellow also for the ESA, and we couldn't not award her for a lifetime achievement also for the CSE and a membership. And so I'm inviting Debbie to come here and the treasurer, please, Paul, to give to Jackie this lifetime achievement award from the CSE. And
Debbie?
picture.
Thank you.
Debbie, do you want to say something about Jackie?
Well, as you can see, she's having quite a bit of trouble now. I think she had a stroke at one point, but she's doing okay. I mean, she's still happy to be involved as much as possible. And so this project of putting together a PowerPoint was, it was inspiring for her. So anyway, she really appreciates being honored at the meeting, and I thank you too.
And I wanted to thank Debbie for all she's been caring for Jackie. She's been with her a lot of the time at her home.
Yeah, she was basically away from the office for a year, and then, at that point she needed to retire.
Yeah.
So,
Thank you, Debbie.
thank you.
Thank you again, Debbie. Also for the presenter here, whenever you present, you can stand right here where I am. You don't need to be on that thing, because we have the speaker there for the people online. Okay. Got about time?
We are ahead of schedule.
We are a little bit ahead of
I
schedule.
have a question. Is there a microphone or there's no microphone? For the people that are speaking here, can they hear in the back?
You do not have a microphone.
Okay.
Please, I guess, just do your best to project your voice for back.
I think we can proceed anyway.
I think with the closed captioning, the mic should pick it up and do the transcription. No.
You're talking about
This-
the people in here.
Yeah, if you're recording it, it'll throw the transcription on screen. Closed
Do
captioning.
people want a transcript appearing under their, are you saying? You're
That
saying
might
people
get in
actually
the way of
just-
some of the, if people have got text, I would just recommend against that, unless some people, well, it might interfere with some people.
That's what I was concerned about. If we have an actual text on screen, it's gonna block text on slides.
Okay.
Unless there's somebody who's got a very quiet voice.
So go to slide.
We do have a slide advancer. It's right there. It's got a laser pointer as
So the
well.
person could speak on the other side.
Yes.
As well.
Right, what people were asking about is not, that speaker should make things very clear for people on Zoom to hear. Someone was asking about a physical microphone, a traditional microphone for people in the back row that might not be able to hear somebody. So we do not have such a microphone. I am just requesting that presenters project enough to hear people in the back.
We also have 10 empty seats.
Yeah. If anybody feels they're hard of hearing and just having people hearing in the back, speak up.
I have one little science question about Jackie's presentation, and that is what Hurricane Dorian did to these populations in the Bahamas. It about wiped them out.
Yeah, we're still evaluating that. Yeah, because Grand Bahama and Abaco got hit pretty bad. That was one of the things that Paul DeLuca was working on, was reassessing the fauna on Grand Bahama. Unfortunately, he passed away last year, so we're sort of in a transition of having a resident entomologist in the Bahamas. His wife is now in charge of the collections at the University of Bahamas, so she'll slowly get into sponsoring projects with people.
Thank you.
Can you also tell us how long she was in Sarasota when she came here?
Oh, Jackie?
Yeah.
I think it was 1972 that she started in Sarasota. She started with the Allen Museum that was established in Chicago, moved to Sarasota, the Sarasota Bank Building, and then a building was put together on the grounds or next to the Sarasota Jungle Gardens, because Mr. Allen also owned the gardens at the time. And so she came to, Mr. Allen donated the collections in 1981. And at that time, Jackie and Lee became faculty members of what was then the Florida State Museum. And they moved the museum contents and everything started moving up here in 2004. So it's been a long process.
You ignore me. Just look at that's.
We are almost ready for the second speaker. Also, another thing, whenever we start to talk, at least for the people live here, please stand by here and let a few seconds until Julieta or maybe Alessandro can take a picture of each one of you presenters, so we can put it into a poster at the end. All right, so I think we can move on with the second talk about Paul Masonick, okay, on the revised classification of the assassin bugs, based on combined analysis of phylogenomics and morphological data. Thank you.
Thank you. You see that laser pointer in the back? Maybe I can point to.
And I'm going to be
Okay.
lowering the
Yes.
lights. You ready?
Yeah.
Thank you.
Yeah, can you hear me in the back okay? All right, awesome. Good to go?
Just hiding the controls. There we go.
Okay. Okay, all right, I'm Paul Masonick. I'm actually a new postdoc in Akito Kawahara's lab here at the McGuire Center, and I'll be working on moth evolution, specifically in the bombicoids. However, today I'll be talking about research that I did during my most recent postdoc in Christiana Weirock's Heteroptera and Systematics lab out at the University of California, Riverside. And my talk today is going to be focusing on assassin bug systematics. This work is not yet published. I'm currently revising the manuscript and will be resubmitting it very shortly here. So I'm going to give you a sneak peek as to what's to come. All right, Reduviidae, the assassin bugs. This is one of my favorite clades of animals. There's about 6,800 described species of Reduviids, divided into currently 22 subfamilies, some of which are para or polyphyletic. And this group constitutes one of the largest radiations of predatory animals. There are many taxa that specialize on specific types of arthropods. There are specialists on ants, specialists on termites, and these Etricodiine millipede assassin bugs that specialize on millipedes. And they engage in this really cool behavior where they some species communally feed on millipedes. There is a clade amongst the Reduviids that are actually blood feeders. So these are the kissing bugs, the Triatomini. There's about 150 roughly described species, and these are medically important species, particularly in the New World tropics, where they are important vectors of the causative agent of Chagas disease, that is the Trypanosoma cruzi. So across Reduviidae, there's a great diversity of morphological and coloration traits happening. There's many species that are aposematically colored, brightly colored, and you can see them from a great distance away in the field. There's many other species that are very cryptically colored as well and difficult to see. And some even engage in this really cool crypsis behavior of piling corpses of dead insects on their backs to help them camouflage. There's a few other lineages that are actually mimics of pompilids and others that are mimics. This up here in the top right. Oh, I'm sorry. Oops. Yeah. right there, that are mimics of mutilids. And then, of course, there's a great diversity of raptorial forelegs that have evolved across assassin bugs. And my favorite example are the ambush bugs, the Thymotynae. This is a group that I did my PhD research on, actually. So I'm particularly biased when it comes to these cute little critters. So there's been many lower level taxonomic studies that have been done over the last several decades that are focused on subfamilies and relationships among those within those subfamilies, tribal level, species level relationships. And some of those have resulted in substantial taxonomic revisions. There's other groups that still require taxonomic revision as well, which I'll be getting to here. There's been a few very broad family-wide analyses based on molecular data. Some of these are dating back to the late 2000s, early 2010s. And these studies worked with specifically with Sanger sequencing data. In the 2010s, we moved into using transcriptomic data to try to resolve the relationships among assassin bugs. And then most recently have incorporated hybrid enrichment data and whole genome sequencing data to even better resolve those relationships. And one of the issues was trying to figure out exactly kind of the relationships along the backbone. And those were difficult to get, particularly with the Sanger data. But we have a much better resolved tree now. One of the things that is common amongst all of these studies is that they've highlighted that there exists some subfamilial and tribal level revision that needs to be done. So the objectives of my talk today. First, I wanted to conduct a detailed survey of Reduviidae morphology to capture features that are phylogenetically informative and diagnostic for major Reduviidae lineages. And then infer the first combined morphological and molecular phylogenetic hypothesis for the group, and reconstruct robust relationships and identify morphological synapomorphies that are useful for recognizing subfamilies and clades. And then lastly, use these phylogenetic results and the optimization from the character data to propose a revised higher level classification of assassin bugs. All right, so my research really leveraged this large phylogenomic data set that was published last year by Neishoff et al. And I added about 80 or so in-group taxa to that, to the taxon sampling for a total of just under 200 Reduvioids that were sampled. On the molecular side of things, we had an alignment that was about 1.8 megabases long, very large molecular alignment that has data generated via whole genome sequencing, anchored hybrid enrichment, and Sanger. On the morphological side of things, I surveyed the diversity of characters on the head, thorax, wings, and abdomen, and in total scored 112 morphological characters for my matrix. And then together integrated the molecular and morphological data into a combined or total evidence approach to reconstruct the phylogeny. And I used a coalescence-based approach as well as a concatenation-based approach to build my trees. With the preferred topology obtained from the analyses, I was then able to identify traits that are useful for diagnosing certain taxonomic groups through character optimization with Wincleda. And then here I was really kind of keying on important nodes, key nodes here that were representative of entire clades to find characters that would be useful for diagnosing those. So what are the phylogenetics results here? Well, many of the groupings that I recovered in this analysis are consistent with those found in previous molecular analyses. One of the big takeaways is that we find that Reduviidae is divided into two main clades. The Thymotyne clade, I'm sorry, this is really small. The Thymotyne clade up here, which is composed of Thymotynae and then 4 other subfamilies. And then the rest of the Reduviidae are lumped into this really large clade that we've referred to in the past as the higher Reduviidae, but we're now calling them the Trichobothria clade because most of these taxa down here possess multiple trichobothria on the pedestal of the antenna. Whereas with the Phymetine clade, those taxa possess just a single trichobothrium on the pedestal. Another major finding here is that the subfamily Reduviidae is it's one of the largest subfamilies and is highly polyphyletic. So all of the taxa denoted here in red are Reduviidae, and it's obvious just from looking back there. You can't read the names or anything, but just seeing the red kind of dispersed throughout the tree, it's very obvious that there needs to be taxonomic revision focused on the Reduviidae. Oh, thank you. Okay, so what are the taxonomic changes that we're making here? All right, so there's a few really easy ones that we were able to make. We erected 3 new subfamilies of Reduviidae. These were orphan Reduviine clades scattered about the tree here. We've named them Nanocarolinae, Passerinae, and Heteropterae. Heteropinae. Heteropinae. I'm not going to go into a lot of the details here in the interest of time, but I'll just highlight one of the cooler groups that we're describing here. These are the Heteropinae, and they are quite remarkable insects. They're very enigmatic. They're from the Afrotropical region, not really present in many collections. They're found under bark. They're very erratid-like in shape and morphology. They're dorsoventrally flattened, quite small. And one of the really strange things about these insects is that they do not possess a striated poststernal stridulatory group. So almost all Reduviids, this is the key synapomorphia of Reduviidae, is that they possess a poststernal stridulatory groove, which they can use to produce sound used by raking the tip of their rostrum across that groove. The Heteropinae do not have a striated groove. Another one of the Reduviine orphan groups that we decided to elevate to subfamilial status are the Syrphidae. And these are easily recognized by the presence of a very elongated first visible labial segment. Okay, what else did we do? So with the subfamily Thermophoridae, we decided this subfamily is kind of obscure and enigmatic. It's they're not really collected. There are just currently just 2 genera recognized within the Thermophoridae, so including Thermophorus here. We decided to expand the concept of the Thermophoridae to include the Epiroterines, which is another subfamily, and also include 2 orphan genera of the polyphyletic Reduviidae. There's 2 really good diagnostic characters that I'll highlight here. One is the really tuberculate cuticle that's found on most of the Thermophorine taxa. And then they also have these really nice elongated subrectangular posterior cells in the forewing. And there's a long emanating vein that sticks out towards the wing membrane here in most taxa as well. Okay, what did we do with the rest of the Reduviidae? Well, we decided to synonymize a bunch of other subfamilies with the remaining Reduviidae. Some of these subfamilies, if you're particularly, if you know really just like Nearctic assassin bugs, these names might not be familiar to you. But regardless, many of these subfamilies we decided just to demote to the level of tribe. So they still exist just at a lower ranking. And now we're recognizing 9 of these tribes within the Reduviidae. And here's a picture of what some of those taxa look like. Reduviidae in general, these are really large, robust assassin bugs, often very beautifully colored. The largest species of assassin bugs is Sitella horrida, which is a very spiny species from West Africa. Very imposing looking. Okay, now the last taxonomic change that I'll talk about here is the probably the most arbitrary and difficult one that we had to make, and this is what to do with the monophyletic, triotomites, the kissing bugs here, those blood feeders. And we have this polyphyletic braid of random Reduviine taxa, and how to rectify that situation. So we could have either, for each of these Reduviine lineages, designated those as a subfamily. But they're very difficult to diagnose on their own, so rather than make that issue, we are just lumping these all together in to revise sense of the kissing bugs, Trichomonas sensu, because we have good diagnostic characters for that entire group. Okay, in closing, we're now recognizing 19 subfamilies of Reduviids, including 40 tribes. And if I have any time left, I'd like to field some questions. Thank you.
Any questions? Paul? I have one.
Yes.
So I saw that you use morphological data as well.
Yes.
Do you think there is some sort of differences in the nomenclature or terminology used for different parts of different subfamilies or taxa? Or is it has been homologous?
Within Reduviidae, it's pretty consistent.
Okay.
Yeah.
Okay.
Yeah, but
That's
as soon as you get out of Reduviidae and into other, even Simicomorphan groups and other heteropterans, the terminology can vary a little bit, but within Reduviidae, it's fairly consistent, except for Phylogenetics. There's some very specific terminology there that varies a little bit, but.
Question.
Yes,
In those 19 subfamilies, you've got how many of those are in the Arctic?
in the Arctic, there are about 8 or so, I think, 7 or 8 that are found in the Arctic. Yeah, most Reduviids, the greatest diversity of Reduviidae is found in the tropical regions around the world. Yes,
Okay, so I've seen Christiana give talks on the wild polyphyly.
yes,
And Phylogenics.
yes, that's, we've known about for a long
Going
time.
back like 15 years.
Right.
It's interesting to see how long and how many other phylogenies it's taken to get even like this talk.
Yes.
When do you actually believe that this will be realized in a paper? Is it in process, or do you think? this is-
I've written the manuscript and already, I have reviewer comments on it, so it will be,
Okay, gotcha.
yeah, it will be published soon, hopefully.
Just it's
Yes.
interesting to see how long these journeys take from the time they
Yes.
know it to the time that
Yeah.
everyone else gets to know it.
Right, and it's not perfect, what we're putting forth is not perfect, but they're working hypotheses that other people can, yeah. Jump in on it.
It's on the Zellurine,
Sorry, which one?
the Zellurine.
Yeah,
Sinking.
yeah,
That was
yeah,
a
that's,
weird adjustment in my collection.
yeah, I'm sorry about that, but that one, yeah, again, that's the most difficult and arbitrary decision that we had to make.
A small question.
Yes,
How are you connecting with Navidae? What is their?
so navids are actually more closely related to so the sister group of Reduviidae are the Taxonomy. Navids are it's another family of Simicomorphans, but they're not too closely related.
Any other questions? No questions?
Okay,
Thank you,
thank you very much.
Right. We're gonna jump on the third one. I invite on the stage James Hayden for the systematics of Glaphyriinae, the cold crambids.
Take a quick picture. Thank you.
Hey, good morning, everyone. Thank you. My name is James Hayden. I work for the Florida Department of Agriculture, Lepidoptera. And I'm going to talk about this enlarged subfamily with some kind of confused but internal classification that should not really be as confused as people think it is. I know this is kind of a lame name, the coal crambids. This is basically a radiation on brassicales. It doesn't really have a common name, so I just made something up. Please suggest better ideas. I mean, even something in a different language, I would welcome. So first of all, let me acknowledge the collections that have supported this by loans of material, the Australian National Insect Collection, the USNM, the State Museum in Stuttgart, and also the local FSCA and MGCL. I also want to thank Jacob Beth at the University of Florida for making some dissections and sorting material. So if you know anything about Crambidae, this is the Pyraloidea, includes 2 families, Pyraloidea and Crambidae. If you know anything about crambid classification, this is kind of the structure of the subfamilies that dominated in the late 20th century up until 2012. And so I'm going to talk about these, let's see, do we have there we go. Back, here we go. These highlighted subfamilies. In 2012, we published a Phylogeny, Regier et al., a number of us published the first big molecular phylogeny of the Pyraloidea. And we found that a number of these things went together, these subfamilies. So we included 3 at the time. And it was interesting. Many of these things, they didn't have a single morphological synapomorphy in common, but they all have partly or entirely feed on Brassicales. Brassicales is a superfamily that produces mustard oil plants by the glucosinolate myrosinase pathway as an insect feeding deterrence. So, okay, so we've accepted that really without any controversy, and I'll show you here some examples. This group includes a lot of pests, things that feed on cabbages and broccoli, and that's why nobody really cares about these pests. So it's kind of a lower priority. Nobody's really done an intensive study of them, but we've made some progress a bit. Theo Leger in Berlin a few years ago published an expanded classification of analysis of the crambidae, including all of the subfamilies that were then recognized, that are raised a few things, sank a few things. But this is kind of this result that we still get. This blue over here, this whole clade is Glaphyriinae sensu lato. We just expanded the Glaphyriinae to accommodate all these other subfamilies. Pretty much every time you find a crambid that's oligophagus on Brassicales, it's going to fall on this clade. This Lanostinae I'll turn toward, which is an unstable, I'll turn toward at the end. Nevertheless, we still can't find a good morphological snake for them. But each of these subordinate clades still includes really good morphological characters that are easily diagnosable. And I want to reiterate them, test them and reiterate them so that you can just identify them, because there has been a bit of confusion. So I threw together this data set after Leanna asked for talks, and this is why it's really poorly supported. But I've been really wanting to do this for a while because these are good characters here. In unconstrained analysis, a couple of the outgroups turn up in the in-group, these here. So I did what I don't want to do. This is not publishable, but I constrained it for in-group monophyly, and I get pretty much the same topology, pretty much the same thing. The diagnoses that I'm going to talk about in the next few minutes are pretty robust, regardless of the internal relationships. So don't get hung up on the internal relationships on these clades. Just look at what I'm seeing. What were subfamilies and what should be maybe revised as tribes or subtribes. I will go through 4 of them really quickly that are more familiar to you before getting into some more interesting things. So the Glaphyriinae sensu stricto, in the old sense, are these cute little colorful moths. It's mainly a New World radiation. Some of them feed on Brassicales, but others, the larvae go on to other weird stuff like cactus flowers and parasitoids, Pallistine nests, feeding on lichens and odd things. The genitalia have the gnathos is absent, pretty much highly reduced, big plicate valvae. The this thing down here is this deep U-shaped thing with little claws on the end. The females are very, very characteristic with this big, the corpus bursae, instead of being a shapeless bag, has this big striations and spines and this big diverticulum. I mean, this is really weird. Now, sister to that is this one genus called Noorda, which is this has been found this is borne out by both the molecular results and the morphology. This is an old world pest on Moringa, the drumstick tree in Asia and in Africa. Pivotal organs are modified. They have the males have these funny sclerites on the vinculum up here, but they have these plicate valvae, which they share with Glaphyriinae sensu stricto. So that's one genus. Evergestines, this group includes several genera, many, probably hundreds of species. It's mainly a whole Arctic radiation with some tropical representatives. They have this long uncus and gnathos with little hairs and stuff on the end. The females have a pair of signa, 2 lozenge-shaped signa in the corpus bursae. I mean, this is really, this should not be controversial, what belongs to this group. Here's a couple of tropical representatives. This is an old world pest, an Asian pest on cabbages in China. Then we have this single thing called Glaphyriinae, which is an alpine thing, 1 or 2 species in the European Alps. The molecules put them together in this group, even though they feed on alpine forbs that are not brassicaceous. There's not really much to say about their morphology generally. A bit reduced. So I'm going to go on to some more interesting things. I just talked about that, and then I'm going to talk about these 2 groups here. This Glaphyriinae, the Cybolomiinae and Linostoinae. So there's a lot of confusion about what Cybolomiinae are. A lot of weird stuff has been dumped in this group as kind of a dustbin. If you want to know, ask the Europeans or people in West Asia, where this group is most diverse. They're little brown moths with very characteristic genitalia. The representatives, what's placed in the Cybolomiinae in other continents, don't pay attention to that. They have really narrow valvae, this little membranous finger on the end. The females have this big corpus bursae with this big signal, one big signum. Uncus and Nathos kind of look like that. So it's actually a pretty boring group. There's one representative in North America, which should be moved back into it. Now, this is what really got me into this talk, what I really wanted to talk about. This is Linostoinae. Linostoinae is one genus of a couple, 2 or 3 species in the neotropics that was a kind of a rogue, bounced around in this group. It's either sister to this whole clade or goes out there. So if we want to improve the molecular sampling, what we really should do is think about, ask what other genera might be related to Linostoinae. So we could add to that. We don't have to go any further than the original proposal of the subfamily by Eugene Monroe. He actually pointed out that they're closely related to Cybolomiines, and particularly this thing called Thyrodifera. And he cited a couple of interesting morphological synapomorphies. But then he said, well, the mouth parts of the tongue is absent, and so therefore, woohoo, new subfamily. Well, this is the 1950s. We don't do that anymore. So I would like to propose that Linostoinae should be expanded to include a bunch of other tropical stuff in taxa, in Madagascar, Australia, the neotropics. All of these things have really good synapomorphies that you can even code these from the literature. Okay, it's just a matter of sitting down with the books and reading them. They have this 2 veins up here, R5 and RS4 and M1 are stalked, often with a fovea. The nathos of the male genitalia is huge, has this great big base. Sometimes with this pyramid-shaped tetrahedral kind of thing going on. It's really strange and unique. The males have shortened hind legs. That's odd. And they're all internal borers, not just on brassicales, they're internal borers in Caparidaceae, as far as where we have knowledge. The largest pyraloids in the Australian fauna belong to this group. I think they should belong to this group. They're currently misplaced in Middleinae. Middleines and neotropical borers in Eraceae, totally monocots, totally unrelated. They've been placed in there, they've been put together for just based on the modification of the tympanoid organ, but the weight of evidence places them more, in my findings, they're more closely related to Linostoinae and so forth. This was actually pointed out by, not long before, by Ian Common, who really knew his stuff. He had placed them in Cybolomiinae back in 1990, and I don't know why people are ignoring that. So a lot of what I'm saying is not new. Many of these large things have been placed in Cybolomiinae in the past. I'll just go back, and I've been bouncing between these 2 phrases here. So the results are that, maybe we could combine them into one big Cybolomiinae sensilato, or maybe 2 separate clades, Cybolomiinae. This is a Linostoinae, whichever, The point is to get some of these things into the data set. Yes. Okay. Typical organs are very typical in this group, and I think this is something that people, these are the ears that they get these things that used to hear. And they have this big capacious posterior part right here. So just to briefly review, genitalia, a lot of these things are really, my data set is heavily based on dissections. Glaphyriinae look like this, Evergestines look like that, Cybolomiinae look like that. Norda has this these big plicate valvae. You don't really have to, this should be very simple. So, okay, this is to reiterate what I just showed before, a couple minutes ago. And now I'm going to go on to two more little groups that are kind of interesting, that are oddly placed. One of them is this. Hydropeonia is a neotropical genus, one genus that ranges up into Arizona. We know that they have been reared boring into like the bark of something called Capparis uniflora in Central America. They're kind of an odd mix of characters. The typical organs look like those of Dichogamma over in the Glaphyriinae with these big cavities here, and this extra pair of cavities down here, these petiole. Females don't really look like any kind of Glaphyriinae, so they kind of bounce around in the group. This, however, is taxonomically a bit more interesting because they have Plantigumia is an obscure genus from the Caribbean, little brown moths, but should be expanded to include a few other things in the neotropics. Some are misplaced, some are undescribed, and others are misplaced at the end of the last neotropical checklist, misplaced without even a genus, there. But if you dissect them, they can be easily accommodated within Plantigumia because they all have tails that look like that, with this big long fibula sticking out here. Apex of the uncus is kind of indented. Something that was really satisfying for me, though, was placing this critter here. Eustyxia pupula is widespread in eastern North America. This little oddity. The caterpillars feed on Lepidium, pepper grass. And Monroe, late world's expert, had transferred them into Glaphyriinae, but years ago, but because he felt that they were more at home in the Glaphyriinae, quote unquote, but they don't really fit with traditional Glaphyriines. But I found a couple of morphological synapomorphies, I think, that unite them with Plantigumia. There's a little CD down here, tufts of CD, and a couple of this uncus is indented. Not much, but the evidence is the synapomorphies seem to be unique. So that's pretty exciting. So finally, to wrap up, this is what we should do going forward. We should reinstate these groups as tribes and formally, reiterate the diagnoses. Clarify the membership of Cybelomiinae and Ledostinae. Improve the taxon sampling of all these. Revise a couple of these genera and try to get some DNA out of them. We have tried. Sequencing hasn't worked for the samples that I've supplied, but we should try again. And finally, also this is beyond the scope of this talk, but the Glaphyriinae sensu stricto, I mentioned some of the interesting post-evolution in that group, should receive some more in-depth study because there's a lot of weird stuff going on with that. So I'm done. Do we have any time for questions? Thank you.
So,
thanks.
Do we have any questions? Yes.
A small one. We grow moringa in Gainesville.
Mhm.
So should we be looking for an introduction?
Well, first question is, what's the regulatory status of moringa? Is that legal?
Loud.
Yeah, I know people do it. I wouldn't worry about it. I don't really have the NORDA moving because yeah, that's the question about our interception records. I don't know. I really doubt it, though. I haven't really heard of it as a pest that's moving around.
We get stuff on moringa. I'm trying to think left-wise paralysis.
Well, I don't know what my relatives. Being very honest.
There was a ketotaxic description of these published a couple of years ago, more of NORDA specifically. But the larvae of this group just don't really have many good ketotaxic characters. I've just gone on and a lot about like really awesome characters in the adults, but no, the larvae, not much is published and they're not really falling into discrete groups.
I have a question. Has anybody done anything about the muscles of the male genitalia?
Yeah, stuff has been published, yes. Here and there, but not enough.
Okay, because that
would be really good for figuring out homologies.
Right.
I've mentioned this structure called the nathos.
Mhm.
For a good time, there is something called the pseudonathos, which is like it's a nathos, but it's fused to the tegumen. It's not articulated. That was, they're synonymous. There's the same structure, just fused or not. And I think that was corroborated by musculature work.
Fantastic.
Yes.
Is
Many studies been done on the mating of any of these corrals.
Have any studies of mating? I don't think so, not to my knowledge.
There was a book that came out in 2015, Feeding of Choice, edited by Bill Everhart, but the Carlos Cordero had a chapter on Lepidoptera, and he illustrated some diversity with really complex structure. Some of them kind of resemble some of the ones you showed.
Yeah, questions about the structure of the bursae, these signa that are very characteristic for each clade. I can't think of anybody who's actually studied them specifically for this in terms of how they function. One question.
Well, yeah. I guess the synapomorphy is the enzymes that they use to detoxify the.
Presumably, and yes, and that's a very big question. I looked and there have been recent reviews of studies of insects that, how do they detoxify the system? How do they, as they say, the pathway is called colloquially the mustard oil bomb. When an insect chews on the plant, these two chemicals come in contact and produce isothiocyanines. How do you diffuse the mustard oil bomb? I don't think anybody has actually studied that in this group, how any of these things do it. It's really surprising considering how diverse they are. Thank you, thanks.
So now we have on Zoom Suk Oh on the molecular phylogeny of Miridae with discussion on its subordinate group classification. I've got a pre-recorded.
Yes, so this is a pre-recorded talk, but is the presenter also on Zoom? Will be fielding questions afterwards?
Yes.
I think so, yes,
Okay.
yes.
Great. Just give me a second. I want to start sharing the audio this time.
Just a reminder to keep your cell phones on vibrate.
Good morning, my name is Suk Oh. Today, I will talk about molecular phylogeny of Miridae with discussion on its subordinate group classification. Miridae is one of the family that comprises Systemicomorpha. In recent classification, this group includes 7 or 8 subfamilies. This family is well known as the most species rich in Heteroptera, more than 11,700 species. Their exceptional diversity is even comparable with other higher Heteroptera groups. In this pie chart, we can see 1/4 of Heteroptera belonging to Miridae. As in their high morphological diversity, Miridae shows diverse habitat and diet preference. This group possesses fungivorous, phytophagous, and predaceous, and sometimes liveous polyphagy. Large number of species can be collected with a light trap. To this day, a total of 7 subfamilies and 45 tribes are recognized. Even after this recent taxonomic arrangement, many phylogenetic studies are continuing to clarify its classification system. Most Mirids can be recognized by 1 to 15 mm size, absence of ocelli, 4 segmented antenna, presence of cuneus, and diverse body shape and coloration. Also, pretarsal structure and genitalia are regarded as key characters. Due to their hyperdiverse and even confusing morphological characteristics, researchers try to define the phylogenetic relationship of Miridae. These 4 cladograms briefly show morphology-based analysis research on Miridae. Largely, morphological analysis implies Isometopinae as a basal group, and the close relationship between Phyllinae, Orthochyllinae, and Miridae Deraiochorinae, and sometimes Bryochorinae and Scylapinae are regarded as non-monophyletic groups, and Scylopinae or Discipinae regarded as distinctive subfamilies displaced from Bryochorinae or Scylapinae. On the other hand, recent molecular-based studies do not fit well with a morphology-based cladogram. Some distinct differences in topology are denied vessel group position of Isometopinae and relationship between Orthocline, Pyrinae, and Deraiocorinae, Miridae. Moreover, monophyly of some subfamilies were not supported. Among these previous works, Xu et al. Focused on Mycomorpha, so their discussion did not address subordinate groups of Miridae in detail. Shortage of Zhang and Li was in the taxon dataset. They represent Bryocorinae, Dilapinae, and Isometopinae with a single genus or few species with 4 ordinary markers. Compared with this, our study doubles the dataset size, 204 species with 7 subfamilies, 37 tribes, and 188 in-group species. Also, 2 additional protein coding genes, histone 2 alpha and histone 3 alpha, were used for analysis. With this dataset, we conducted maximum likelihood, Bayesian inference, and maximum parsimony with following programs. Based on these efforts, we could get the following results. Among our 3 analysis results, BI tree was selected as the main tree. When simplifying this cladogram with merging clades to tribe level, some distinct tendencies can be observed. At first, Phyloidea, suggested by Xu et al. Was well supported, and the sister group confirmed as Stingily. Miridae was revealed as strict monophyly in all analysis. However, in maximum parsimony analysis, Stingily displaced from Phyloidea and mixed with other semicomorphan groups. But this displacement recovered its monophyly in implied weight analysis in TNT. Among the in-group of Miridae, part of bryocorine tribes like Monallonini and Discipini construct the early divergent clade in Miridae in-group, and Isometopina was not placed as the earliest diverged group of Miridae, as in recent molecular phylogenetic analysis of Xu and Zhang and Li. Bryocorinae fragmented into several clades, and its monophyly did not support it in all analysis. Some tribes like Discipini and Eccritotarsini were found as non-monophyletic tribes, and some differences were observed at tarsal structure between separated clades. However, due to lack of species and supporting values, it is hard to reclassify each separated clade to valid taxon. Similarly, Stylapinae also shows unstable topology, shown as 5 respective clades. Due to unstable topology between analysis to analysis and insufficient supporting value, we could not ascertain the actual position of each node. However, it is obvious that Bryocorinae and Stylapinae need to be regarded as non-monophyletic groups. Otherly, Corydromini and Dermatopilini were displaced from their original subfamily Deraiocorinae and Orthocline, which they imply these 2 groups need new taxonomic status. However, we did not merge these 2 tribes as a single group because their biology and external characters showed large differences. When we see the lower side of the cladogram, 4 subfamilies, Orthocline, Pyrinae, Deraiocorinae, and Miridae are represented by 4 separate clades. In each clade, distinct non-monophyly was observed at clades for some tribes, such as Deraiocorinae, Miridae, Orthocline, and Pyrinae. These tribes need thorough reclassification because some species groups are classified only relying on suspicious diagnostic characters. Also, our research reconfirmed the sister group relationship between Orthocline, Pyrinae, and Deraiocorinae, Miridae. These results were recently denied by phylogenetic work of Zhang and Li, but recovered by subsequent work of Menard et al. At 2014 and Kim and Jung at 2019. As this recent result, our cladogram recovered Orthocline and Pyrinae sister group relationship and the sister group relationship between the major clade of Pyraloidea and Miridae in all analysis. In Pyrinae, Menard et al. Recently reconstructed the phylogenetic relationship using total evidence. Our results largely follow this, but some differences occur in the placement of Decomini and Nasocorini. Malaysia Miris and Casmiphyllus were displaced from their respective original tribe clades Decomini and Nasocorini, and it seems that this genera need their location based on subsequent research. Pyraloidea construct major clade except Dermatopyrini, an important discussion point of this clade is the paraphyly of Pyraloidea. Ant-like habitus Angelianus and Fingulus displaced from other Pyraloidea, and it came at us recent phylogenetic work for Pyraloidea. They suggest the real location of Fingulus to Myrmecomorph tribe Surinamellini. But precise relationship between Pyraloidea and Surinamellini is still in vague, and for that, to conclude the actual position of Angelianus and Fingulus, further research is necessary. Miridae shows strict monophyly, but the taxonomic position of Miridae was highly unstable. Among the clades of polyphyletic Miridae clades, the Lygos complex shows stable position and supporting value in all analyses. As in Kim's and Jung's recent work, Methysocellini fused into Stenodemini, and one of the interesting points was Hylopepilini. We used two Hylopepilini species, Hylopepilus similis and Cosmiomiris sp. These two species did not construct a single clade and placed at clearly separated position. This may imply Hylopepilini needs thorough re-examination on its identity. In summary, we reconstructed the cladogram of Miridae with more comprehensive dataset. As a result, 4 subfamilies were revealed as non-monophyletic groups. Also, we reviewed the sister group relationship of some closely allied subfamilies. However, due to lack of species and supporting values, we could not provide alternative classification which can cover all phylogenetic problems, but implies the need for more comprehensive research into unstable groups. For example, the actual placement of Corydromini and Dermatopilini is still vague. Also, tentatively non-monophyletic subfamilies like Bryocorinae and Xylapinae have frequently been considered as monophyletic in recent phylogenetic works, but our work suggests that those presumptions will require additional reassessment. This is the end of my presentation. Thank you for listening. Thank you.
Okay, we have the presenter online. If someone wants to ask some questions, oops. I have a question. Can you hear me? How long has been all this work put together for how many years? If years?
Actually, I started this work from 2019, early 2019, and this work was published at July 2023, so nearly 4 to 5 years, 4 years to take all this work, compile and finalize.
Fantastic.
Thank you.
Great work. Any questions? Right. Thank you so much. So next one in line before the break is a researcher from Italy, a friend of mine, Daniele Sommaggio, is going to present live about the old data, new information, the Syrphidae of Italy project.
Okay, good morning to everybody. Do you hear me?
Yes.
Yes, we have you.
Okay,
you
thank you
have-
very much.
uh,
And you,
Do you have slides you're going
Can
to? Excellent.
can you see my screen?
Yes.
Okay. So thank you very much to the organizer to give me the opportunity to be here with you online, unfortunately. And just a second, sorry. Okay. Now, sorry for my terrible English, and sorry for if my presentation is quite local because it's focused on Italy, but I hope that some idea can be useful for also for not only for Italian people. Now, the focus taxon of my presentation are overflies, or better flower flies. And these are very wonderful creatures and very nice. And despite the fact that they are Diptera, and they attract a lot of attention also by amateur during the years, and a lot of research also thanks to the many ecological services that they provide. For example, pollination, very important recently, and also pest biological control. Now, for all these reasons, the research of overfly in Europe, in worldwide, but in particular in Europe, received a lot of attention. And here we have a publication of last year, and in which we put the checklist of all the European countries. Now, also in Italy, we have a lot of research because Italy has the second richest number of species record with 538 species. But the story of research in overflies in our country was undergone two different periods, well separated one from the other. The first period was dominated by two giants in the research of Diptera, which were Camillo Rondani and Mario Bezzi. They not only studied overflies, but they studied largely Diptera, and not only, of course. Just to give you an idea, Rondani described 1,150 nomenclature species in Diptera and many other also in other order. Here is an epitaphium on a monument dedicated to Camillo Rondani and is called correctly Principle of Diptera. And they have in common several things, but one is very strange because both of them were not professionists in entomology. They were teachers in secondary school. This may seem quite strange if you look at the most important publication of Bezzi in about Diptera fauna in Italy. And here you can see that it covers almost all our country. But this is easy to understand because in that period, teachers were moved all over our country, which reached the unification only 30 years before. And so in his new school, Bezzi started to collect Diptera and publish then an important contribution about Diptera in that area. But the fact that they were not professionists may be also a problem. And this is very evident in the case of Camillo Rondani, because there was a single institution, maybe a university or a museum which hold and all his private collection. And this is particularly critical in the case of Rondani, because in 1863, for reasons that we have not yet understood, he decided to sold his private public collection to different museums in Italy and also to some friend in Europe. So here you can see the place where the types of Camillo rondani species are located. And we can say that they are spread all over Europe. And for this reason, 2 years ago, I and Alessandra Sforzi decided to publish a catalog of Camillo rondani types. This was a very big work because we checked more than 3,000 types. And in green, you can see the types we personally checked. And in red, the types that we received information from the institution. This is a publication of 350 pages, just to say where are the type of rondani species. And just a small curiosity, during the publication of this research, I and Alessandra have one thing in common with Rondani and Betsy, because both of us was teacher in secondary school, and so we realized this publication in our free time. So the study of old collection and the study of also of old publication help us to solve some problem about the identity of species described by these very important dipterologists. I give you just one example, which is Paragus coadonatus. Rondani introduced this new name because he found one male of Paragus amorous and a female of Paragus obscurus coupling together. And so he correctly said that this should not be 2 separate species, but only one. And incorrectly, he decided not to use one of these 2 names, but to introduce a new name, which is Paragus coadonatus. Coadonatus in Latin means unified. Okay. Now, this may seem very strange for us today, but we should remember that in this period, the principle of priority was not mandatory. So Rondani decided to introduce a new name for 2 species that yet exist. And later, fortunately, later on, in his wonderful publication about Paragus, the genus Paragus in Europe, Jacqueline de Tifano examined the collection in Florence, and he found 6 specimens under the name Paragus coadonatus, and 2 of these from Malta have male genitalia different from Paragus amorous. So he decided to use the name Paragus coadonatus for this new species, but this was not correct, since Paragus coadonatus was not a new species, but only a new name. And this decision created a lot of confusion, because here, for example, is the distribution map in the Mediterranean area of all the records as Paragus coadonatus. And you can see we have some records from North Italy, but these are records of Mario Bezzi, who used the name in sensu rondani, which means Paragus coadonatus is Paragus amorous. And down, you can see in the south is records of Paragus coadonatus sensu Jacqueline de Tifano. So for this reason, we decide to consider Paragus coadonatus sensu Jacqueline de Tifano as a yet described new species, which is Paragus mundus. And similar things happen also for other species. This is, for example, Merodon italicus. It's the same thing. Okay, we have two species, a male of Merodon melancholicus and a female of Merodon natans, which Rondani found in coupling, and so he decided to use a new name, Merodon italicus. Unfortunately, in the collection of Rondani, there are some males of identified as Merodon italicus, which is different, something different, which is Merodon longicornis. And this name is used as a senior synonym of Merodon longicornis, also in the recent European red list of overflies. So these are only two examples. You the idea that for the research of overflies in Italy, it's very important to collect information from, of course, old collection, but also from the original publication. And this information need to integrate the one with the other. But a piece of a puzzle is still missing, and it's the new research. What happened after Rondani and Betsy? For more than 70 years, we have in Italy no specialist on overflies. And so the research in Italy, unfortunately, dropped down to only few sporadic records made by or other entomologists, specialists in other groups, or from foreign people which come in Italy to collect. But fortunately, starting from Mauro D'Accordi, and more recently in the last decades, a group of syrphidologists are now working in Italy. And also thanks to the change sensibility in political, on the political side, we have the opportunity to improve our research about overflies in Italy. Here, for example, is a map with the active monitoring schemes from my university and in collaboration with the Bologna University. And so you can see we have quite a good coverage of most of our country. And the new research help us also to shed light on past species. This is the case, for example, of Pilosia atriceta. Here you can see a female of this species, probably collected in the Dolomiti Bellunesi National Park 4 years ago in a male strap. This was the single female collected in this area, which fit very well with the description of Pilosia atriceta made by Oldenburg 100 years ago. The problem is that Pilosia atriceta was described on the holotype, and no other specimen has been collected after the description of Oldenburg. And so today we have only two specimens of this particular Pilosia, and both of them are from northeastern of Italy, very near the one with the other. And the fact that we have no additional specimen of Pilosia atriceta put several authors to consider Pilosia atriceta as a doubtful species, maybe a teratological condition, and so on. And thanks to the new specimen collect, now we are studying the, also from a molecular point of view, the identity of Pilosia atriceta, which is unfortunately the type species of a subgenus Teniopilosia. So if it's really a teratological condition, some important nomenclature change will happen. Finally, we have a lot of data today. We have data from old collection, which we are checking. We have data from the check also of publication and also data from new research. And my idea was to put all this information in a database which make available all this information for everybody interested in overflies. And thanks to the help of Davide, I found a very good opportunity in taxon work, because they opened a new project, Syrphidae of Italy, where the idea is to put inside all the information about overflies. This is some page, the over of our project. And as you can see, this has been updated until a few hours ago. And I have not yet completed the introduction of all species, but I'm not so distant to complete this work. And you can see also the large number of scientific names necessary to introduce these 400 species and the citation and so on. And what is very good for me in Syrphidae of Italy, in taxon work, is that the taxonomic approach is very precise and very correct. And so it's not only a database which gives you the opportunity to have distribution, but also to build the taxonomic history of each single species. Here are just two examples. This is Crysotoxum cisalpinum. Here you have the distribution map, the new records in the red, and also the type records in blue. The photo of some specimen, which can give you idea about the look of the species, and also some important features to the identification of this wonderful Crysotoxum species. And also the repository of species, of types, and also the nomenclature history of the species, which in the case of Crysotoxum cisalpinum is very simple, but if you go to Paragus mundus, it's very complicated. And here you have also the Paragus coadonatus sensu Gödelin de Tifano. When you type in the system coadonatus, he asks you if you want to go to Paragus coadonatus sensu Rondani or to Paragus coadonatus sensu Gödelin de Tifano. And in the second case, you have a distribution map, which is really much more correct, not a map where this species is present in the North Italy. So I hope that this project can increase in future more and more information about taxonomy, distribution, but also biology. And I hope that Syrphidae of Italy can become a useful tool for everybody interested to study these wonderful creatures in our wonderful country. So thank you very much for your attention.
Grazie, Daniele. We have time for one question. So how long did it take you to compile that taxonomic paper on the Rondani collection, Daniele?
15 years, a long time. And what is very nice is that in all this research, we have received no financial support. So everything has been done by me and Alessandra only for the love of Camillo Rondani, which has been a very great entomologist.
Thank you, Daniele. And now there is time for 15 minutes coffee break. So we're going to reconvene here around 10:45, please. 10 minutes. Yes.
Question. Could you put
Adam Wallner, on the phylogenetics and taxonomy of the edible stinkbugs genus Ascra and its two species group, bifida and privata. Please, Adam, that's yours.
Thank you. And I hope I'm audible.
Yeah, yes, we can hear you.
I don't know how to switch on I'm a Luddite by nature, so I don't know how to switch on that video, but it's probably best that you don't see my face. So I did notice that there was a slight typo in the title of my talk. I forgot to use the adjective preliminary, because this is a preliminary phylogenetic analysis of the edible stink bug genus Ascra. And it's to species groups bifida and privata. And I would also like to acknowledge my Brazilian collaborators who, of course, without their help, this talk would not have been possible. And so, get on with the show. So, Ascra is one genus out of 18 currently described genera within a stink bug subfamily called Edessinae. It's a monophyletic group thus far. And as you can see in these representations, they come in a variety of shapes and sizes and ornamentations. And within that diversity, there's several diagnostic features or several synapomorphies that unite them. And as many of you are aware of the group, one of those is this metasternal process on the ventral surface of the thorax. And sometimes it's bifurcating, sometimes it's a spine, and sometimes the process can be a combination of these two states. The diversity of this subfamily is strictly the New World, extends from the United States all the way down to the Tierra del Fuego in Argentina. But most of that diversity is housed in Central America, Mexico, and South America. And interestingly, anecdotally, there are a few groups that have cultural importance that are actually consumed in large quantities. And my USDA colleague, John Lemongood, knows that they are commonly intercepted at US ports of entry. And one of those genera is Ascra. And I hope this works. I hope the sound works. Can you hear it? So just put it down a little bit. So these bugs are harvested in bags and baskets and from their host trees. They're actually eaten alive. And they're sometimes cooked with chiles or tortillas. Personally, I have never eaten them myself. But in addition to what they bring to the culinary world, there's also quite a bit of work that has been done on their distribution and diversity. And one of the main authors is Santos et al., who did a paper in 2015 and found that what elevated Ascra from a subgenus to a genus, changed the status, and hypothesized that there were 7 species.
Adam, you just muted yourself.
Oh, can you hear me now?
Yes, it's only for a few seconds.
Oh, okay. like I said, my enemy is technology, so bear with me. So these authors recognized 7 species they put in the bifida group, and they also hypothesized another group called privata, and they put an equal number of species, 7 species in that, although that's never been published. And the distribution for this group is mainly in Central America, specifically Mexico. And of course, there's one species, Ascra bifida, which we'll get to later, that has a range of 13 countries. Oh, it's not. There we go. Okay. These authors also found several characteristics that unite these 14 species that are diagnostic for the group, for the genus. Some of those are a swollen pronotum, the proctogenitor of the male penis that surpasses the posterior plane, the pygopher, as well as a swollen proctogenitor with a horizontal crest or a carina. Yet, despite this taxonomic work, there wasn't any phylogenetics that was done to test the monophyly of the group. And so in our research, we sought to explore that, to examine whether or not these are Ascra bifida and privata are monophyletic or natural groups, as well as in a process of elucidating its evolution relationships, explore any kind of applications. So in examining the monophyly, we selected Neotibullus as the outgroup and 5 genera as the in-group, including all 14 species of Ascra. And that taxonomic selection was based on communication with Jose Fernandez and scientific literature. We examined 51 specimens, including types and paratypes from 12 institutions, both private and public. And from that material, we had just the amount of morphological characters. We discovered about 50 of them that seemed to have some phylogenetic signal. 23 of those were external. Is there any way I can move this? There's a screen that's cutting off some of the text. I don't know how to get rid of that.
I don't know what text you're referring to, but you can see all the text on our end.
Okay.
So what we're seeing is not necessarily the exact same as what you're seeing on your computer.
Oh, okay. So I found half of those were external features, specifically the pronotum and the abdomen. And the remainder of these, I should say the bulk of them were terminality features, specifically the male and female genitalia, especially the pygopher and the proctogenitor seem to be rich in characters, as well as some of the internal features within the female, like the intima vaginal. And that seemed to be good for separating out Ascra from other related genera. So once these characters, we use Mesquite to assemble the matrix and test the homoplasia and retention of synapomorphies. And then the meat of the project, of course, was to test the monophyly of the group. So we used TNT phylogenetic software. And I just want to preface this by saying this is this is a preliminary analysis. So bear that in mind. And this is the strict consensus tree of the morphology that we currently have. And it's a strict consensus of 4 most parsimonious trees. And there's a few takeaways that I'd like to share with you to deduce what we found. First was that Ascra seems to be moderately supported by Bootstrap and Brenner. And we found some retention of this synapomorphy of the genus itself, which was a swollen procedure as well as that horizontal carina. The second is we did find branch support for the privata group, suggesting that it's a natural group, and detected a few synapomorphies to characterize that group, which was these paramers that are oppressed to the procedure, as well as the pygophora with its ventral surface, half of which is covered with punctures. The bifida group, of course, collapsed. There was no support. The characters are very wobbly, had very high homoplasia, and so we were not confident thus far in the analysis. And there's a series of explanations that may explain why there's no support for bifida. And one of them could be the statistical artifacts within maximum parsimony. As many of you know, some incredible talks before me, maximum parsimony can sometimes get trapped in some optimal tree space or tree islands, so it may not be able to find the most parsimonious tree. Although it did find support, good resolution for the privata group. But I think it'd be worthwhile to examine whether or not bifida's natural group using more computationally intensive methods, probability algorithms like maximum likelihood and Bayesian. So the statistics may not be to blame. It's possible that there could be replete with homoplasias characters. And I did notice that there's a handful that seem to reflect more of adaptation and environment rather than the center modification for the bifida group. So, for example, pigmentation and punctures. But then in some species within bifida and related species, I found that there was a stasis within the male genitalia. It was difficult to tease apart characters and character states within some of these species. Now, I don't know if there's deep homologies underneath those homoplasias characters. And so some of the future work I would like to explore would be to use molecular markers that I think could elucidate relationships at the species level, such as CO1 and CO2 and the cytochrome B gene. Now, there is another explanation that I'd like to share with you. And I think it's the most interesting. I hypothesized that the bifida group is in fact a species complex, a cryptic species complex. And I think the best candidate of that is Ascra bifida. And that's because if you looked at the Santos paper, it is distributed in 13 countries. Now, I did an extensive literature search on anything related to introductions. And I only found one paper which mentioned that bifida was introduced to from Mexico to the United States. That was pretty much it. Every other species of those 14, their distribution was just retained in Mexico or Mexico and a Central American country. So bifida seems to be the outlier. And I just want to state that I'm not familiar with the coalescent models. I understand that Paul has experience, so I'd love to pick his brain. And so I would like to use these coalescent models to test whether or not the bifida group is in fact a cryptic species complex. Because I'd like to explore, as some of you may know, there's these coalescent methods that use these genetic markers and run through models to test if evolutionary lineages are in fact independent. You can do it at the population level and the species level.
Adam, your time is almost up.
Oh, I'm sorry. So I think this this big analysis could benefit not only understanding the genealogy of the bifida group, but also reveal the taxonomy, maybe stabilize it to validate some of these species and provide applications for regulatory agencies by providing valid names and a better understanding of the host plant and countries. And with that, I would conclude and entertain any questions that you may have.
Thank you. May you have time for one question? Yes.
Even though you have not consumed any, do you know what they taste like?
They taste a little salty. I've noticed they taste a little salty.
If I can throw in one more quick question, where would I find them? What plants?
Hungry?
So I've and I don't know if John also may have information on this, since he gets a lot of those interceptions, but I do seem to, when I explore the literature, they do seem to occur on oak trees within Mexico. I don't know if John's found any other host plants.
Morning glories here.
Morning glories? Okay.
All our interceptions that I've received have literally been tubs filled with upwards of tens of thousands of specimens.
Yeah.
Like you took a scoop of nuts at Costco and dumped it in the bucket, and by the time they arrive and they've been by CBP, they're a little slow moving, but they've just been putting alive. So we don't know about how they're rearing it. They're just shipping anything for fresh food or for you to have a starter in a greenhouse. So John, how do you get rid of those? Eat them. I just identify them. I can order a few and eat them.
All right, we should move on. Sorry about this. We can talk about eating those later. Thank you, Adam.
You're welcome.
Adam, please end your screen share before we move on to the next presenter.
Rosanna, if you have some questions, just put them in the chat and maybe Adam can get back to you later. We need to move on. We have another talk from Italy for another friend, Filippo Di Giovanni, about the Italian recipe for a checklist, an updated list of the Italian Darwin ones, which are the Ichneumonidae. I've got a pre-recorded one.
Good morning, everybody. I will share my screen. Here it is. I'm Filippo Di Giovanni. I am a researcher in entomology at the University of Siena in Italy. And today I'm going to briefly outline a project I'm currently carrying on with Davide Dal Pos from the University of Central Florida in Orlando, concerning the checklist of Italian fauna of the so-called Darwin wasps, Hemiptera Ichneumonidae. Well, Ichneumonidae is a family of parasitoid wasps that include about 30,000 described species worldwide. It is believed to be one of the largest family in insects, with an estimated diversity of 60 to 100,000 species. It includes parasitoids of holometabolous insects or spiders, and it is one of the most frequent taxa encountered in collections or in samplings, for example, via Malaysia traps. Well, they have been recently nicknamed Darwin wasps in reference to Darwin's famous letter to Asa Gray, and the group includes species that play a key role in as controllers of harmful pest populations in agriculture and forestry. However, despite their importance, the knowledge about their diversity, geographical distribution, and biology is still severely lacking. There are few specialists to deal with this group. There is a lack of up-to-date checklists and catalogs, and accurate information in the biology of only a few species of economic interest is available. Well, so because of this paradox, we can say, between the enormous diversity in species on one hand and the paucity of knowledge on the other, ichneumonids are counted amongst the so-called dark taxa. The lack of accessible resources, such as checklists or catalogs, coupled with a fragmented and often hard to find literature, is a barrier that discourages many from tackling the study of this group. So why talking about checklists? Checklists are basic tools consisting of a simple list of species, of known species for a given geographical area. They are apparently simple list to compile, but today I want to talk about our experience with the checklist of the Italian fauna and talk to you about some practices that I think should be avoided when compiling a checklist, and suggest others that may be useful in achieving a better result. I want to start with the Italian fauna because Italy is a peculiar reality. There have been no great specialists of ichneumonids in the recent past. Data on the occurrence of species are scattered in agricultural entomology papers, and there are no national museums in Italy, but rather a plethora of small to medium-sized collections hosted by small museums or universities. By the way, Italy was the first country in Europe, as far as I know, to make a complete inventory of all the animal species in its territory. The project of the checklist of Italian fauna took place between 1993 and 1995 and was carried out by Minelli and Ruffo. It entailed the involvement of more than 250 specialists from 15 different countries and the publication of over 100 papers. Mainland Italy was divided into two regions: North Italy, including the Alpine regions, and South Italy, and then the two major islands, Sicily and Sardinia. The Mediterranean also has been divided into three macro areas: the West Mediterranean, South Mediterranean, and East Mediterranean. In order to finish the project within the established time frame, the notes and synonyms have been kept to a minimum. Concerning ichneumonids, the checklist was compiled by Scaramozzino in 1995. It included 150 species, 70% of which have been recorded from North Italy. Fewer than 200 species have been recorded for South Italy and major islands. Although the checklist represented an ambitious masterpiece for their time, some aspects of this work are questionable. First of all, the checklist was published in Italian and printed and in printed format, which led to its very limited diffusion. Also, taxa have been listed in phylogenetic order, so that it becomes necessary to know the systematic classification of the time in order to find a name in the list. And lastly, no references are included. Therefore, it is not possible to ascertain the origin of records and evaluate their validity. The checklist was also converted into a website, but again, also in the website, taxa are listed in phylogenetic order without a search button also. And the site last experienced an update in 2003, after which it was abandoned. Fortunately for ichneumonids, 2012 saw the birth of Taxapad, an interactive digital catalog, which encompasses all the information on the systematic, morphology, biology, and geographic distribution of Braconidae and Ichneumonidae, based on more than 33,000 references. This catalog soon become, and still is, must-have for those who, like me or Davide, are involved with the study of this group. And of course, amongst its functions, Taxapad allows to draw up a list of species for each country based on the known literature. Unfortunately, Taxapad lacks a few publications on agricultural entomology and completely ignore the checklist of 1995. The last update of Taxapad dates back to 2016. After this year, it received no further updates and the project was dismissed. So comparing the checklist of 1995 and the information of Taxapad, we can note a clear discrepancy in the data. Taxapad, though later than the first, reports fewer species for Italy overall, but almost doubled, for example, the number of species for major islands like Sicily. So the picture that emerges is that we need, for a consistent and updated list of ichneumoniid species for the Italian fauna. This is particularly true in Italy, in the situation of Italy, because Italy is an area that has a complex biogeographical history. Orography and latitudinal extension of Italy requires record to be reported by regions or macro areas in order to understand species distributional patterns. Also, each record must be accompanied by a reference so that it can be always verifiable. This is what makes a record scientific, basically, as in science, data must be always questionable. In a checklist, it is not necessary to report the entire nomenclature history of a taxon, as for a catalog, but at least the names under which the taxon has been reported in the papers in which it has been cited. Wishingly, a checklist should be also constantly available and updatable. For all these reasons, together with Davide, we started to make a new checklist of the Italian fauna of ichneumonids, trying to fix all the critical aspects we encountered in the previous versions. So we are making a list of all the species known for Italy, listed by region, and with each record linked to the paper in which it has been reported. For each species, the synonyms and incorrect epithets with which a species has been reported in the cited paper have been included. In this way, for each record, it will be possible to trace it back to the original source, knowing the name with which a species has been reported and possible verify the validity of the record itself. Finally, in order to make the checklist available and constantly updatable, all the work will be eventually transferred to WID, the World Ichneumonic Database, a new database with information on the systematic morphology, biology, and distribution of known ichneumonic species worldwide, which is currently under construction by me and Davide and other colleagues in Taxonwork platform. So to conclude, checklists are one of the fundamental tools to approach a study of a difficult group like such as ichneumonids. Their apparent simplicity of checklists must not distract from some good practice that can make checklists valuable scientific tools for taxonomists. Checklists can boost interest in taxa that are still little known and stimulate sampling efforts in those areas whose diversity is still largely underestimated. And that's it. And thanks for your attention. Thank you.
Thank you, Filippo. Do we have any question for Filippo? Should be online. Is it online? Yes. Are you online, Filippo? Yes.
Yeah, Davide. Can you hear me?
Yes. There is one in the chat. No, not for you. I have a question for you. How far are we to the completion of the checklist?
What, Davide? I didn't hear you.
Completion of the checklist.
How far? Well, I don't know. I think we can manage to finish the checklist soon. We still I think we already overtake the numbers of the previous checklist in these new versions, and so we have more than 2,000 species for sure. But yes, I think still a lot of work need to be done to complete everything.
Roughly 18 pages of references that we need to go through.
I have a question. How did you organize the synonyms? Because the problem with all these checklists is you get a species, it's not a valid name anymore, but that's what you have, and you go to the checklist, but there's nowhere to be found. So is it in the index, or is it listed separately, referring to the valid name, or how do you understand what I'm saying? Yeah.
Did you get it, Filippo?
Just a part of the speech. Can you repeat, Davide, please?
Yes. So he's asking, how can we trace back the name? Let's assume that there is a synonym. How can we trace it back in the checklist so we know what we are referring to?
Oh, well, I hope to answer the question. We checked all the names. I mean, when we found a name, we always check if the name is valid, is correct. We fortunately have the catalog of Taxapad. We already did a great work with all the synonyms and with a history of nomenclature of species. But then in the checklist, we need just to report the name that was used in the reference we are reporting, we are listing. So we don't need a complete nomenclature history because it's great, a huge work. I mean, this is something you do usually for a catalog, but we just need the reader to understand that in that publication at that time, the name the authors used is this one instead of that one. This is the piece of information you need in order to understand who said what in the past, and to have the history of your name. That piece of the history of your name, you need to understand the checklist, what you found, basically. I hope I have answered the question.
Yes, yes. So yeah, we're recording in that reference, what name has been used, and we're placing it underneath. Yeah. Luckily, we have Taxapad, so we can trace it back ourselves because it's becoming complex. And then we're building also the catalog, so it's all come together at some point. 10 years maybe. Okay. Thank you, Filippo.
Thanks again.
We have another Zoom presentation by Anna Namiatova on the total evidence phylogeny of the superfamily Syrphidae and the divergent states of its subgroupings. Here it goes, Anna.
Hello. The title of my presentation today is Total Evidence Phylogeny of the subfamily Syrphidae and the divergent states of its subgroupings. Syrphidae belong to the family Miridae of plant bugs. This group is within 20 largest families and includes more than 11,000 species. Miridae is a very diverse group. Miridae includes 8 subfamilies. Syrphidae is among the smallest groups. It shows affinities with 2 other small groups, Isometopidae and Psallopidae. It is very unlikely that Syrphidae are monophyletic. Its representatives are mostly tropical and subtropical. Syrphidae includes around 70 genera and 500 species. Molecular-based phylogenetic study for this subfamily with all its tribes was not performed before. Syrphidae live in the leaf litter and under the bark. That explains why there are many known fossil taxa for this group. Because of their cryptic habits, it is almost impossible to collect Syrphidae with the entomological net. The special collecting methods are needed to collect this group. On the slide, it is shown how I collected Syrphidae in Australia. In the collections, the species of Syrphidae are usually presented with a single or few specimens. In Syrphidae, there are 5 recognized tribes. Among them, Fulviinae is the largest. Both Fulviinae and Syrphidae are most probably non-monophyletic. Subfamily Psallopidae is also sometimes included within Syrphidae as a tribe. Oh et al. In 2023 provided the phylogeny of Miridae and included 4 tribes of Syrphidae there. Others showed that the subfamily is non-monophyletic. However, the relationships between the tribes are unclear because they included only 1 or 2 genera from Vaniaini, Syrphidae, and Reduviidae. Representatives of Bosphoromirae and Psallopidae were not included into this study. So let's look at the tribes of Syrphidae more closely. First of them is Syrphidae. It is distributed in South Asia, Japan, Australia, and South America. There is a morphological phylogeny which showed that paraphyly of this this group is paraphyletic. And Vaniaini were nested within Syrphidae. Molecular data for Syrphidae exist only for 2 genera. Tribe is characterized by vertical head, the antennae attached above clypeus base, labium relatively short in this group. Its segments are not subdivided. Antennae are often as long as body or longer, and there are no characters which can be considered a strict synapomorphism for this group. Second type is Vanyaini. It is distributed in tropics and subtropics around the world. Its monophyly was shown in the morphology-based phylogeny. Morphology shows that Vanyaini can be nested within Coelophyni. Molecular data exist only for 2 genera. All genera within Vanyaini have characteristic red or brown color pattern. In all other respects, this group indeed is very similar to Coelophyni. However, within Vanyaini, some genera have very different male genitalia structures. The third type is Miridae. It is known from South Asia, Australia, and Japan, and one genus was described from Africa. Monophyly of this group was shown in the morphology-based phylogeny, and molecular data are also present only for 2 genera. Miridae are similar to Coelophyni and Vanyaini morphologically. However, all Miridae are very similar to each other, and they have oval and punctate body with short antennae. The fourth type is Reduviidae. It includes only 4 genera, and it's known only from Africa. This group is likely to be monophyletic, and other molecular data exist only for genus Rhinomidis. Reduviidae are similar with Coelophyni and Vanyaini in long antennae. However, they have a different structure of head, which is sub-horizontal. Antennae are attached near the glabrous base, and some labial segments are subdivided. And the last tribe is the largest, which is Fulviini. It is known from all continents except for Antarctica. Polyphyly of this group was previously shown with morphological and molecular data. In Fulviini, the head is horizontal or sub-horizontal. The antennae is shorter than body and attached below glabrous base, and labial segments 1 and 2 are often subdivided. This group is very diverse morphologically. So, are there any characters which can unite all those different Coelophyna groups? The only candidate for synapomorphy is a structure of pretarsus. As you can see on this slide, Fulviini, Coelophyni, Reduviidae, and Miridae have an identical pretarsus structure, which is different from other Mirid subfamilies. However, in Vanyaini, pretarsus is different from other Coelophyna groups. The same structure of pretarsus as in most Coelophynes is present in Salophyna, which is sometimes placed within Coelophyna. Additionally, Isometophyna also have the same structure of pretarsus. However, representatives of Isometophynes have ocelli, which are absent in all other plant bugs, and it is very unlikely that Isometophynes are nested within Coelophyna. Therefore, the structure of pretarsus is most probably a plesiomorphic state in this case. So, what do we know about the origin of Miridae? According to the previous studies and fossil data, their group originated in the early or middle Jurassic. Right here. According to Oh et al., Fulviini originated in the early Cretaceous. Therefore, the main problems addressed in our study are the monophyly of Coelophyna and its subgroupings, the relationships of Tsilapina tribes with each other and other plant bug groupings, and the origin time of Tsilapina and its tribes. This slide shows methods. We used both morphological and molecular data, and we also performed the total evidence analysis. The results show that Tsilapina are non-monophyletic, Boswellianae are monophyletic, Tsilapinae are non-monophyletic and split into 3 groups. Some Tsilapinae genera form sister group relationships with Boswellianae. Vanniae form a clade with a group of Tsilapinae species called Tsilapus complex. Vanniae are paraphyletic in all molecular-based phylogenies. Position of Rhinomirae is unclear. Vulviae are non-monophyletic and form 2 well-supported clades. The first clade is the largest and includes the most Vulviae genera, and the second clade includes the Australian species only. As in Oh et al., the largest clade of Vulviae is close to Isometapina, but with lower supports, and our study shows that position of Psalapina is unclear. The large clades are distributed in the different continents, originated at the border between Mesozoic and Cenozoic, which can explain their broad distribution because the continents were still at least partly connected at that time. More locally distributed clades originated later in the late Cretaceous or beginning of Paleogene. 2 Australian clades in our phylogeny originated at the same time, which correspond to the Paleogene or first half of Neogene. Australia was the most isolated at that time, and the most probably many native taxa originated during this period of time. And 2 cosmopolitan genera, Pyrithropis and Psalops, originated in Paleogene, beginning of Neogene. The continents were mostly separated at the time periods. This means that at least partly dispersal is responsible for the wide distribution of those 2 genera. Thank you for your attention.
Thank you, Anna. There is any question for Anna?
Hello. Where did you get those really
Hello.
nice reconstructions for the continents through time? I like those images a lot.
Yeah, where I did those pictures. Yes, I found them online. There is a resource. I think it is American resource. I can send you a link if you want. It is free.
Okay, yes, please. Maybe put it in the chat or something.
Yeah, put it in the chat maybe. And then
Okay, I
we'll
will.
do it to Paul.
Yes.
Thank you, Anna.
Thank you.
So we are slightly late, 8 minutes late. We're gonna crunch it from the lunch period. Now we have Will's flowers. Dangerous liaisons from quickly female choice to medieval battlefields in genital evolution of Glaphyriinae.
Thank you.
Yes.
This talk is kind of a follow-up of a presentation I gave 3 years ago at this conference. And that was a survey of the genital structures that have evolved in the endophallus of male Gallerusines. And I first got into this topic when I was doing a sabbatical in Costa Rica with Bill Eberhard on just a general survey of copulation in Chrysomelidae. And I was out collecting and found a pair of small Galluricines copulating, and this is in the genus Monolepta. And you should remember that name for this talk. But anyway, I flash froze the pair and dissected them, and I found this situation. The male median lobe is on the left, and it is inserted into the female genitalia. The principal organ in the female is the bursa with a spermatophore. But at the tip of the male endophallus are this cluster of long needles, and you can see several places those needles are actually sticking through the female bursal tissue. Shortly after we published our general study in Chrysomelidae, a Japanese team did a study on another genus, the genus Pyralta. And they found that the male has, inside the endophallus of the male is a sclerotized ribbon with a string of curved teeth, kind of like snake teeth. And when they dissected the females, they found that the females had scars on the bursa, which they inferred were caused by copulation with these males. And some years after this, I came back to this topic and started looking at the specimens available to me, and I have a little collection, and also here in the FSCA collection. One of the species I looked at was the elm leaf beetle, Xantha gallirusa. And Xantha gallirusa is very close to Pyralta. And in fact, Xantha gallirusa has a endophallic sclerite system very close to Pyralta. And in the female elm leaf beetles, I found evidence of scar tissue. So in the talk I gave before, I went through a list of different genera in the Galluricines, and these are some of the structures that I found on the male endophallus. Here's some more, and yet more. Now for the vast majority of these, we have no idea what's going on during mating and what these structures might be used for. We do have one study of mating in Galluricines, and this is for the southern corn rootworm. And Talamy and his group have done some very careful studies of the copulation procedure in the species. What they found was that the female, the duct from the orifice to the bursa takes a couple of very sharp zigzags, and there's a muscle which the female, when the female contracts it, it completely shuts that corridor down, and the male is completely unable to mate with the female. The male uses antennal stroking to court the female, and if he does a good job and the female approves, she'll relax these muscles and gives the male access. I found providentially in the collection a pair of southern corn rootworms that had been collected in copula and dry mounted, still connected together. When I softened and cleared the specimens, this is the male median lobe entering the female. I also found the endophallus is everted, and there's a series of robust little spines on the endophallus. They also found signs of scarring on the female. None of this is mentioned in Ptolemy's study. So I said, remember Monolepta. Monolepta belongs to a group called the Monoleptides. This is a very speciose pantropical complex of the Galluricines. And this is where the medieval arms locker type of male modifications comes to the fore. Virtually all of the specimens that I looked at, plus the specimens where they've been published, Tom Wagner and his group have done a lot of revision on the African Monolepta. And you have kind of endophallus look like this. The different grades of spines and needles and hooks. This is up on top is a species from Vietnam. Down below is a female, and there's lots of apparent scar tissue. But another thing on the females is that they also have sclerotized structures. And I don't know if you're up in the front row, you can probably see that on there's two pairs. There's a larger lower pair and a darker upper pair. You know, this here you can see it has its own set of teeth. Mapping. This is a recent molecular phylogeny of the Galluricines. And just mapping on them where you find modifications of the male genitalia. It's just scattered all the way across the phylogenetic space. Now we do have a model organism for traumatic mating in beetles, and it's also a Galluricine. This is the bruchid Callicebruchus maculatus, and for about 20 years, it's been, some teams in Europe have been intensively studying the mating behaviors of this species. And the male has a little spherical pad of sharp spines at the tip of its endophallus. And during mating, this is apparently an actual photo during an actual real-time mating. You can see the spines sticking through the female's bursa. And what they've concluded is that this is still an ongoing project. As the headline showed, there's the spines injure the female. They've demonstrated that the male the seminal fluid from the male passes through those wounds during copulation. The female plugs the wounds up basically as quickly as she can once she gets rid of the male. The female kicks the male, tries to kick him off during copulation, and apparently the longer the male stays attached, which he can kind of do with its little anchor spines, the harder the female kicks, and this kind of like a negative feedback further damages the female. Since the last few years, there's been a lot increase in interest in this type of traumatic mating in arthropods, of which Callicebruchus is an example. And there have been two major theories put forth to try to explain what's going on. Sexually antagonistic coevolution. This appears to be what's going on in Callicebruchus, and this is kind of like the default explanation when you have kind of the structures that we've seen in other Gallerusines. Cryptic female choice is kind of is gaining some attention as a possible alternate explanation. And that seems to be what's going on in the Diabrotica, because the female can basically internally shut the whole process down, which is like how the male's been stroking. On the other hand, the Ptolemy's explanations don't take into account the possible use of those endophallic spines. And another aspect is that some of these male modifications may appear to be antagonistic, but also possibly also stimulatory. And we got another example in Gallerusines in the Phyllecthrus. The median lobe and the endophallus are like above, and an enlarged view of the endophallus is here on the bottom. There's a big sharp spine sticking out in one direction, but a series of very fine teeth, which kind of implies a stimulatory aspect on a different side of the same plate. And there were two copulating pairs of Phyllecthrus preserved in the FSCA collection. And one of them, you indeed see the spine sticking up through the female bursa, but on the other one, this apical sclera is completely enclosed. Another aspect of a possible cryptic female choice is that the female is also evolving structures in response to male evolution of copulatory structures. And guess what? Here in the Gallerusinae, these are a series of modified female bursa that I found. And again, in the genus Monolepta, these modifications are the most elaborate. Here again, you can actually see in once the teeth in female structures. And it's kind of obscured, but does damage always follow when females mate with a well-armed male? The answer is no. In both the elm-leaf beetle and also one of the monoleptas that I was able to, I had access to multiple females that I could identify with certainty belong to that particular male. In most cases, I found that the females had scars on the bursa, but I also found cases where the females were undamaged. This is particularly important because this female was undamaged, and it also shows signs of spermatophore material, so it wasn't undamaged because it's virgin. And the title here is a bit off topic, but how does chromatic mating do under climate change? This is a paper that's just come out. I found online. It was again Callus abrucus. They took some genetic lines that they'd been rearing polygamously and monogamously, and I don't really understand exactly how they did that, but they took beetles from both of them and also subdivided that into two different habitats, complex and simple. And then they hit them all with a simulated heat emergency. And they found out that the group with in the more complex habitat was able to maintain their population through this multi-generation climate emergency a little better. That was the last slide anyway.
Click right now.
Okay. I'm just saying this kind of bad news for the future, not only of the Gallerusines, but everything else. The photos on top are some of the genera in this study, and they all come from Borneo. And the background photo is what Borneo is turning into, which is a very simplified habitat, and global heating's coming for not only the Galarucines, but all of us. So.
Do you suspect that any of these taxa, the male endosome or endophallus is actually maybe adapted to break off and remain in the female and prevent further mating?
I haven't seen anything. I haven't seen anything like that. Something that, in Hayden's talk earlier, he had some bursa in the Lepidoptera with these highly elaborate signa. There's this Carlos Cordero who was working on kind of the female cryptic choice thing. Apparently, in some Lepidoptera, the males have these little needly spines, and they do break off in the female. I haven't seen anything like that yet.
Yeah. Just wondering. Mating rates for residents as it relates to.
Nothing to that. I haven't really seen anything like that in the Calisabrucus papers. There's a book on cryptic female choice in arthropods, edited by Bill Eberhardt. That apparently is a thing in odonates, for example, among other things. Some spiders also, I imagine. One thing I wanted to, I also should point out is some of those earlier photos of these male endophallus is all spread out, and that's me doing it in the lab with nothing around the veil. What they look like inside a female bursa could be something entirely different.
Thank you, Wills. Got our last talk before lunch. Felipe Dames, Phylogeny and Biogeography of Florida species of Lepidoptera. Yes.
Hopefully this will be a less traumatic presentation right before lunch. What I'm going to talk to you about today, what I'm going to present is the conclusion of a talk I gave 2 years ago about the Lepidoptera. And I justify using this group because it's one of the largest of Collembola with about 300 described species. It is relatively easy to recognize the genus and the species because they have color patterns and shapes that make them fairly easy to recognize in leaf litter samples. And they're very abundant and easy to collect. So it's a convenient group to work with. Last time I mentioned that despite the fact that there are about 56 species described from the New World, only 3 have been recorded from Florida. That seems to me absurd because of the diversity of ecosystem and the range of temperatures in the state. This is a map. This is the last map on temperature plant hardiness. This is new. There used to be this peninsula here is new. It used to be that this temperature range was continuous, but I think that's Orlando metropolitan area is making everything hot. Yes. We collected leaf litter samples in about 2 counties. Not surprisingly, mostly in north central Florida. I have a few from the Panhandle and a few from down south from Monroe and Miami-Dade County. And so far, I mentioned this last time, we have 13 species. I found the 3 original ones. There are 7 new records and 3 new species that I have been trying to describe like for 4 years, but they will be published this year, I hope. Here, Phinus is highlighted because I found this in crushed leaf ponds in Washington Oaks about a month ago. So this is, well, it was in December. So this is a recent finding. So I'm sure I'm going to find more species in this genus in Florida. Now, Florida has gone through a series of cycles of being submerged and subaerial throughout the past several 100 million years. And each time the fauna disappeared and it had to be recolonized. So the last time this happened, the last time that Florida was open for business for us was about 2.5 million years ago when we had the glacial period. So the sea, all the water was sequestered in the glaciers, and Florida was huge at that time. So the next question after I have all the species, so which of the biogeographic regions has contributed more to the fauna of Lepidocyrtus in Florida? Is it mostly in the Arctic because we have the connection directly with the rest of the continent, neotropical, with things coming from the south? Remember, Florida used to be much wider, but it's just a crossroads, some from the north and some from the south. And I'm going to divide this in 2 sections. I'm going to talk about the non-endemic species separate from the endemic species. And I'm going to address the question using geographical distribution with the knowledge we have about the non-endemic species and with phylogeny. And then at the end, I was hoping that I could use geographical distribution as a proxy for phylogenetic affinities, but well, we'll see. Now, so starting with the non-endemic species, there are 1, 2, 3, 4, 5 species with Nearctic distribution and 3 of them with neotropical distribution. And I have highlighted here Phimiculus because Phimiculus was described from Puerto Rico, but that's the only record from a tropical place. All the other records are from the continent here, from North America. So even though it was collected, it was first in the tropics, I don't think it's really a tropical species. And this is the distribution of the species, that I have Nearctic distribution that we know. As you can see, all of them are clustered on the upper part of the state. The only one that is down here is Phimiculus, which is sort of everywhere here in the center of the state, and that one there. Now, Phimiculus, it's a little difficult for me. You see, there are 2 color forms. I'm not sure that is really Phimiculus, but at this point, I don't have a good character to distinguish them, except the color pattern. This form is only up here. This form is here and there. So we don't know what's happening. And the neotropical species, they're only in the south. There's nothing really here in the northern counties. So now, if we look at the distribution of the endemic species, the 3 new species, Floridensis and Milsey, which were described in the 60s, they're everywhere. So they are more abundant than the Arctic species. This species, Bogwood trophy, is everywhere from the south, here in the north. Remember, that is the gap. I don't have samples from there. Brambili is only in the northern part. Mike Tomasi, that is a very strange species. I found one in Melbourne, here in Brevard County, and then from Monroe County. That's the only 2 populations. Floridensis is the only species of the endemic species that is only found in the southern part of the state. And Milsey is only found on the northern part of the state. So by using distribution, and if we speculate what is the origin of this species based on the distribution, so there are two species that have a putative Nearctic origin, Brambili and Milsey. Floridensis is a putative neotropical origin, and Bob Woodruffy and Mike Tomasi, I don't know, because they're everywhere. So what do? Well, I do phylogeny, okay. I did a parsimony analysis based on 39 morphological characters. I had, in fact, like 120 characters, but I wanted characters that were described for all the species. I didn't have specimens of all the species that are included in the analysis. I had to get the characters from the literature. And the characters, this is from two publications, Marimut 1980, 88, on the Lepidoptera of Puerto Rico. This is Cipolla 2022 from some Lepidoptera from Brazil. And there are two sets of characters. There are ketotaxi characters, as the number, shape, position of the zeta, microketa. This is the distribution of microketa. These are details of the ketotaxi on the second and third abdominal segment, the head. And then there are structural characters, what I call structural characters, like whether there is a vesicle at the tip of the fourth abdominal of the antennal segment, the shape of the claw, number of teeth, their size, their distribution, their spread, the shape of the mucro, and there is many species have like a tubercle at the base of the dents. That is that part there, which is assumed to serve to help with traction when the animal hits the ground with the furcula to jump. So I included 43 species of New World Lepidopterus. Okay. And I only included 43 because I didn't want the matrix to have many holes. So if it had more than 3 empty cells, I discarded that species, which resulted in having 11 species of the Arctic of the 18 and 26 of the 36 neotropical species described. All the other species, these are species that we can diagnose, but I cannot get enough characters to include it in the data matrix. And I included as the outgroup 6 species of Pseudocinella. Pseudocinella is clearly derived from Lepidopterus. The only difference between Pseudocinella and Lepidopterus is that Pseudocinella has fewer than 8 eyes. There are no other characters that distinguish the two genera. These things have radiated in caves. There are 700 species of that thing across, mostly in Europe, because that's the place where people go into caves. So let's look at the tree. I got lucky. So this is a strict consensus tree. This is a parsimony tree of 94 parsimony trees. And I divided by color. So the neotropical species are in blue, the Nearctic species are in red. And the first thing that you can see is that it's a good partition of the groups, fairly good. I don't have bootstrap values. It would have still been running, I can assure you. But I ran the analysis several times and I got the same 94 trees. So I'm confident that at least the big groups would be retained in a bootstrap analysis. Anyway, so the neotropical species form a group here. Nearctic species down here. There are only two exceptions. Phynos, which by distribution, you would think it's the Arctic, is here nested in the Neotropical, and I'm not surprised because that species occurs in Mexico too. The other species is Diminutus, which is a Neotropical species, but it nests freely here within the Arctic species. So if we look at the distribution of the endemic species, surprise, surprise to me, most of them are nested in the clade of the Neotropical species. The only species that is not with the Neotropical species is Milsey. But all the other species are in the Neotropical group. I like this group here. They are all the species are here. Bogudrophy, Phynos, and Floridensis. So going back to the question, which biogeographic region has contributed most species to the concurrent fauna? For the non-endemic, based on phylogeny, both have about equal number of species. No? Lepidoptera phynos has a Nearctic distribution, but the phylogenetic affinities are clearly were Neotropical, and as I mentioned, it occurs all the way in Mexico, and I can easily see how it would have originated down in Central America, Mexico, and was distributed through Texas and Louisiana. In terms of the endemic species, most of them belong in a Neotropical lineage. Milsey is the only species that is endemic with a strong Nearctic affinity. So in general, which region has contributed more? Florida definitely is a crossroad, but 8 of the 13 species have Neotropical affinities. And the second question, is a geographical distribution a satisfactory proxy for phylogenetic affinities? Nah, no, not really. But I guess I was well known a long time ago. And that is my presentation. And the only thing that I wanted to show you at the end is the fact that what you see in alcohol is not what the animal looks like when it's alive. So this is Phimiculus alive with all the, when it has all the scales, it has this color pattern. It's just like that in alcohol. And this is Phynos with that beautiful pattern. And when in alcohol, it has nothing. Thank you very much. Huh? Yeah, yeah. Yep.
Any question for Felipe?
Thank you for talking loud enough for us to hear back.
You're welcome. Okay.
Are you going to collect on those counties that you?
Yeah, I have to keep collecting. South of the line between Pinellas and what is it, Brevard County, in the middle there between. And I have to be more specific about habitats too, because you saw what happens with the new species from plants. Yeah.
That was gonna be my question. You looked at your vegetation map. Do you ever know what kind of leaf litter and the primary vegetation that's happening above?
I just write down whether it's hardwoods or pines. I really don't distinguish the type of tree. But that is something that can be looked at, and the pH of the soil. Yeah. Yes.
How difficult is it to get a picture of the more colorful species?
So if you can collect them alive fairly easily, and they molt every 2 or 3 days. So even if you mess them up a little when you're collecting them, wait until they molt and you can take a picture. Yeah, put them in the refrigerator to cool them down because they move very fast. Yeah.
Thank you, Felipe. We're done for the first morning, and so we are ready for lunch. Julieta, you wanna?
5 minutes to open everything up. Look at the post.
Everyone, welcome back. I hope you enjoyed
lunch. I have a few reminders for you. We have a beautiful book by William Cooper. He is present here. So feel free to take a free copy. William, you can stand up if you want. And he can also sign it for you. So feel free to take a free copy. Another reminder, we have our business meeting at 6. So if you want to stay, feel free to stay. It is open. And after the meeting at 7, we were going to Bahama Breeze to have dinner. Just think about that, and I will remind it to you later so that we can count you. And if you want to join us, yes, at least we know how many. Okay, so we can start with the first presentation of our afternoon section, Punet, taxonomic revision of the ant genus Nylanderia, Hemiptera formicidae in Vietnam.
Thank you all. I'm Ashut Punet. You can call me Ash. I'm a third-year PhD candidate in the Lucky Lab at the University of Florida. After a nice lunch, I'm seeking your attention to tiny but crazy ants of the genus Nylanderia. More specifically, I'm going to talk about integrative taxonomy revision of Nylanderia in Vietnam. Nylanderia is a nearly globally distributed ant genus with nearly 130 species, and many are known as globetrotters because they travel via human-mediated dispersal pathways. Nylanderia has centers of diversity in Neotropics, Southeast Asia, and Australasia. Taxonomically, there are like subtle morphological variations and cryptic diversity. How can we identify Nylanderia? These are small to medium-sized ants. It belongs to the family Formicinae, so you can see a pseudopose at the tip of the gaster. The mandibles with 6 teeth, dorsal mesosomal setae arranged in loose pairs, and propodium lacking gerect setae. Overall, mesosoma will be compact. Considering the taxonomic importance of Nylanderia, this is many species are known as invasive. They displace native species, causes household damages, and serve as agricultural pests. Interestingly, a large proportion of ants intercepted ports of entry worldwide are Nylanderia. And there are difficulties in species delimitation and identification because of limited resources. There we are, the Nylanderia research team at the University of Florida and Towson University. With the NSF support, we are providing global revision of Nylanderia and also providing taxonomy training to overcome the taxonomic impediment in the genus. I want to focus on the importance of Nylanderia as an invasive species. So you can see Nylanderia borbonica here. This is one of the widely distributed species in the ant genus. It known to have origin somewhere in Southeast Asia. However, the Thai specimen is located in a non-native region, Reunion Island. Taxonomists are so confused whether this wide distribution is represented by a single species or Nylanderia borbonica is represented by multiple cryptic species. So I did this sub-project on Nylanderia borbonica species complex. So it belongs to Nylanderia borbonica belongs to Nylanderia borbonica species complex, which consists of 2 species, borbonica and vega. But with the molecular analysis, we found at least 5 species in the species complex. Apart from that, there are several morphologically similar species. We call them borbonica-like species, frequently misidentified as Nylanderia borbonica. So there comes the importance of taxonomy in this genus. So for my PhD research, I'm focusing on Asian and Australasian Nylanderia revision. As part of the Asian Nylanderia revision, we got a large specimen from Vietnam last year. So most of the specimens are collected during International Ant Course, which is organized by the California Academy of Sciences. To give an overview of the ant fauna in Vietnam, we have 373 species of ants known so far. Out of them, 5 species are from Nylanderia, but these are not published records, these are museum records. So this present study provides the comprehensive taxonomy revision of Nylanderia from Vietnam. So all the specimens in this study are mostly collected from Cuc Phuong National Park. It's located in the Ninh Binh province in Vietnam. You can see the forest consists of broadleaf tropical evergreen lowland and situated in the foothills of the northern Annamite range with elevation 150 meters to 656 meters. And thanks to my supervisor, Dr. Andrew Lucky, and also Dr. Phil Watt from UC Davis, my lab mate, Leo Hammer, who collected the specimens from Vietnam. After getting all the specimens, I did the basic taxonomic procedures like sorting, identification, imaging, and also like building their illustrated identification keys. For an integrative approach, we generated the UC data, ultra conserved elements data. These are highly conserved regions within the genome and a modern tool for sequencing multiple independent genome regions. And after getting this UC data, we generated the phylogeny tree using maximum likelihood approach in IQ-tree2 with the US supercomputer, the hypergator. Coming to the results, we found 10 species in Vietnam. Out of them, the last 2 are not collected in this study, but they are museum records of the species. Here is the phylogeny based on UC data. Just to show you like those phylogenetically distinct species are well morphologically distinct, except few species at the top. A species account of Nalendaria in Vietnam. You can see Nalendaria birmana. This is a very peculiar species with micro reticulate sculpture on its head and mesosoma, and the pronotum and mesonotum with 4 to 8 erect macrocyte. Nalendaria bourbonica, as I mentioned, this is one of the globetrotting ant species with long and dark brown to almost black in color, and overall cuticle smooth, glossy, and lacking surface sculpturing. One important feature of this species is that abundant red black hair-like macrocyte and gray pubescence on head, mesosoma, gastro, and legs. This is my favorite Nalendaria MA. Yellow to light brown body with oval-shaped head. Interestingly, one of the striking features of this species is the striations on its mesopleura, and also you can see the same on propodium. Nalendaria picta, as I mentioned before, most of the Nalendaria species are medium to large size, but this one is a small size Nalendaria species with small eyes. And they have short pipe ears and the first segment of gastro without pubescence or scarce pubescence. Coming to the potential new species from the region, very interesting yellow to brownish yellow body colored Nalendaria with shining cuticle, darker erect macrocyte across body. One of the interesting features of this species, which separates from other Indo-Malayan species, you can see the dome-shaped podium, which makes it a new species. Nalendaria, another species with darker brown body, and you can see like lack of pubescence on body except propodium and a subrectangular head. Here is another species, as I mentioned before, there are several species, especially from southeastern China, which resembles Nalendaria bourbonica in their appearance. So this is one of the new species which closely resembles Nalendaria bourbonica. They have a darker brown body with abundant erect. One character that differentiates this from Nalendaria bourbonica is their smooth cuticle with a blue iridescence and lack of pubescence. Very cool species, tricolored body with abundant erect macrocyte on head and gastro, distinctly paired macrocyte on its pronotum and mesonotum, and an oval-shaped head with sparse pubescence. These are museum records of 2 more Nalendaria species from Vietnam. Nalendaria vega, just like Nalendaria bourbonica, these are globetrotting ant species with overall dark brown color and dorsal face of propodium gently rounded in profile. You can see here. And mesopleuron without pubescence. Nalendaria gibordina. This is a brownish yellow bodied Nalendaria species with darker gastro oval head and punted sculpture on head, tip of castor and legs. Well, that's not the end of the story. We got another very cool, interesting Nalandera species, but with unusual Nalandera characters. So we thought of running a general level phylogeny with the UC data, and we found Nalandera indigera, no longer a Nalandera species, it belongs to Zedania. So that means the genus Zedania is it's the first time it's reporting from Asia. Zedania is a relatively a new genus, previously known from the greater Andes and Central America. There are 6 species in this genus, with mesonotum distinctly elongated, pronotum and mesonotum long, and pronotum rises less than 45-degree angle to mesonotum. So considering Zedania indigera, these species are color orange yellow. Mesopropodial suture will be like not deeply impressed. So we also found another Nalandera species, Nalandera opisophthalmia. We found the characters of Nalandera opisophthalmia more close to Zedania. So we are providing another combination. So Nalandera opisophthalmia will be in Zedania. So Williams and Lapola, my teammates, they published this paper in 2016. They also mentioned Nalandera opisophthalmia is more close to Zedania. But during that time, Zedania was not reported from Asia. But now we know Zedania is there in Asia, so we are providing a new combination. So we can identify Zedania opisophthalmia based on their abundant pubescence on the head, scapes with no erect-city, 7 to 8 teeth on the masticatory margin. Well, the story is not over yet. So we got another very interesting species, a new species of Paratrachina from Vietnam. You can see the hairy body, so that makes it different from other Paratrachina species known so far. Well, I presented different aspects of the research, but everything points to the fact that Nalandera genus presents taxonomy challenges due to its diverse morphological variations. So there are multiple cryptic species within species complexes. So, but morphologically, it's often hard to differentiate species, but an integrative approach is promising tool for delimiting species boundaries. So most of the specimens are collected from Vietnam during the ANCOS, but last year I was fortunate to participate in the last international ANCOS, which happened in Papua New Guinea. So hopefully in the next CSE conference, I will be here to share my fieldwork experience and research findings on Papua New Guinea and island area. With that, thank you so much.
We have time for questions?
Yeah.
The globe-trotting island area?
Yes.
Is it known how they are moving?
Mostly through cargo shipping. When we are like transporting items, they are inside.
So there's like actual nests are getting, whole nests are getting moved?
Maybe, yeah, when we are, especially when we are transporting like plant materials or something. Okay. Yeah. So we have collaborators at ports of entries, so always they ask us how to identify these species. So especially from Australian ports of entries. Yeah. Yeah.
Are you identifying only females or also males?
We can identify actually. So for Nalandera, for Bonica Nalandera vega, these are morphologically very similar species. So how you exactly identify these two species is based on their male gene value. And we also have one project in the lab currently looking at Nalandera fulva and Nalandera pubens, which morphologically is similar when you look at the worker specimens, but they are different based on their genital structures. Yeah.
Do you collect all your specimens like via aspirated?
Yeah, we can color. So these are like 5, one of the 5 most abundant, I mean, leaf litter ants. So whenever you collect like litter samples, you can encounter, we usually encounter Nalandera ants, so, but you can collect with the aspirator as well. Yeah.
Awesome, thank you, Ash.
Thank you.
Thank you very much. Okay, the next speaker is Paulina Zelali, the integrative study of the subgenus Orthops, Heteroptera Miridae, and we should have a pre-recorded presentation.
Good afternoon, colleagues. I'll tell you about my research as integrative study of the subgenus Orthops. Palearctic is the largest biogeographic realm with many biomes and climatic zones. The Palearctic insect fauna is considered to be the best studied compared to other regions. Most species are diagnosed by morphological characters only. This is not sufficient to study interspecific variation and cryptic species. Many abundant and widely distributed species in the Palearctic have been poorly studied. Also, the boundaries between species are not always clear. Some of them are extremely variable externally. The integrative approach is needed to disentangle the taxonomy of such taxa. The studies on the widely distributed species in the Palearctic are rare and usually are performed on the European taxa. We choose plant bugs from the Miridae family as our subjects. This is a large family. It accounts for a quarter of the total diversity of Hemiptera. There are 2,800 species of Mirides in the Palearctic. This family is diverse in all continents except for Antarctica. Among plant bugs, subfamily Miridae is the most diverse. It includes many widespread species, including trans-Palearctic, trans-Haloarctic, and those that inhabit 3 or more continents. Orthops is a genus within Lygus complex. The Lygus complex within Miridae consists of about 50 genera, what are very similar to each other, and many of them are taxonomically challenging. Orthops is one of them. Orthops is distributed in the entire Palearctic, Africa, and North America. The genus comprises 36 species, which are usually medium-sized plant bugs. Orthops has 2 subgenera, Orthops and Montana Orthops. They are mostly recorded from the family Apiaceae. The subgenus Orthops included 7 Palearctic widespread species: Orthops basalis, Orthops campestris, Orthops frenatus, Orthops calmeo, and Orthops cutellatus. According to the previous works, the differences between the species within this group are small. There is no molecular work testing the boundaries of these species. Within this subgenus, Orthops basalis, Orthops campestris, and Orthops calmeo are widely distributed and known from the Western Europe to Western Siberia. Orthops cutellatus is known from the large areas in the eastern and western Siberia, East Asia, and Nearctic. Frenatus inhabits mostly Caucasus and Central Asia. Orthops vitipsus and Orthops ferrugineus inhabit China. According to the previous works, the differences between the species within this group are small. The 5 widely distributed species differ mainly in the front scala and the structure of parameres. It remains unclear whether these characters are species-specific or represent the interspecific variability. So far, there have been no published comparative study on the Ediagus and the female genitalia for the species of the subgenus Orthops. These structures may be important for species discrimination within the Miridae. The aim of the current study is to test the species bodies in the subgenus Orthops using the morphological and molecular data and to test the phylogenetic relationships between them. The regions studied include material from various regions of Russia, as well as from Europe, Asia, and North America. Our methods include examination of external morphology and molecular genetic analysis. To study morphology, we used standard preparation techniques, light microscopy, scanning electron microscopy, and morphometry. Our molecular techniques are mitochondrial and nuclear markers, construction of phylogenetic reconstruction by Bayesian and maximum likelihood techniques, and species boundaries with automatic species separation methods. Now let's move to results. The traditional features for identifying orthop species were following: the coloration of hemelytron, the coloration pattern on fronts, the vertex with ideal diameter ratio, the shape of parameres, and stripe on tibia absent or present. We identified potential diagnostic features, the differences in the structure of Ediagus, and the differences in the female genitalia. Hemelytron coloration can be useful in some cases for species identification. However, it should not be used alone in the case of widespread species. Shape of the markings on the fronts can be misleading. The paired markings on fronts were diagnostic for Orthopsis basalis, but this condition was found in other species too, and some specimens of Orthopsis basalis have single markings. The round markings on fronts were diagnostic for Orthopsis calmi, but sometimes it can be absent. The Y-shaped markings on the fronts of Orthopsis cutellatus may be absent. Orthopsis campestris usually doesn't have markings on the fronts, but Daca form may have one. The most reliable character complex for the species discrimination are the shape of both parameres. However, in some cases, the differences are very small and should be used in several specimens of the different species can be compared. Genital capsule and theca are identical in all species. We have studied the structure of the Ediagus for the first time. Ediagus structure is less variable and is identical in Campestris, Calmi, and Scutellatus. The most species except for the Orthopsis basalis and Orthopsis calmi can be identified using the female genitalia, dorsal labiate plate, and posterior wall of bursa copulatrix characters. We constructed phylogenetic trees based on 2 mitochondrial and 2 nuclear markers. In general, the results for the different markers are very similar. Specimens of Orthopsis palus from Indian Ocean islands do not form a clade with other Orthopsis species. Our phylogenetic tree suggests that generic assignment of the species should be reconsidered in the future. Orthopsis calmi and Orthopsis campestris were monophyletic in all phylogenetic analysis. Orthopsis basalis formed a clade in most phylogenetic trees. Orthopsis cutellatus was divided into 2 clades, palearctic and nearctic. The species delimitation analysis also confirmed this. The representatives of these 2 clades show slight differences in the parameters. We did the phylogeny with fewer samples but for markers. Except for Orthopsis cutellatus, all species formed isolated clades with height supports. Orthopsis cutellatus sequences break up into 2 clades from different continents. This may indicate that the species actually represents 2 different species. We don't have fresh samples for the 2 species from China. However, we have recently obtained results for the species Orthopsis frenatus. It forms a sister clade with Orthopsis campestris and palearctic Orthopsis cutellatus. Thank you for your attention.
Thank you, Paulina. Is she online? Paulina?
Yeah, hello.
Okay. Hi. Any questions?
Hi.
Paulina? Okay, we have a question. Yes.
So I didn't catch whether you synonymized those species or not.
Did you hear, Paulina?
Yeah, I can hear you. Thank you for the question. Could you repeat, please?
I didn't pick up whether you synonymized those species or not.
Paulina, did you synonymize those species?
Yeah. No, we didn't synonymize these species, but it's our future plans. Thank you.
Thank you.
The other question about the American Scutularis, will somebody pursue that to be different?
Did you hear, Paulina?
Yeah, I can hear.
Can you repeat the question?
The American Orthops Scutularis was
Yeah, Orthops scutulatus.
from the others, so will you make it a joint study with somebody here to clarify its status?
Yes, the clarification of status of Orthops scutulatus. It's our future plans. We have old material, and we hope to update material for molecular work, new samples for molecular work, and it's our future plans. Thank you for the question.
Thank you, Paulina. Our next speaker is John Watts. I didn't know butterflies did that. Interesting and unusual behaviors among the scutters.
In case you're wondering that word scudder, I found that in some very old literature. It was another word for the true butterflies that separate them from skippers.
Yeah.
A little trivial thing.
John, can you look this way?
Thank you. Okay.
John, so just so everybody understands,
Magic get in there.
we have some supplementary videos that are not part of the slide file, so we're going to share the entire screen. And John, when you want me to switch to the video, I will open it up. So I just downloaded these. I have not had time to actually open them and verify they work. So we're gonna hope for the best.
Let's hope.
But I downloaded the files you need.
Okay, appreciate that. Okay, so this is going to be a very quick run through of butterfly behavior and some of the stuff I've discovered in conservatories, in addition to some stuff you already know from wild butterflies. What do I have to change? Oh, sorry. Big one? Okay. Okay, as you can see, I've broken them up into 6 sections. We'll start with feeding strategies. Of course, most of us know that the majority of butterflies, in fact, all butterflies are restricted pretty much to a liquid diet, and they tend to go to flowers, and they do have preferences for certain colors as well as shapes to them. Usually they're shades of lavender, pink, orange, and yellow. But I've also observed on more than one occasion some butterflies going to philodendron sphax. Here we have some Archiprepona and some Mycelia enthusa visiting some of these philodendrons I got inside the conservatory, and they actually indeed produce seed afterwards. Males of Ithomines and Danaeines can often be found visiting certain flowers to acquire chemical compounds and components utilized in pheromone production. And in particular, they seem to be most interested in Heliotropium, certain Eupatoriae composites, which includes the bone sets and Joe Pye weeds, as well as Solidago, and certain Solanaceous genera, such as Brutmansia, as you can see the Tithoria on the right, are visiting to get these substances. Heliconius is one of those genera of nymphalid butterflies that deliberately collect pollen on the proboscis. Most other butterflies do not do so, it just ends up there accidentally. But they do this on purpose, and we call it a pollen load. And the reason they do that is in order to regurgitate some of their stomach enzymes. It will digest the pollen externally, and then they get amino acids from the slurry that they suck back in. And this allows them to live up 4 to 5 months instead of the usual 2 to 3 weeks. As a result, longwings are extremely intelligent for butterflies, as you will find in some of the other slides. In fact, Doris's longwing will go a step further than other longwings, and that it will deliberately hide the floor that is on from other longwings when they come in to try and feed on the same flower. I'll call this pollen hoarding, and it's only this particular species that I've seen do this. Of course, not all the butterflies feed on flowers. In fact, when you go to the tropics, because there is such a high diversity of flowering plants, you might not have a whole lot of one particular species blooming in a very close region. So a lot of the tropical species have instead adapted to feeding on the juices of overripe fruit as well as sap oozing from trees. Some of these fruit feeders have thick, shortened proboscis that are strong enough to actually poke through the skins of the fruit. And in particular, Caraxes, all the Caraxines are pretty much that way. Defecation strategies. You don't usually think of butterflies as using the bathroom, except when they first come out of the chrysalis, in which case we call that substance that they drip out of the end of their abdomen meconium. But they still have to use the bathroom once they've even passed that point. Because even though nectar is solid, there's still going to be waste products to it. And most species just shoot it out the back end. And they don't really care where it ends up at. I've actually seen that happen in flight as well. I've seen certain swallowtails just flying in the air and right out the back end. But what about those butterflies that deliberately land upside down? If they were to do it the usual way, it comes back down on them, right? So what they do instead is they open the wings up, curve the abdomen over the front, and shoot it forward. And I've seen this both in Catanephly and in Mycelia. Watch butterflies. They do all kinds of neat things. Aggregation strategies. We all know, or at least male butterflies of many groups, do indeed aggregate at moist and wet places to drink the water, imbibe it, and what they're doing, of course, is collecting the salts and minerals that are left behind whenever the water evaporates away. And they use those substances in sperm production. So it's usually only the males that do the puddling. But there are other types of aggregations. Everyone's, of course, familiar with the aggregations of the monarch butterflies, and especially in Mexico. And it's one of their ways of being together at the right time to mate at the end of winter, as well as having the visual effect of their aposematism being much larger and therefore easier to see by predators. Longwings, too, have communal sleeping roosts. And due to the fact that they live longer and have developed a longer memory than other butterflies, they return faithfully to the same spot every night. And in fact, these roosting aggregations have been found to last not only several months, but up to years, with individuals replacing the older ones as they expire. So the new ones find the aggregation spots by watching the older ones where they go. Told you they're smart butterflies. And there are instances of interspecies congregations in longwings, where it's not just one species of longwing, but multiple species. And there are also examples too of things not even closely related, like Idea and Pterimella, which are in the Danaid group, also aggregating along with the longwings. Figure that one out. Sound production. This is where we hope that we got what we want. I want here. You may already be familiar with Hamadryas, the cracker butterflies. They get that name because they can make audible crackling noises, and the males use that in courtship fights. And you can actually hear it very easily when they're spiraling up in the air doing their territorial aspects. And it's supposedly a result of a mechanism at the base of the swollen front costal veins. But there are other species that I've discovered in conservatories that make audible noise as well. And these are the two here, some of the craxies and some of the caligos. And let's see if we can get the caligo, Memnon. Sorry. Well, that's okay. I'm talking faster than I anticipated,
Okay, so you want
probably because I'm expecting questions.
You want the caligo file, you said?
Caligo first, please. There you go. Did you hear that? Okay, and this obviously isn't territorial because these are the first day these butterflies emerged and they haven't yet set up a territory. So it's probably for predator avoidance. Okay, let's go to the next slide. And now do the craxies, Veranis, please. Pretty cool, huh? And it seems to be, from what I can see, a rapidly vibrating of the wings. There may be something else going on there. I know that caligo have extra organs on their abdomen and hind wings called a Vogel's organ that I don't think anyone's ever paid much attention to as to what its structure is for. But the caraxes does not have that. I'm so glad that worked. I was afraid I had to have you think, just believe me, not know it actually is true. Bless your heart, man. Appreciate that. And I believe this was actually discovered here at the McGuire Center not too long ago, that the blue and white longwing, Heliconia cygno, produces audible clicking noises too. And they do that when they're coming in to roost, probably like sonar to find each other. Territorial mating behaviors. Of course, most species of butterflies and males just do this spiraling up and down. That's how you can tell they're two males fighting over territory and not courtship, like a lot of people think it is. But there are other species that are more aggressive. Cracker butterflies. Again, we already learned about these. The males only have the crackling mechanism, apparently. And of course, they use that when they're fighting over territory. My guess is that the swollen part of the veins on the forewing also act like a tympanum of sorts, that they can feel the vibrations through them. Rajas, which are caraxes, some of the species, the males have very sharp teeth on or scales that on the edge of the forewing that stand on edge. And if you look at it on a microscope pictures, they look like saw teeth. And what they do is deliberately scratch each other's wings up when they're fighting over territory. An older male will have less scales on the wings and therefore look less fit to the females. Of course, we know color patterns in butterflies are often used in courtship. I mean, they're extremely visual creatures. Our eyes have 3 cell types in them. Theirs have between 6 and 8 different cell types, so they see way more colors than our eyes can detect. As a result, males tend to be more brilliantly colored than the females, just like with other animals. Females have all the strength when it comes to determining which male they want to mate with. In addition to bright colors, a lot of species of male butterflies have specialized scales, and what we call androconial patches. Some of them have hair pencils that come out of the abdomen and even on top of the hind wing and tithoria. And these scales produce a cologne or perfume scent attractive to females. In fact, a number of the species, the scents are quite powerful enough that we can detect them with our noses. The eastern tiger swallowtail is one of those. As a kid, I used to love to catch a fresh male, stick him up my nose and smell his hind wing, and it smells like crepe myrtle blossoms. And Phoebus agarithi, the large orange sulfur of South Florida, they smell like nacho cheese, Doritos. It does. It smells like nacho cheese. At least it does to me. Or maybe it's just because of the color. I'm not sure. Since butterflies hold the wings upright when they're resting, they can't mate up one on top of the other like most insects do. They have to do theirs end to end in what we call tandem. Of course, And that can take several hours. If they have to fly from a predator, it is the female that does the flying. The male just sits with his wings closed and legs pulled up next to the body and gets dragged along while she's flying. Females have all the power. I don't know if you're familiar with the Parnassian butterflies. They are very primitive swallowtails. They're mostly found in mountainous regions. In the US, they're mostly the Rockies and the western states. They go all the way up to Alaska and so forth. But the males, what they do to insert a paternity is they secrete the substance that hardens into what we call a structure called a spragus. And it has a tube down the center that the egg just rolls out. No other male can go in there and scoop out any of the other male's sperm, which happens in some other insects. And so that is his way of making sure that his genes are the ones passed along and not another male's. Monarchs don't even court anymore. I suspect that one of the reasons that they have developed this is because when you're all together with hundreds of millions of butterflies at one spot, how is a male going to court a single female when you got hundreds of millions of other males? So I think what has happened here is they have completely decided not even to mate anymore, the usual way of courting, and they just wrestle the female out of the air and mate with her in sort of a rape type technique. Their androconial patches no longer produce pheromones, whereas the queen, which is closely related, still does it the old fashioned way. And there's even one clade of longwings that practice pupil raping. The males force themselves into the chrysalis just before the female emerges. I guess the female is still putting out pheromones while she's in that chrysalis. Otherwise, you might not know it's a female. I'm assuming that, but then if somebody here has been studying the longwings, they can let me know if that's true or not.
Is that the zebra butterfly?
That is the zebra longwing, yes, we have here. Predator avoidance. Of course, a lot of butterflies use camouflage as their way of keeping from being predated upon because like they have to rest, all of them have to rest. So if you look like your background, things don't see you. You know, you look like dead or green leaves, bark of trees. And of course, predators walk right by you, they do not see you unless you're moving. The glassy wing butterflies, Iphomiines, a lot of them are see-through, that's how we call them glassy wings. They are able to be camouflaged pretty much wherever they want to rest. That's pretty advantageous if you stop and think about it. Close.
Sorry, yes, time's up.
Time's up. Oh, bummer. Well, we'll run through the slides real quick.
You can have 1 more minute, yeah.
Okay, so this is really interesting. The mosaic butterfly, Colibera dursae. This butterfly is one of those that rests upside down. In addition to that, the stripes converge so that it points your eyesight to the fake head. And they even move backwards a little bit to fool you even further, thinking the back end is actually the front end. Because most birds think most insects land with the head up. And then we got the aposematism. These are bad. These want you to think they're bad by looking like the models. And these combinations of black, red, orange, and yellow, white is what we universally call aposematism. Not just with butterflies, coral snakes, bees, wasps, ants, things like that. Ladybugs. And then Bayesian mimicry is where you have one that is a model that's inedible, and the other one is perfectly palatable to predators. It was discovered by Henry Walter Bates. Right, good. Another one is Mullerian mimicry. And in this one, what you have is multiple species that are unpalatable, but they've converged on the same pattern. Franz Muller discovered this in the same place that Bates did his studies. And these were done in the late 1800s. But we're back to longwings again. I love longwings. There are two species, Errata and Melpomene, that wherever they occur together, they have converged on the same color pattern. This one is Melpomene on the left. That's Errata on the right. And this one is Melpomene, that one's Errato, Melpomene, Errato. The sunset and sunrise postman, the piano key postman, and harpsichord postman. And I believe that's actually the last slide. It is.
Thank you very much. Any questions?
Yes, sir.
I have a question about stridulation. Yes.
Yes, sir.
Can you tell me about the extent of stridulation in these butterflies?
Only that the only ones I know of are the Homodrius and now Caligo and Caraxes. I do not know how much further it is in other species. The butterfly farmers that I buy from, none of them knew this happened. They've been breeding them for decades and they've never taken the time to watch what they're rearing, see what they do. There's so much room for study in these groups, so much room. Yes, sir.
Former squirting and defecation be used as a defensive method?
I suppose it could. In the case of conservatory, there wasn't really anything they could use it on except me or any visitor, I suppose. And I suppose they might want to do that among the visitors are always trying to pick them up. Yes, sir.
Quick question, I didn't know about the zebra longwing. The males have a special like appendage to get inside the chrysalis, or like on the end of their abdomen to get it. Do they have a?
My guess is that they're taking their claspers and ripping it open to get in. I mean, the exoskeleton on the chrysalis is very thin to begin with, and when they're getting ready to come out, it's even softer than it was whenever they were still kind of goo stage. I don't know what other words to put for that. Yes, sir.
What conservatory did you mean studies at?
This is at two different conservatories. The Butterfly Pavilion in what used to be Westminster, Colorado, now they're in Broomfield. And the one I'm at now, Texas Discovery Gardens.
Awesome. Thank you, John. Thank you very much. Our next presenter is Samuel Bolton. Why one believes in mite monophily just die already? Let's see.
So,
Oh, how do I, oh, sorry, it's sorry.
The big one is.
The big one, and then how do I get it back if I wanted, oh, that one, okay, thank you.
So. Thank you.
All right, so this talk really sums up my my very balanced, objective, and completely impartial viewpoint on this highly contentious topic of whether mites are or are not monophyletic. And basically, we can start with, well, essentially the definition of what counts as a mite has really not changed in about 200 years. So mites have belong in one of these two lineages have always been put, you know, in Acarae, and are essentially arachnids that fall outside of that, outside of either of these two lineages. They've been placed outside of Acarae as well. So it stayed the same, but the characters that have been used to support this grouping have not been good. They've not been very strong. And so basically just to emphasize this, you know, essentially this has been borne out recently in the last few years. Molecular phylogenomic analysis have not supported mite monophyly at all. There's been one or two, but not very good in terms of being sort of unbiased or taxonomically comprehensive. So I think pretty much so that really sums up in terms of is mite monophyly true or not? It's blatantly not true, in my opinion, and I'm sure there are other anthropologists who disagree with this, but it's obviously invalid, which means that we can explore the more interesting question. Okay, so this question, I know it's a horrible question, it's a horribly phrased title, and I'm sorry I couldn't come up with anything better. I put some effort into it and then just eventually thought, sod it, let's just go with this. So, but the thing is, it's a correct question. It's a good question from my point of view. And maybe a better phrasing would be, how did we get it so wrong for so long? Okay, because 200 years, right? We just, you know, we got it wrong. And the characters that have been used, I mean, it's true that not everyone has sort of gotten behind monophyly, and there have been a few dissenters. But the consensus has been that mites are monophyletic, and the people that have been sort of arguing this have sort of one decade maybe used maybe a small number of characters, and then maybe a couple of decades later somebody else has come along and used different characters, and they've just all been kind of weak and feeble, except for this one really important character that's kind of been reiterated and re-emphasized over and over and over again.
And yeah, that's the character I want to talk about today. It's the best character. It's the quintessential character for mite monophyly. And people will say, oh, well, don't worry about everything else because this is the character. So what is that character? Well, this is the technical bit of the talk coming up. This is the head of an arachnid. And this is just a generic arachnid. It's got chelicerae palps. And we're going to look into a little bit more detail on the palps. And the idea is that in the early evolution of mites, the palpal cocci, which are these little segments at the base here, they expanded and fused at the midline, expanded around the chelicerae, and formed this sort of pseudo-pneuma that we call the gnathosoma. And that dramatic expansion of the palpal cocci that's the bingo character, the golden character that says, look, mites are monophyletic, there's no dispute about it. So I want to get to the root of this character, and to do that, I want to go all the way back to the early, to the 1700s. Sorry, it's a detour, but it's I like detours, I like stories, you like stories, so let's go with this. Okay, so right, well, this picture is depicted in 1755. It's a bit early, so I want you to imagine it's not 1755, but 1798. And this harbor, this is Toulon, this is the south coast of France. It's a beautiful harbor town. And in this harbor, just imagine in 1798, in the 19th of May, 1798, this harbor would have been brimming full of boats and ships loaded up with French soldiers that were ready to embark on this adventure to go all the way to the Middle East, to Egypt and Syria, where they're going to conquer lots and lots of territory and expand the French Empire, right? But this wasn't just a military campaign. This was also a scientific campaign. And dotted among those soldiers would have been 150 savants, okay, brilliant young minds, mostly in their 20s and 30s, some geniuses among them. One of them was Jean-Baptiste Fourier, who was the famous mathematician and physicist. Now, so this is an important campaign. It had people that changed the world in the coming century. Now, this campaign was headed up by none other than Napoleon Bonaparte. And Napoleon basically, he at this time, he wasn't the leader of France. He wasn't the first consul, let alone the emperor. That wouldn't happen for a few more years. At this time, he was a high-ranking general in the army. And he had recruited among his savants, among his 150 savants, he had Etienne Geoffroy Saint-Hilaire, Marie-Jules Savigny, those important zoologists that were charged with the zoological collecting, such as they went out and collected a bunch of material. This campaign launched their careers, and it was very influential on them. It allowed them, not only did it launch their careers, the material that they brought back from Egypt, mostly Egypt, they studied it in Paris, and it basically was, it brought forth a bunch of ideas and innovations and thoughts and discoveries that went on to shape the course of evolutionary biology and evolutionary entomology throughout the 19th century. So this campaign was really, really important for that reason alone. And Etienne Geoffroy, well, Savigny was an entomologist. Maybe you haven't heard of him. Geoffroy, probably a few of you must know at least Geoffroy, because he was the guy that was behind, anyone want to venture, maybe saltation, the birth of hopeful monsters, a very important evolutionary biologist, and an incredibly interesting guy, right? So you think about evolution and the idea of evolutionary biology, these giant leaps and bounds. Well, Geoffroy was behind that. And he was very passionate, very eccentric. That's why I put sound effects, give that sort of extra feel or whatever. But he was a very passionate guy. And while he was, and after the campaign, when he recounted it, he basically said, knowledge is so sweet that I was able to put out of my mind that a bomb might explode at any moment and instantly hurl me and my evidence into the abyss. Okay, what about that for passion? Okay. So while he was luxuriating in this atmosphere of violence, mayhem, bombs, explosions, and killing and whatnot, okay, he was also coming up with some really interesting insights, some really interesting ideas. And one of the ideas that he was developing when he was out there, and also a little bit before, was this idea of unity of plant. So unity of plant, maybe probably a lot of you know about this. It's a simple idea, but the idea is that the components that make up one organism are also present maybe in a very different organism or a different animal, and in the same basic arrangement. But the form of those components can vary dramatically. Okay, so for example, the walking legs of one animal can become pincer-like structures, say, in a different animal. Okay. Now, what Savigny did, okay, he developed this idea, extended it to insects and extrapolated it, I guess, to insects and other arthropods. And he realized very astutely that the mouth parts of insects are homologous with the walking limbs of other arthropods. And he also recognized there was 3 pairs of appendages that make up the mouthparts, and they have all these different funky forms throughout insects. These 3 pairs are the mandibles, the maxilla, and the labium. And he also recognized on top of this that there's a lower lip of insects, a lower lip in insects that's formed through the fusion of a pair of appendages. Okay, so, and all of those discoveries, they were all correct. Like he was really on the ball. This is like the very early 1800s, and he really figured out an awful lot at a very early time in his career. And it's quite stupendous just to think about it back now, how much of it, now some of his stuff was wrong, but a lot of it was correct. Now, in the decades that followed, at this point also, mites were also being recognized as a taxon in the way that we sort of recognize it today. And in the decades that followed, some entomologists and some acarologists, or early ones, were starting to think about mice as this sort of transitional organism between insects and other arachnids. Now, of course, this was wrong, but what was important about it was that it led to this search for similarities between these 2 groups. Okay, and one of the things that they picked up on was the lower lip. They realized, you know, they were aware of what Savigny did, and they thought, oh, well, look, there's a lower lip formed from fused appendages in insects, and there's a lower lip that's formed from fused appendages in mites. And that is correct. It's specifically correct with respect to these things called endites, these lobes that project up out of these appendages, okay? And so you do get this lower lip. Now, this isn't actually very significant in terms of the monophyly of mites, in terms of plugging the monophyly of mites, because, well, there's other arachnids that also have fused endites, so like camel spiders and solophages, for example. So, of course, what's important from our point of view, well, what's going on with respect to the palpal cocci? If you remember back to that animation, the expansion and fusion of the palpal cocci is what creates that sort of synapomorphy, that golden character that essentially we use to distinguish mites from other arachnids. Now, the problem is, as you can see this border between the endites, the border stops before you hit the cocci. So we don't know what's going on with the cocci. We don't know if the palpal cocci are fused or not because, you know, there's no medial line between them. Now, what these early acarologists did about 120 years ago, they just kind of said, well, look, let's just sort of extrapolate all the way down here and just assume that the palpal cocci are fused, right? And basically, we've kind of gone with that. For 120 years, we've just gone with this, yeah, that'll do, you know, that's the carrot, that it works, right? The problem is there are other mites, including some orobatid mites, that have a different type of mouthpart morphology, and they have these oblique borders that, you know, separate away from the endite border or diverge away, and these oblique borders, well, and don't tell me why, but it took us roughly 100 years. In 1996, a very astute acarologist homologized these borders with essentially the edges of the palpal cocci. And he realized, well, you know, he sort of suspected, he said, well, look, these palpal cocci are probably, you know, these palpal cocci are probably separate, and therefore the inner borders, this is the sternum. And so, and not only that, but he also suggested that this is probably the plesiomorphic state, the primitive state for all mites, okay? Which of course means that this character just doesn't work, it fails, okay? If the palpal cocci are separate, they kind of expand, they kind of fuse, they kind of form this sort of gnethosoma in the way that we envisage it, that golden character does not work. Now that was 28 years ago. And for some reason, that paper that he published has just been ignored, like the papers that have been published since, the books, the standard textbooks of mite morphology, all palpal cocci are fused, you know, the discussions of phylogenetic characters, they don't mention this once. Morphological matrices that have been generated, all palpal cocci are fused, even in orobatids, all palpal cocci are fused. Which is kind of weird, because we now discovered that there's no evidence for mite monophyly, right? So what I did was I'll briefly talk about my work, which is basically that I've been working on Endiostigmata, which is this sort of grade relative to all other Charaformes, this sort of basal lineage relative to everything else. And what I found also was that, yes, mite after mite, you get these oblique borders that separate, that show sort of separate palpal cocci. And the reason why I'm sure that they show separate palpal cocci, among many other reasons, is that here's a spider, right? And you've got what are unambiguously palpal cocci, and their inner borders are oblique. And what's good for the goose is good for the gander. We can say that these are also, you know, palpal cocci, and that they're separate. Okay, and What's more interesting, when you start looking not only at endostics, but across Acariformes, and I basically undertook a sort of survey across not just endostics, but these Trombidiformes and various other groups, and started using confocal microscopy, looked at academia and whatnot. And when I could see a state that was exhibited confidently, or when it was obvious, it was almost invariably separate. Now there's a few cases of fused, these black areas fused, right, fused species, but they're rare and they're derived. So this character that's supposed to be primitive to all, or supposed to be sort of the synapomorphy that unites all mites, it's a nonsense character. And so yes, going back to that question, well, you know, how do we get it so wrong for so long? I got 2 minutes. How do we get it? I'm just about to finish. How do we get it so wrong for so long? Well, the answer is we just haven't been looking properly. The evidence has been around us for 120, for 200 years, and we just haven't been examining and scrutinizing the characters. All right, thank you. Sorry, that's it. Any questions?
Any questions? Yes.
Are you planning to do maybe even some muscle studies? Because
No, somebody should. Yeah, you're right. Somebody, I mean, there's, you know, but I've been working on this for a while, and yeah, no, I'm ready to move on. The paper just been submitted, so sorry. Yeah, but yes. What was your question? Your more specific question was?
No, because I'm thinking about also Syrphidae, and back in the days there were some ideas of that the Omatidia, exactly those are not Omatidia, and have been actually Ocelli, and they've been discovered that by Snodgrass already in the 30s.
Right.
Because looking at muscle and where is the clepeus and stuff like that. So maybe you can find even more characters that points to your conclusion or to.
Sure, yeah, and it's, I mean, there's a lot of things that would be really good to look at muscles, but so, yes, another next question.
Yeah, Brian Weedman.
Hey, Brian Weed.
Weedman on.
No, that's the German, good Wiegmann, German, German acrologist, yeah, or about it, biology. Sorry, it's totally different work. Yeah, we go.
Thank you, Sam. Thank you very much. Next speaker is Florencia Montierico. Beyond the Root, are revealing the insect and other invertebrate fauna associated with Systematic in Northeast Argentina. And we should have the pre-recorded presentation, but we also have the poster outside.
Hello everyone, my name is Florencia Monti, extending warm greetings from the northeast of Argentina, placed in the lovely Corrientes province, just a stone throw away from the Iguazu Falls, one of the 7 new natural wonders of the modern world. So I mentioned this geographical context to provide you with a sense of where I'm speaking from. At present, I'm pursuing my PhD in biology en el Laboratorio de Biología de los Invertebrados y Protistas de la Universidad Nacional del Nordeste y en el Centro de Ecología Aplicada al Litoral. Here, my focus revolves around the ecology and biology of protist-ciliates, particularly their role as bioindicators in wetlands ecosystem. So today, I'm delighted to offer you a glimpse into my ongoing research with this presentation titled Beyond the Roots, Unrevealing the Insect and Other Invertebrate Fauna Associated with Systematic in Northeast Argentina. So let's start. Well, I have structured my presentation in 4 main sections. To begin, I will introduce the focal topic. So let's go on an exploration of our research concerning dynamics that exist between invertebrates and plant communities, particularly those featuring post-Systematic communities in rhiophyletic aquatic environment in Northeast Argentina. Well, in the heart of the diverse landscape of Northeast Argentina, my research takes place in the Misiones rainforest. Here we embrace a multidisciplinary approach to understanding the delicate balance and intricate relationship within these ecosystems. The Misiones rainforest hold immense ecological significance due to its exceptional biodiversity and distinctive features. This unique ecosystem is characterized by a plethora of plant and animal species, including endangered species such as the jaguar and tapir, which contribute to the overall health of the environment. One of the standout landscapes of the Misiones rainforest is its intricate network of watercourses, including rivers, streams, and waterfalls. These water bodies play a vital role in sustaining the ecosystem by providing habitat for aquatic species, ensuring water availability for flora and fauna, and contributing to the overall balance of the ecosystem. The waterfalls not only enhance the visual beauty of the rainforest, but also support diverse aquatic ecosystems. In these ecosystems, aquatic macrophytes, as many of you may already know, play a pivotal role in shaping and sustaining aquatic ecosystems. They create microenvironments that foster a rich diversity of organisms, offering fundamental resources for the surrounding fauna. Our exploration today takes place on the unique Postemasias communities in northeast Argentina, which have amazingly adapted to distinct biotopes due to their exposure to water drag forces. It is a unique family of aquatic plants that bloom in fast-flowing water. These plants create microhabitat with specific ecological conditions, forming the foundation for an array of fascinating invertebrate communities. In this environment, abundant growth forms extensive beds that offer refuge, protection against predators, breeding sites, and a source of food for a diverse array of organisms. The various species within this family hold an interesting and lesser-known community of invertebrates that exert a significant effect on the trophic webs within these particular environments. So despite the many of studies on Postemasias in South America, there exists a conspicuous gap in information regarding associated invertebrates, especially insects, in the northeastern Argentine region. So our investigation seeks to address this gap. In Argentina, the few published studies correspond to research conducted in 2 streams in the province of Misiones. In general, diverse studies show a marked predominance of immature stage of insects. So other invertebrates such as sponges, turbellarians, oligochaetes, and mollusks, as well as other arthropods, are also recorded. So understanding the composition and function of the insect community in relation to Podastemasia in this environment will enrich our comprehension of the ecology and dynamics of these aquatic systems. So with this background, the principal objective of this research is to examine into the intricate interplay between invertebrates and Postemasias associated plant communities. Through this exploration of unexplored territory, our goal is to clear up the biodiversity, ecological functions, and conservation importance of these frequently disregarded ecosystems. Within the scope of this investigation lies the specific aim of presenting preliminary findings concerning the diversity of insect and other invertebrate groups linked with Postemasias in the province of Misiones. Moving on to methodology, we explore 3 prominent Postemasias species from the collection of the Herbarium Humboldtianum, which are preserved in 1700 households: Postemon molleri, Tristichia trifaria, and Apinagia iwasuensis. Within the years from 2003 and 2007, we collect samples from 3 distinct locations, forming the foundation of our investigation. Central to our methodology was the identification of the fauna inhabiting this ecosystem. We followed specific literature and taxonomic keys to each group. We examined the specimens using stereoscopic microscopy, ensuring accuracy in our taxonomy assessments. Additionally, a photographic record was carried out, serving as a visual repository of our analysis. Our methodology integrates laboratory analysis and collaboration with experts in plant ecology and entomology. This multidimensional approach enables us to unreveal the complex of relationships that define these unique ecosystems. Well, the results were as follows. The preliminary results reveal a fascinating array of insect life with a notable prevalence of insect larval stages, particularly Dipterans and Trichopterans. Additionally, we observed the presence of adult beetles and the presence of invertebrates such as gastrop mollusks and rotifers. Also, we registered some protist communities. Here you will find a list of insect orders covering both larval and adult stages, alongside accompanying photograph of these organisms, or some of these organisms. Well, but what sets our findings apart is the revelation of galls on Posthemacia, which for the first time in Argentina contain chironomid larvae of purpey. So this introduced a novel element to our existing knowledge and underlines the uniqueness of these ecosystems. While our work is ongoing, preliminary discoveries indicate the rich biodiversity and ecological shades within the invertebrate communities associated with Posthemacia. These findings hold promise for enhancing our understanding of both local and global ecological dynamics. Finally, this investigation not only bridges an information gap in the region, but also highlights the significance of understanding the ecology of these aquatic communities in terms of associated insect diversity. So the complexity and uniqueness of these communities, as shown up by our preliminary findings, provide a valuable foundation for future research aimed at the preservation of this environment and a deeper understanding of their ecology. Understanding the invertebrates associated with plant communities featuring Posthemacia is not only a scientific pursuit, but also carries profound implications for conservation and ecosystem management. So finally, from myself and the entire research team, I extend profound gratitude to the esteemed members of the conference organizing committee to their effort in organizing these remarkable events. So for any further question, please don't hesitate to contact me directly on fmon16@hotmail.com or reach out to our research group at biologia.imp.pro@gmail.com. Also, you can follow us on our Instagram profile, as you can see there. So, thank you very much for your attention, and once again, thank you for your invaluable contribution to the scientific community, and we look forward to the continued success of this conference. Thank you very much.
So we don't have Florencia online, right?
We do.
Oh, okay. Yes. Any questions for Florencia? we have any questions online?
No.
Okay. So we should be on time. First contribution to the Plant B interaction network of El Impenetrable National Park, Chaco, Argentina. And we have the notes, right? And we also have the poster outside the room.
Hi everyone, I am Guillermo Ábalos, a biologist from Universidad Nacional del Nordeste, Argentina, land of the tango, de mate, and dulce de leche. For those who don't know, Argentina, ecologically speaking, is a country that distinguishes itself from its large scale, which houses a total of 16 ecoregions, clearly defined by its unique biogeographic characteristics and living things that are found in each one of them. This heterogeneity has allowed the development of agroindustrial, mining, and fishing operations across our country, as well as the creation of sanctuaries for the protection of wildlife in each region. In this context, national and provincial parks are not only important from the ecological point of view, but also have become widely used tourist attractions. Despite the natural wonders, Argentina has important challenges related to the preservation of species and their habitat. The illegal logging, intentional fire in natural areas, the indiscriminate use of agrochemicals, and the contamination represent some of the threats that several ecosystems must overcome. These problems are also aggravated by the lack of awareness and limited information about the biodiversity in the different ecoregions. A determining factor that contributes to this lack of awareness is the limited access to a few of those areas, extensive dirt roads, and the absence of basic infrastructure such as water or electricity, as well as the distance in connection with communities or towns, are the obstacles that limit the correct study and monitoring of the local biodiversity. With this in mind, I will speak about my work in one of the biggest national parks in Argentina, the National Park El Impenetrable. Located in the north of our country inside of the dry Chaco ecoregion, in the province of Chaco, the park is known for its vast grassland, molded by act of the rivers and for being a biologic corridor between the jungles of Yungas and Paranaense. We can find temporary and permanent water bodies that, along with the traffic condition and historical use of fire, have generated mosaic vegetation, where we can commonly find xerophic and subxerophic forests interlaid with forests in gallery, savannas, and grasslands. Inside the park, we can find a huge diversity of flora and fauna, with a lot of endemic species that, in general, have been poorly studied due to the limited access to it. Among the species of insects of interest in the region, the ones that stand out are the bees. In Argentina, are distributed throughout the country and are an extremely diverse group, with around 1,200 described species spread into 5 families: Andrenidae, Apidae, Coletidae, Halictidae, and Megachilidae. Despite these groups' cultural, economic, and ecological relevance, only a few studies exist that deal with their diversity and ecology in the Grand Chaco. Under these circumstances, the objective of my work is to make the preliminary data known according to the biodiversity of the bees in the impenetrable chaqueño, but also the plants possibly associated with them. For these reasons, samples have been undertaken between the years of 2021 and 2023 inside the park, where different sampling sites were selected, in which some of flowers or water bodies were observed, since those are determinants for the presence of these insects. We made different types of samples on those places to be able to capture the largest number of insects in the least possible amount of time. Those bees with pollen on their body were kept in individual containers for later analysis. At the lab, The bees were classified thanks to the use of keys and bibliography about the bees' diversity in the nootropics. In the case of those with pollen on their body, they were placed on glass slides, cleaned, and the remaining pollen left on the slide was processed with the conventional techniques of palynology and dyed with safranin for this subsequent observation under microscope. The pollen grains were identified using a specific bibliography and the use of virtual palynotecs of the nootropics. After that, we were able to put together a grid named Spectropolínico, and with the use of different statistics indexes, we could observe the plant pollinator network. As a result, we classified 11 bee taxa, from which only one of them was named in the líneas bases de biodiversidad del parque, being 90% of the species here presented new records for the area. According to the pollinic spectrum, only 9 taxa were found with pollen on the body, and we were able to identify 24 pollinic taxa. The resulting network only leaves evidence of the plants associated with these bees. Moreover, it can be observed that during the time of sampling, the bees forage the botanical resources in a differential way. In addition, according to the statistical indexes used to describe the interaction network, the StatiNet tends to present itself as a specialized and cohesive network with low nesting values. It can be appreciated that bee species with bigger number of interactions were Scaptotrigona fukuyensis, Aulochlora, and Tetragonisca febrile, which are catalogued as generalist bees. As for the stingless bee species, like Scaptotrigona fukuyensis and Tetragonisca febrile, they are preferred trees with massive bloom, like Sapionis machofermum, Libidibia parvagensis, Acacia species, and Mitrocarpus hirtus. Another key point is announcing for the first time in Argentina the botanic resources foraged by the bees of the Spinionella genera. On the other hand, the botanic families that had a high number of interactions were Asteraceae and Fabaceae. In conclusion, this interaction network tends to be of the specialized and cohesive type, which is expected from a plant pollinator network. This means that the species are highly interrelated, for which any disturbance in the system could affect significantly to some of the species, resulting in the partial or total loss of some of them. However, there is a lack of information about the impact of Apis mellifera in the territory, and how it influences of the overlap of niches or displacement of a species. For the years to come, we expect to identify, through the samples obtained throughout these years, all bees, wasps, and plants associated with them, to complete the established network up to this moment. Recognize the rest of the pollinating arthropods and their role inside the park, since there already exists a cup of information on other pollinating arthropods and the flora associated with them. Evaluate and establish if the bees respond to the environmental changes and the different use of the ground, and propose action of conservation and sustainable usage of the resources. Make an illustrated guide of the diversity of the social bees and wasps, showing their interaction with the different plants. That can help the spreading and understanding of the relevance of this plant-pollinator relationship. Overall, I would like to thank the Center for Systematic Entomology and the organization of this meeting for giving me the opportunity for displaying the progress of my work, to the Universidad Nacional del Nordeste and the Laboratorio de Biología de los Invertebrados, from which I am part of, to the Secretaría General de Ciencia y Técnica, as well the Picto UNE 2019-00015, for financing this research, but also to the people present here for listening to my presentation. Additionally, I leave my email at your disposal and the last email for any future questions. Finally, I also leave my Instagram in case you like any of the pictures and drawings shown in this presentation. Thank you very much.
We have a nice talk. Any questions? Okay, yes, we have a question.
How many wasps did you collect, and do you have any idea of what tax that you have?
Hi, everyone. I collected to this moment 6 species of wasps.
I have a question. Do you have a reference of all the plants that you are expecting? I mean, the pollens, or is still a work in progress?
Can you repeat, please?
Do you have a reference for all the pollens that of the plants that you found, or is still a work in progress?
It's like half and half. I have the list of the plant of the park, and also I have a pollen reference in the Palinothèque.
Okay, thank you. Thank you,
Thanks.
Guillermo. Any questions?
Thanks.
Okay, thank you. Okay, our next speaker is Matthew Moore. You can run, but you will never escape. A new species of Sylphidae parasitoid of the classical biological control agent, Boreogrypsis meleleuke in Florida. Stage yours.
Thank you.
Great. Okay, thank you. First, I guess I need to acknowledge my Australian co-authors on here. Without their extreme willingness to help us, we would have never resolved this mystery at all. That would be Elena, Bradley, and Matt from CSIRO and University of Adelaide. Matt Hentz, a USDA inspector down south, tipped us off to this. Starting back in 2020, he sent us the original specimens, but he's been sending us more. And then we've got a team at DPI that's moved this forward. So this is a simple talk, but there's three main takeaways. There's a new adventive wasp, which is a hemisphere record that is now established in Florida that is almost certainly affecting a very important legacy biological control program of a terribly noxious weed here in the state. So I'll appeal to anyone in the audience who has interest in biological control or parasitoid ecology. By all means, go study this species in the wild here in Florida, because it needs to be studied probably. Second takeaway is don't give up on really difficult taxonomic questions, because sometimes the help you need is just a well-timed email away. And third, don't ignore predators as adventive or invasive species. I think a lot of times we're really biased towards autophagous insects, but really I think the way that the evidence is showing that predators are moving around the world just as much. So we'll start with some sage wisdom from APHIS, 1998, Ancient History. Introduced organism recognizes no political boundaries, and its introduction needs to be considered on a continental basis. Now, this is in the context of biological control, but I mean, it's absolutely true. There's a bit of hubris involved that we can just pull apart an ecological interaction in one part of the world and put it in another and just be like, oh, it's stable. It will serve us forever. Obviously, this is wrong. Okay, it's not geographically stable. Biological control agents move. Neither is it ecologically stable. It can be disrupted very easily in a new environment and collapse. So to put this new introduction to Florida in context for North America, there's some good work recently that allows us to do this. I mean, it's just one species, it's a drop in a giant bucket. US Geological Service in 2021 published a list, a compendium of what they think are verified introductions into North America. Alaska, 71 species of insects introduced, but Hawaii, most of the damage has been done. Over 3,000 verified introductions to the Hawaiian Islands. In the lower 48, we're looking at about 3,600 possibly introduced. If you remember my talk from last year, there's a paper published now based on that if you want to follow up in Neobiota. But my idea was like, well, let's count these things as molecular operational taxonomic units in databases. And so potentially in North America, there's over 12,000 of these MOTUs that are sort of shared between North America and other continents or island chains. There's no directionality there, but there's many ways to sort of quantify this phenomenon around the globe. So here's our subject of interest. Sylphidae. This genus is a huge problem. It's hyperdiverse. Over 200 species currently described, but there's many, many, many more. Its center of diversity is in Australia, where the legacy biodiversity research is not very helpful in identifying any of the material at all. There was a key to the Palearctic for all insertions, but included Sylphidae. That was Russian language, so not a lot of access. That was recently translated into English, so that's a little bit better. You can kind of work with that now. New World genus is very poorly known. So in 2020, Matt Hentz sent out 50 specimens. This is a female. It's a very handsome wasp, little guy, reared out of Oreocaspis melaleuca, which is a psyllid used as a biological control agent of Melaleuca tree, invasive in South Florida. So this is a very handsome wasp, I'm told, by my Australian colleagues. They said, oh, this is one of the nicest ones we've ever seen. Oh, great. The characters that separate Sylphidae species, coloration of both the body and the appendages, the shape of the antennae, their insertion into the face, cetacean, kind of the arrangement of the ocelli in the eyes. A lot of the old descriptions are fairly superficial. So there's a lot to learn still about the genus. The male is largely the same as the female, except for the antennae are way different. And luckily for us, this antennal morphology of the flagellomeres here, where there's kind of this lobe extension off of them, seems to be rare in the genus. So it kind of sets this apart a little bit and helped us kind of narrow things down in the literature, which was a mess. But here's the system. Melaleuca quinquenervia, it's known as punk tree, paper bark tree, has a long history in a state very, very invasive in the Everglades. So it was estimated at the height of its invasiveness that there was about 400,000 hectares of just monoculture. So this is an image around Lake Okeechobee. All of this dense green here is the same tree, Melaleuca quinquenervia, in kind of a monoculture there. It's very resistant to fire and cutting. And so this was recognized early on. Biological control agents for the plant were aggressively pursued starting in the late 80s. Trees native to Australia, so that's where they looked. There's 4 agents now in the state. A weevil, Oreoglycapsis melaleuca, is a psyllid, and 2 species of Lophodiclosis, gall midges, used to help control the tree. And it's been very successful. Aggressive removal of the trees plus deployment of all these biological control agents has really knocked the tree down. It's really worked, which is why it's alarming that something could be disrupting the program. So the psyllid itself, ecology and host range were first described in 1997. Very well, it seems to complete its development really only on Melaleuca quinquenervia. There's some oviposition on other Melaleucas, but there doesn't seem to be any survival of the nymphs. Importantly, the nymphs cause the most damage to saplings, young trees. So that's kind of their role in the biological control suite. It's like it hammers young trees. Heavy infestations can kill them. It was first released in Florida in 2002, and it subsequently spread to 22 counties or something like that. So the wasp, the Sylphidae, these are images sent to us from Matt Hentz, the USDA inspector. That's a healthy nymph here. And these show parasitized nymphs. And then these arrows, so it's kind of grainy, but you can see the exit holes there and nymphs. And you notice this, and he sent us the wasps. And we are absolutely dead in the water trying to identify the thing. We got a bunch of sequence data, didn't match anything. You're scraping through the literature, could come to no firm conclusions. Well, lucky for us, we had the bright idea, okay, it's an Australian tree, an Australian psyllid. What's the chance that wasp is Australian, right? Let's be real here. Sure, it could have been a native thing that jumped over. There's precedent for that. But we thought kind of that's our null hypothesis. The wasp is Australian. So we reached out to CSIRO, which is the original group involved with doing the ecological studies on the biocontrol agent. University of Adelaide, luckily for us, there's a PhD student now working on the genus in Australia. Woo! Because otherwise, we would have had no chance. And then Queensland Museum, Chris Burwell, may have seen these wasps back in the 90s. There is a reference in the literature to, oh, we reared a Silophagus out of these. We sent them for ID. There was no ID at the time. We wrote him and said, hey, have you seen these wasps? That didn't go anywhere, but it's okay. We reached out to CSIRO just through an email and said, hey, can you rear some wasps out of Boreoclacaspis on Melaleuca? And they said, oh, we got that right outside the front door. Yeah, we'll go get some and we'll see what comes out. And then a couple months later, they sent us these pictures, just taken through a microscope camera. We were very excited because what are the chances that they would just go out their door and collect the wasp like that? It seemed very improbable at the time. And we were like, oh, these are dead ringers. These are the same wasps. It's gotta be, right? It's wild. So Elena now is handling the description of these and comparing the material from across the world. And we're pretty convinced now that, yeah, these are the same species. We did a small genetic comparison, ribosomal data and mitochondrial data from across the basically opposite ends of the world. Ribosomal data are all the same. They're different mitochondrial haplotypes, but there's not that much variation there. We're fairly convinced that we have somehow an introduction of this wasp subsequently into Florida. The type series is being assembled now and the description is nearly complete. And we're going to try to share specimens across various places where we know the tree occurs and where the psyllid occurs, so people can kind of be ready for this thing to show up. Susan Halbert has been identifying psyllids from traps in South Florida for many, many years. And one of the things she's been counting are numbers of Melaleuca, or excuse me, Boreoclacaspis melaleucae in these traps, various traps in Winter Haven, Immokalee, and Miami, going all the way back to 2003. But biocontrol agent populations wax and wane in every single way that natural populations do. So to see them go up and down isn't a shock, right? But we see a troubling thing, which is that in the traps, we're not collecting very many Boreoclacaspis melaleucae anymore, which makes sense because there's way less of the tree, right? However, the introduction of the wasp, a parasitoid of the psyllid around this time, probably indicates that those numbers are never going to go up again. At least that would be my prediction. So it's possible that this aspect of the biocontrol program just might never recover. How did this occur? How did the wasp get to Florida? This is a great mystery. The tree has a long history here, dating back to the late 19th century. It was imported into nurseries, kind of across the coast of Florida. Some settlements used it for ornamental purposes, quickly escaped cultivation, and they knew it was a problem by the 20s. They're like, oh yeah, this tree's not going away, it's growing everywhere. Became invasive in Florida. The tree is also present in California, Louisiana, Texas, Caribbean, South Africa. It's not very invasive in those places. It seems to be the Florida Everglades are just the perfect habitat for the tree. The psyllid was introduced in 2002, mysteriously showed up in California in 2009, Puerto Rico in 2006. It's not really explained how that happened. And then now we have a wasp in 2020. So clearly, this system hasn't been stable since the start. The biocontrol agent's been moving all over this side of the world. And I'll end with this. This is still a mystery how that's occurring, but there is a connection, I think, a weird one. We'll throw it out there. Eucalyptus, kind of a similar situation to Melaleuca. They're both Australian rhotaceous trees. There's been some griping that possibly eucalyptus in California may have been subject to some vigilante biological control. Basically, people unhappy about eucalyptus being introduced and propagated in California intentionally introduced a bunch of pests of eucalyptus into the state. Evidence for this is scant, except for that the accumulation of pests has sort of defied models of how pests accumulate. So when you have just, 15 new pests of eucalyptus show up in a short period of time, you're like, oh, somebody did that on purpose, basically. There were some other features about those introductions that didn't make sense. They didn't have any parasites at the time. So you would expect the pests to have parasites, but they were clean. And then a bunch of Silophagus have now started being collected in Australia, or in California, excuse me, that were not intentionally introduced there. And there's probably more that are undescribed and uncertain research is being worked out. But it makes me wonder is if the sharing of the psyllid in California turned up there 7 years after release in Florida, wonder if it was done on purpose too. And could that be how we're getting Silophagus in the New World attacking Boreal black aspis? I don't know. I don't have any evidence for that, but it's one of the ways. The colony could have not been clean to start with. That doesn't seem probable. Melaleuca is strictly prohibited from importation anymore. So somebody could have illegally moved a plant. Okay, but from Australia? It's kind of hard to smuggle. I don't know. There's a bunch of features to this that don't make a lot of sense. We're still trying to come up with scenarios about how it occurred. And so with that, I'll end my talk and open it up for questions. Thanks.
Thank you.
Yeah.
How many specimens do you have of the wasp right now so far collected?
We have a few vials from St. Lucie County and Palm Beach. Maybe 70 or 80
So do you
total.
think they're established sort of?
Yes,
Okay.
because we got subsequent collections year after year now. Unfortunately. Yeah.
Any chance of hyperparasitoids with your Australian colleague?
Hyperparasitoids of,
Parasitoids of the uncertainty?
they haven't mentioned anything yet. So no, the answer is no. Don't know.
I'm collecting eggs of the wasps of the city.
Yes, good.
My married.
Yeah.
No more questions? Thank you. Our next presenter is John Livingood. Studies in the New World tribe Anisochelini, Heteroptera coraidi.
John. Thank you.
All right, so I added a little silly subtitle, nothing clever about it, a 10-year journey. There's a lot to that. This isn't the kind of project that takes 10 years. It took me 10 years. It's not for the reasons you normally think a taxonomic project should take 10 years. But with that, I'd also like to add that I'm going to tell a little bit about my journey that led me to this 10-year journey as well. And that's my little story that I want to tell. So a lot of years sitting out here, John's talking about true bugs. Depending on how you know John, you think that, John, aren't you a beetle guy? The answer is, yeah, most of the time, maybe 90% of the time in my head and 70% of my time at work. But true bugs have kind of infiltrated like an invasive thing in my mind, and they've held and established. And now I spend a lot of my time actually thinking about two orders of insects, even on a research basis. Now, I should say that rather shamefully, there is a person in this audience who co-organized this symposium, who spends his weeks day in, day out, working on only one order of insects, and that's Davidae. He works on Hymenoptera all the time. And I say for shame, because Massimiliano Spinola, a fellow Italian, worked not only on Hymenoptera, but even more on true bugs and yet more on beetles, specifically checkered beetles. So I feel that any stigma of John, what the hell are you doing spending all this time on true bugs is unfounded. There's precedent from 1844 when this guy dropped 2 books on clarids, and it is still the largest single work on clarids ever composed by anyone published at one time. I'd also like to take a moment to directly thank Weston Opitz, who gifted me his copy of this last year, and I didn't want to thank him until it was in person. And this felt like the right way to do it. This book isn't only rare because it's from 1844 and an original printing. It's rare, it has its own kind of conspiracy story. But this book is even more rare than other books of its time in nature because someone was trying to drive up the price of these books for personal gain. So I have that, and now Weston, after hearing that, you have to sit through a Korean talk. And
Glad to hear it.
And you're gonna hear my story of why. And everyone else in this audience who just happens to be here will have to suffer through it because they showed up, and it's rude to get up now, you have to hear it. So back in 2010, this guy who looks like my son, but I swear it's actually me, went to Costa Rica. He went to Costa Rica for an organization of tropical studies class called the Biodiversity of Heteroptera. This was led by big names, Cristiano Wierach and Toby Shu. On this course, there were a lot of people who were up and comers who today are big names, like Michael Forthman. Oh, look, I have a pointer, don't I? Michael Forthman, Wei Song Huang over in Singapore now, Laura Renio-Correo and Dimitri Ferrero, both in Colombia now, and Cristiano Feldman-Shortner, who was really like my conference buddy for a long time after this, who's now in Sao Paulo. He was a student of Joselia Gracia, and now he's in one of the leading pentatomoid labs in the New World. So I got to know all these people. Now, what's funny is that I got permits for myself that I arranged separate from this class for all insect collecting. The course provided one just for true bugs. I just thought I'd give it a shot. I got it the day before I went to Costa Rica. And so I'm walking around collecting beetles, and everyone on this course from day one is saying, we're not allowed to do that. I said, no, I have permits. And then they just got the idea that John just wanted to collect beetles on a course with an itinerary, and all these other people who can't collect beetles will give it to them. But within a couple days, and it wasn't totally wrong, but in a couple days, they realized that I could talk the talk a bit, because about 10 years before that trip from DPI, I got my free books when I became a research associate. And among them were the Lygeidae, now functionally the Lygioidea of Florida, and the Charaidea of Florida. And I got pretty good at them, that just because you catch a bunch of stuff. And when you have keys to things, you try to identify those things. When you don't have keys and you're, green to the field, you're not going through descriptions, you're using what you have. So it was scarabs and these at the time. So I could say, hey, in the field, that looks like this genus of Charaidea. That looks like that tribe of true bug. I, as a perfectly palatable coleopterist, was able to blend in with the unpalatable heteropterist. I was a Batesian mimic. We all know what that is now because that was explained in the weird Lepidoptera behavior talk. So now I know you're thinking, John, like you are the perfect emissary and acolyte for Coleoptera to go into the true bug nest and kill the queen. I didn't do it. I didn't. It took everything in me. They proselytized me. It wasn't fair. I gave into the dark side. And I actually got invited to teach a course on family identification later that same year in Heteroptera in Grenada. And this was to agriculturists and entomologists from all over the Caribbean, a few from Northern South America. This is because Wierach and Shu couldn't do it. They recommended me. Thanks. I wonder how many people had to turn that down for it to come to me. They gave me all of their family and morphology lectures. And still, I knew I needed help. I mean, I'm a graduate student. I haven't done anything. So I recruited Susan Halbert. She's right there. And she really saved me on this. I don't know if she realizes it or realized it then, but I could talk about families in and out. I was an ace at a key. I was already the guy in my department that everyone went to, usually instead of the systematics professor, of what family this is, not what species. Susan is the person who knows the pests, knows what's spreading where, knows what host they're on, knows about plant pathogens affecting crops. I know none of this, and I knew that would matter. And thank God she joined me. She really saved me. So thank you, Susan, yet again, 14 years later. So I'm getting into true bug. Somehow I'm making a name for myself, even though I'm already about 10 years into checkered beetles. And then I get hired by the USDA. We noticed that I'm getting a little bigger. The hair is going away, a little grayer. We're starting to slowly morph into present day. So when you become an identifier with the USDA, it's long been a standard that there's this training that you go through to have a certain pay grade jump. They call it an advancement plan. And with that, you pick a taxonomic specialty. Some identifiers will embrace that and run with it on a avocational almost interest level and not publish. Some will run with it and publish. They may have even studied this in grad school. For some people, it's just a mechanism to learn a new group and learn how it is to get into the literature and dive deep into a group, because maybe all they ever did was take an insect taxonomy course. But whatever the case, I had to pick one. So I said, how about cerambycity? I quite like them. I catch a lot of them catching clarids. They said, no, we have steep-winged culture. I said scarabs. They said, no, we have a policy. They're easy to deal with. I said click beetles. They said, nah, it's not really a big enough problem. Okay. Bastrichids, scelidids, general weevils. No, 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 for various reasons. Some I agreed with, some I was bothered by, but that's okay. I said, all right, how about Lygioidea? They said, no, it's not a big enough problem. But okay. Cydnids, Pentatomidae, still no. Do you hear how many things I'm saying? So people were already doing this, or it wasn't a big priority based on interception data, I suppose. I got to Coraids. They said not a big enough group. So it's Choroidea and Pyraloidea. I only know about Coraids. I just want that on the record. Don't call me about any of these other groups. So I picked that, and picking that, having never published anything on true bugs at all, I want to do something with this. I like publishing papers. I like projects, a tiny bit ego, a tiny bit legacy, a little bit of its love of the game. But if I have to learn this, I want to do something with it. So I set some goals. I want to do a key to the genera of the tribe Acanthocephalaeni plus Placosolini. That's a contested status, whether or not it's a part of that tribe. I want to do the same with Anisocelaeni plus Leptosolini, same situation. And by the way, that would mean making keys to 1/3 of the New World Coraids. The goal of the guy who just now is, I guess I'm going to do more than of Florida when I work on true bugs. So we're going to call these some pretty big dreams. And okay, well, either way, I set these goals, I'm going to do it. I know it's going to take me a really, really, really long time. By the way, I have a job that's not a research job. A lot of my spare time when I have it is already going to checker beetles, and I'm not favoring them over that. So this is getting about 1/10 of my time. That might explain the 10 years. I arranged some training trips to get to learn these things. A couple of trips to Texas A&M, a couple to the Smithsonian, getting to know people, exhaustively going through the material. And I found Anisocellus affinis in the Sabal Palm Grove in both the Smithsonian and Texas A&M's material. There's specimens going back to the 70s. How do we not know this is here? This is one of the coolest new country records that I could find in Coraidae. And I was excited. I added it to a manuscript draft. And then 2016, this paper comes out. And it's a key to the Anisocellus. And among them were distributional records with the exact label data verbatim that I wrote down at those museums. Culprit was Harry Brylovsky. Very nice man in reality, but at the time I was very annoyed. Very annoyed. So, okay, we gotta just move on with our little, part-time Coraid life. Another trip to the Smithsonian. This was the last, this project trip, I suppose I had to the Smithsonian. You see here, there's a lot going on in the margins, the track changes of my working key. I had working keys from my first visit in 2014. Couldn't use them very well, but a lot of notes. I couldn't see about 1/3 of the genera of these respective tribes because they were described by, wait for it, Harry Brylovsky. And they were usually described on bases of 1 to 3 species that each had 1 to 2 specimens that were either in his collection or spread around Vienna, Paris, Bucharest, wherever. And since I'm not a productive heteropterist yet, I don't want to pester someone for all these photos and say, no, I can't quite see it. Other side, how about another set of photos? And then have them wait and say, John, did you do anything yet? Did you do anything yet? Can you thank me yet? Can you thank the museum? And the answer is going to be no, because I'm on a 10th time basis here. But I'm doing the circuit. I'm going to the International Heteroptera Society meetings in DC, in Argentina, in Spain. I'm meeting people, I'm telling people what I'm doing. Everyone's saying, that's great. Do you know Harry? I said, well, I've emailed him a few times. By the way, there's Joe hanging out, and there's Julieta. I don't know why I don't like using the laser printer pointer. But so either way, I'm really getting deeply invested in this whole Heteroptera community deal. And eventually I published a small paper with a whole lot of new country records. It's a big deal to me. I realized that Harry Brylovsky has published more revisions than the number of country records I have in this paper, but still, and around this time I wanted to give a talk based on my frustrations called A Perilous Journey, Lessons Learned in Trying to Construct Keys to the New World, Acanthocephalaeni and Anisocelaeni. got canceled because of COVID, but again, I'm struggling with all these genera I can't see. I keep hoping I'm gonna find them in a museum loan, identify it myself. Never happened with any of these, like 16 genera across these two tribes. And then this happened. Illustrated key to the genera of Acanthocephaline, skip to the end, key to the genera of Placoceline. Harry scoops me again. And this is funny because some people could say, John, why didn't you ever ask him what he was working on? Harry is a describer and a reviser or reviewer of genera. He had to date never in over 100 Coreid papers published a paper anything like this. A key to a whole bunch of genera of a tribe he had never done. So I didn't expect that. So maybe my own fault, but again, I'm afraid to reach out sometimes. So just a reminder, that's those tribes. So we can just simplify my goals. My dreams are a little more manageable now. I met Harry a little later and I was saying to him, Harry, this is in Spain. I wanted to do these keys to the tribes, and I was working on them for a really long time. I had keys made. Whoops, how do I go back? There we go. I had keys made, and then you did a key. And he says, oh gosh, I didn't know you were working on it. Yeah, I know. I know. So right now I'm still working on and have been for a really long time at Isocline. He says, I'm going to do that next. I said, okay, well, look, I've already spent a lot of time and I have this functional key. It has a lot of problems because the genera I haven't seen. Would you look at it and let me know if maybe we could do like somehow do our own kind of aspects of this? And he said, why don't we just work on it together? I wanted to give that man a hug so bad. Because to me, like I'm Frodo and he's like Gandalf, you know, like I don't know anything about anything and he was all about this world, right? Everyone didn't laugh last time. So I'm like Luke Skywalker to the people who didn't get that. And he's like Obi-Wan. Like I'm Skywalker back when he was a moisture farmer on Alderaan and didn't know anything about the Force. Is that okay? I think I got almost everybody. I'm happy with that. So there's Michael Forthman too. He's going to come into play a little bit on the left. Michael Forthman actually worked at DPI and over at UF for a bit. But Harry and I set some goals. These are the typical goals. Well-illustrated key, illustrate every genus. One of the problems that I had was that a lot of these genera Harry described were back before we had good photography. They were a grainy black and white photo or some line drawings of a parameer. I couldn't do a lot with it. And I could extrapolate a bit from his diagnoses and descriptions, but then I look at all these other things. Like, I guess I could make that this genus, but that's not this. So I really needed him, it turns out, and I really should have reached out a lot earlier. We were going to publish loads of new country records. I just like that. It's fun. And whatever else we stumble across along the way. But there was a big problem. And it's a problem that almost everyone faces with almost any big taxonomic group with a lot of problems in it. What exactly is an Isocline? We've seen talks today on Silapinae in the Miridae. We've seen talks today that assess all of Reduviidae. We saw what happened with Reduviidae. That's like 86 tribes. So this is another such problem because Michael Forthman did this phylogenomics project on Coreinae. And even though it's just one subfamily, for you guys who aren't in the know, that's like 88% of Coreinae. And here is a rather ill-sampled, but still, an Isocline. It's coming out in two places. By the way, those two places are not Lepidoptera and Isocline, they're both and both. So if we sampled more, I don't necessarily think it's going to pull it together. It might just be yet another clade. Hemiptera, very speciose group, that's where that's coming out. Here's two more tribes being rendered paraphyletic, and I'm just picking some of the highlights. What's more is that a year before this, he actually did a smaller analysis and included Elydidae. And back when he did that, he found that Elydidae rendered Coreids paraphyletic. So now we have to ask ourselves, what's a Coreid? What's a Coreine? And what's an anisocline? So what do you do when this problem comes about? If you're a grad student, you think, gosh, John, this is the end of it. If you've been in taxonomy for 20 years, you're like, yeah, you just keep going. Because what are you going to do? No diagnostic resources ever until we fix this problem? No, we weren't waiting around for the high proselytizer of genomics to fix taxonomy for us. We had to go do the dirty work with morphology. So we're going to move on anyway. And still, we do have to ask ourselves, what is an Isocline? I wanted to say plus or including Lepidoptera because Harry Barlowski would say those are two tribes. Rick Bukowskis would say, I synonymized that in 2008. It's not a tribe, and Harry didn't re-elevate it. So to not bother anyone, I chose this verbiage, and that's the verbiage that's right now in the Zootaxa proof that got accepted. So here's couplet one of the key. This is just how you separate these two groups. Again, these are not the two natural groups in Orthman et al.'s phylogeny, unfortunately. They're basically separated by whether or not they have simple, all on the left, simple hind TB, or some kind of flattened long ridge or foliation. This is the only guy on the right who has simple. So that's these two groups. By the way, I was mostly the key guy. I seem to be really good at ferreting out problems with keys. And Harry is the guy who knows these things so well that he's not looking as closely as he should, I think, sometimes. So I kept coming to him fixing the keys, but I also had to keep coming to him saying, Harry, I don't know what these things look like. Say, John, here's a picture. Thank you. Wish I had that 20 years ago. So Bellaminochorus and Ugneus, here are two weirdos on the left. We included them because Bukowskis moved them to Anisocylinae. Previously, they were in Acanthocephalinae and Coriinae, very different tribes. On the bottom, we have Amblyomia. Same paper, Bukowskis kicked that out of Anisocylinae, but despite that, that event, he didn't put it anywhere. It's in Syrphidae. So we decided to include it, or at least that was what I wanted to do because of so much literature historically associated with Anisocylinae. I just want it there for everyone who isn't looking at every little paper. Bukowskis' 2008 paper where he did this is actually a rather small paper where he just threw in some synonymies. There's no taxonomy in it, if you will, for the non-Korean specialists to want to seek it out for an identification resource. I want this to be accessible. I want this to be doable. And I also don't want to use the really hard technical characteristics that Harry wanted to use in the key. I wanted them to be things that are approachable by someone who just has a good grasp on Heteroptera. Mamurius, there's another weirdo, Mictini, Metapodini, Acanthocephalinae. These are some very different tribes it was associated with. I think, by the way, that Wallner, for a long time with his Brazilian colleagues, was testing the monophyly of Nematopodini. If I remember that correctly, forgive me if I'm wrong, but if he is, I'd be very curious how many clades that's forming or how that's rendered paraphyletic by things. We synonymized two genera. I was pretty proud of this. This was a suspicion of mine. I thought either I've nailed it or I just have one species in front of me and not the other one, and I just can't tell the difference. So we sunk Belanoma sinulated which was this monotypic critter, into Malvana, which only had 3 species, one of which is very, well, more common. The other is very elusive. There were like no good specimens of this critter. Just the pictures you see took 3 specimens to provide. And even though there were a lot of recently collected specimens, this taxon is cursed. It's like how legs just fall off a Miridae or a Typulid when you look at them. We gave a complete distribution, et cetera. It wasn't so exhaustive on the systematic side, though. This was just more about cleaning things up for a key. We resurrected the genus Bita. That was Harry's baby. He really wanted to do that. He treated that as a subgenus back when he scooped me in that Anisocylus paper back in 2016. And so we re-elevated some names, yanked something out of synonymy. On the right is Anisocylus, on the left is Bita. I think there are really fewer species than those pictures are suggesting, but that wasn't my war. I also noticed a few issues with his old treatment of Anisocylus, and he acknowledged that some of those things were problematic enough that he just wanted to redo the key. So here's couplet 1 of that key, and that's why this paper is a key to the genera of Anisocylinae, yada, 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 skip to the end, and the key to the species of Anisocylus and Bita is in the title of this paper. So it made it a little more substantial. Here are some more genera that snuck their way in for a total of 29 genera and Anisocylinae plus Lepidoptera. There are some weird looking critters here. Like this guy here, don't know what's going on, but no one has ever contested its placement since Harry described it. Over here, pretty flashy compared to some of these other guys. But again, there's a lot going on here. It's not so surprising this thing isn't necessarily a monophyletic group, maybe even more clades than Forthman had dug up. We found 90 new country records between us for 44 species. Each of those species gets an exhaustively explained distribution. We're not just saying this is a new country record and leaving it on you to know where else the thing occurs. We also found some informally published data. from students who had gotten a grant from the Hemiptera Reserve Society. And you see the screenshot here. This is actually just identifying the people who got the award and what fiscal quarter it was, basically. It doesn't say anything about the taxa. There's no place on the website to search for taxa. I luckily found this on Google Scholar. And it's just a PDF with pictures and names a lot of times and a very brief write-up. But these things are the kinds of things that would typically get overlooked when you're assembling literature or a distribution, and we want these students to get some credit for it. Same thing goes with these field guides on the Field Museum website. Some of these are as simple as the lichens at this Eco Lodge property. Other times they're the birds of this province of Peru. But again, they're not exhaustively vetted, but they're still nice products. So we included those. Little field time gave me some fresh specimens to look at. They didn't provide a whole lot of new records for me for the papers, but they gave me good material to work with for photography for this paper. And just to familiarize myself, it's a little more exciting when it's your own specimen. There's Joe and I having a car trouble in Paraguay. Some of you may recognize Gareth Powell when he worked here. He just left in November for North Carolina, a professorship. There's an Anisocellus right there. And there's one of the Acanthocephaline in a paper I'm not on because I got scooped. This was just Diactor bilineatus. It's just a cool thing. It only hung out. I spent 2 weeks on this property on these plants that were overgrowing the showers. And they were only on the inside at night, moon bathing on the wet leaves in the shower area. Just a cool aside. I did not find one of these anywhere else. I found 8 of these showering over the course of 2 weeks. It's really awkward when you're naked and you don't have a Snapchat file. And you're like, and I have a small towel. I don't know if I can hold the towel and walk back holding the thing. And it's like trying to kick its legs. You're worried it's gonna break off its delicate legs. And no, and you let it go or drop your towel. But it's remote proof, so there's no one there, so it's fine. You're still embarrassed, but it's fine. And about 8, 9, 10 days ago, I got this proof. And this is for our paper. This is right now 2 months away from 10 years to the day of my first trip for this advancement plan project. And again, that's not a requirement that we do this project. This is just what I wanted to put into it. This was my heart and soul, this project. And again, we've got the key to the Trinity of the tribe. And so I got a nice little and in there. Little jab back at Anisocellus. Replaces that other paper. And Harry Brolowski's a friend now. And our interactions are so different now. Instead of, hey, do you know what this is based on a photo? And I'll say, sure, here it is. And I don't really know if I'm bothering him or not. Now he's just excited about what we're going to do next. I'm like, slow down. This was 10 years of my life. And I realize you're not going to scoop me anymore, but yeah, it's very exciting though, when you make a friend out of these projects, even if it's almost entirely over email and briefly meeting at a conference. So I got to be very grateful to my past and present supervisors in the USDA who humored these museum trips as training for a new identifier. But I did work with some other people like Tom Henry and got some guidance from Heteroptera along the way. This is not something that I get to continue doing. This is not our job. Again, it's not a research job. It's not a museum job. I do like my job, but this is my spare time. My brethren of the net who helped me in the field, who helped me find stuff and go on adventures with me. So thanks, Joe, for the one who's here, and all the people who helped me out on museum visits. And that's all I got. Thanks for your attention. Any questions? Take a little coffee.
Questions?
Probably not, because that's just like totally resolved now.
Lots of pictures.
Yes.
All good?
Now I have a question.
Okay.
Do you prefer beetles or?
Beetles.
Okay. Thank you.
Yep, Susan?
Joe, what are those leaf feet used for? They do.
So there are a few things, and so Miller at UF, some of her phylogenetics work is looking more into like behavioral aspects, and that's what Portman was doing with her. So some of it is actually just defensive, where then those legs come off easily. It's like a lizard tail in some taxa, but not all taxa in their respective clades. So sometimes they kick with it and it'll come off. So they're going to try and deter the predator If it's not working, it'll break off and then predator might look at the flashy, foliate leg. There's a little bit of mate competition involved in things like that, but usually that's more in the acanthocephalines, which have much stronger foliations in their hind legs, where they're just kind of rear boxing at each other, like donkey kicking. But that's the extent that I could say of it. It's not like a nuptial presentation. They're not like, you'd want to hope they're like birds of paradise or a peacock spider, but none of that.
Is there any aposematism involved in it?
So, yes, I don't think, well, I might have had one picture up very briefly, but there is a genus Hemiptera, where if you were to look up that genus, that's just about the one in this whole group where the Hemiptera are clear, even though they have some blackening over the cell margins. And so their wings are clear, and their coloration, it looks a lot like ichneumonides in the area. So, and different species of Hemiptera actually will look like different ichneumonides, even with a little bit of like white and black alternations on the antennae, yellowish or reddish versus white and black from the dorsal coloration, bright red or bright yellow abdomen. So that genus especially has even been cited as kind of a probable mimic there. But I'd say it's mostly that genus. So the others, other than maybe Amblyomia, the one I pointed to, which has that pastel bright orange and yellow.
Awesome. Thank you, John. Our next presenter is Michael Bernal, the broad-nosed weevils of Florida. The stage is yours.
Thank you, thank you.
Can you send me your slide so you can take a picture?
Oh, sure, sure.
Thank you.
All right, good afternoon, everybody. Like she just said, my name is Michael Bernal. I'm a graduate student at the University of Florida. I'm here to talk more on the progress I've made on the broad-nosed weevils of Florida, which is the focus of my thesis. So broad-nosed weevils are a subfamily of Herculeonidae, which, as some of you might know, is considered the largest animal family in the world, with 62,000 described and an estimated 220,000 extant species. And that includes undescribed taxa. In North America, there's an estimated 2,500 species across 19 subfamilies of 480 genera. This is just a diversity snapshot of the entomines you can currently find here in Florida. So as I said, Herculeonidae, numerous known pests. They are mostly phytophagous with a narrow host plant range. Sometimes this makes them a little easier to find for research purposes. So that considered, you may be asking, why entomines? Entomines are polyphagous, so they are actually thought to be generalist feeders. They will eat on any plant that they deem fit, which really makes them a pest of many crops of interest, such as ornamentals, trees of forestry interest, or even just found feeding on flowers like this little guy here. Enough of them have gathered enough attention, caused enough problems that they've been added to what I like to refer to as the IFAS's most wanted list, which is the featured creature page. This is just 3 examples of some that you can find on there. As some of you may recognize, Diaprepes abbreviatus, it's a big baddie right now. So these are the objectives for my thesis. They range from compiling all literature to providing county-based distribution maps for each species, providing updated standard differential diagnoses, images for each species, and a key to the genera and species of Entomomia florida. This is an example of the 2 descriptions you can really kind of find right now for a lot of entomines. You either have a really old one that's only a couple lines long and can be misleading once it's translated from its original language. And then you have a more recent, this is from, I believe, the late 70s, that is very detailed. I want to make something that's kind of in between, because I'm not re-describing all these species. I want to make them easier to identify and separate out from other species that they could be confused with. Right now, there's no literature that just focuses on the Entomology of Florida. The two closest things we can use right now are the Weevils of South Carolina, which is one in a series of 9 books that is it has some issues. It was made by a non-legal taxonomist, and the other books that she actually was a part of in this don't even cover weevils. So it's likely that any issues that were discovered and added in this book, they won't be circling back to try to fix them. The other one on the left here, we have American beetles, which it's an amazing resource, but it really is more for the established beetle taxonomists. This is just the last shot of the key to the genera of Entomini, and this is all of North America. So that right there is 123 couplets covering 124 genera, and that's assuming each one only keys out once. So as I mentioned before, I want to provide photos for each species, comprehensive photo sets. As I have here, I've got the lateral habitus, the dorsal habitus, and a shot of its mandibles. The reason why I kind of want to include the mandibles is there's an interesting feature right here that you can't really see here, but you can see better here. In Entomini, there is a characteristic that does not occur across any other part of Crocodiliontidae, which are when they emerge out of the ground as adults, they have these gnarly looking deciduous teeth. The overall theory on this is that they use these to break out of the pupal cases and crawl out of the ground, but then shortly after they emerge, they just fall off and they're left with scars. Just side note, going through over 4,000 species, over 4,000 specimens of entomines that I got from the FSCA. It wasn't until last week that I came across that one right there, which is Syrphidae castaneus with its teeth. Needless to say, I was very excited in the lab when I found it. I let out a little scream. Luckily, my colleague John was not there, so I didn't totally embarrass myself. So another example as to why I want to provide comprehensive photos. These are 3 separate specimens of Mylostra undesiculatus, the Sri Lankan weevil. You can see there's color variation that has to do with how many of its scales are rubbed off, and if you can really see that black integument underneath. This species gets misidentified pretty often with Artipus floridanus, which is a non-inventive species here in Florida. And here's another example of Artipus floridanus from the same shot from that dorsal habitus. There's a lot of differences that separate out these two, but to the lesser trained entomologist, it's an easy misidentification. Thus far, I have visited 4 different collections. The first one I went to was the FSCA here in Gainesville. I went over to the UCFC over in Orlando. And then recently, back in September, I got to visit the Canadian Museum of Nature in Ontario, Canada, and the Canadian National Collection of Insects, which is also in Ontario. During my first visit to the FSCA, I got to meet Dr. Bob Anderson, a fellow weevil taxonomist, which was exciting. Don't find a lot of them near me, at least. And then I got to have an early meeting with one of my committee members, Paul Skelly. And as you can see here, you can gauge how long the two of us have respectively worked for the US government based on the size of our coffee receptacles. I here with my 3.5 years with a tall, if you will, from Starbucks, and Paul with an entire coffee pot to indicate his decades of servitude. Thank you, Paul. So I mentioned I went to the UCFC. I got to meet Sean Kelly. UCFC, although it's a smaller collection, important to visit because they do have specimens from the scrubbier habitats of Florida that are usually just harder to collect. My visit to the CMSC back in September, I was hoping it would be colder. It was actually surprisingly hot for Canada. But I got to go through all of their drawers, work alongside Bob Anderson. And then I also got to meet, to be honest, Rolf over here. That was on our last day before I was heading home. Then during that same visit, I got to go to the CNC, as I mentioned. And during my time there, going through their drawers and copying down all the label data for specimens they had for Florida entomines, I came across Naupactus minor, which up until then, 8 months into my thesis, I had not laid eyes on a specimen yet. So that was another exciting moment. Just a snapshot of the data that I am recording. It's verbatim label data that's covering the county, any locality notes, who the collector was, time of year. I'm just copying down all this data, hopefully looking for any consistencies or trends that I can find. I've been doing field work as well to collect in an effort to collect fresh specimens. That's a whole nother story. So I'm currently involved in a project collecting at the Chinsagut Wildlife Preserve. And in addition to my responsibilities with that project, I'm also running barrier pitfall traps, as you can see down here. We're setting up light traps, sweeping in fields with various vegetation, collecting at sheets at night and running malaise traps. And then in addition to all of that, I'm also doing leaf litter sampling. And this is just an example of the barrier pitfall trap here with me for reference of size, where we incorporated decaying logs to help divert more specimens to the collection receptacles. This is a little snapshot of some of the work I've done. So luckily for me, right after I started my thesis, Brian Riley's dissertation was published on the redescription of Pacnius. And kind of boiling down it's massive. It's not approachable for anybody who doesn't already know weevils. So I was able to boil down the information to essentially be able to identify the 3 Pacnius that currently occur in Florida. So Pacnius lightus over here on the top left has these forward-facing margin, these angles on the humeri, whereas in the other 2 it's very rounded and smooth. And then to separate out Azurescens from Opalis, it's more rostral characteristics. So in Opalis, it's a shorter rostrum. It's kind of hard to see here, but it's got several carinae, and it's not as round or nearly as long as it is in Azurescens. So Azurescens, it's just more rounded. There's no carinae. So that was kind of, I spent a lot of time going through that dissertation to kind of figure that information out. As I'm sure a lot of you are familiar with the Ligiidae of Florida, or at least some of you are, or the scarab beetles of Florida. These are kind of like my goals of what I want to create. So I want to create a product that's approachable to both the average entomologist, but also trustworthy enough for the more experienced taxonomists that they would feel comfortable using it or confident using it. Like I said, we've got the county-based distribution maps, taxonomic notes, and then, where they have drawings is where I want to incorporate these high-res, these photos for each one. I'd like to thank all my committee members, Nicole Quinn, John Levengood, Dr. Paul Skelly. Robert Anderson has been a great help for me with general questions and also during my time up in the CMNC. And I especially needed to shout out to my very supportive and patient wife and my children who have been very understanding in all the work I've had to take away, come to Gainesville to present. And they've just been nothing but supportive in this project. So. Questions?
You mentioned that this particular group is mainly Polyphagus. Are you including the list of host plants?
Yeah, that is part of the label data that I am collecting. The known host plants, and then also any host plants that they were found feeding on, or sometimes even just collected on, just so I'm really covering all my bases. Yes.
How is easy that the scales on Purpurgonia is right 90?
Say that again?
How easy that the scales on your group is falling off because of collecting maybe ethanol?
I haven't noticed them rubbing off a whole lot. Unfortunately, I haven't been able to catch a whole lot of them. A lot of what I've caught is Epicurus formidolosus or Pyraloidea hilarius, and they're, for the most part, they're all in great shape. They're just falling into the malaise traps. I haven't really seen the rubbing off too much. I imagine it's something that's actually occurring more while they're alive and they're moving around and they're coming up.
Okay, thank you. There is a question.
Yes, please.
There is a question online by Bob Anderson.
Of course.
Have you found any new species in Florida?
Oh, yes, thank you, Bob. Yes, so while I was at, I completely blanked on this. While I was at the CMNC, I did come across a new state record for Florida for Cercopius scalii that is actually only known from North Carolina at the moment. So, but we have a record of it that was identified and confirmed by Bob for me during my visit to the CMNC. So yes, so that's a new state record for Florida.
Yes.
Do you have pictures of the damage also? Like Michael Sayers, the damage is absolutely characteristic.
No, actually, that's not something that I have, but I will add that to the final product. That's a great idea. Thank you.
So thank you very much. We have our last presenter before the coffee break is Andrew Monk. Faster Z, under the sea. Genetic drift drives sex chromosome evolution in salmon lice.
Okay. So yeah, this is going to be a little bit different, kind of on the edge of entomology and on the edge of systematics here. So because of that, I'm going to spend a good portion of this setting up why we care about sex chromosome evolution at all. If I can convince you that we should, why we care about sex chromosome evolution in sea lice in particular. And then finally, if you're still with me, what we actually learned about sex chromosome evolution from the sea lice. So starting just generally, why do we care about sex chromosome evolution? Well, hopefully just because we care about evolution in general. To be a little bit less cute about this and a little bit more detailed, generally, as we're trying to describe the diversity of life on the planet, things change either through random chance, there we go, drift, or selective processes like adaptation. So this applies to phenotypes like the color of a beetle, for instance, can change in frequency in the population over the generations, either through a drift event, like all of the green beetles accidentally getting stepped on, or some selective process like predation from a lizard, changing the color of beetles over the generations. And this is true of complex phenotypes, but it's also true of the sequences of DNA that give rise to those phenotypes. So as a molecular ecologist, my interest is really which of these forces, drift or selection, wins and under what conditions. And so there's a whole host of conditions that might favor one or the other. I won't exhaustively go through this list, but genetic drift tends to be stronger in things that have low population sizes. Genes that are only conditionally expressed rather than expressed under a number of tissues or conditions are less exposed to selection and are more driven by drift. On the other side, adaptation really benefits from high rates of recombination. Things like haploid expression and selection can also make selection more efficient. And the reason I have this whole long list up here to get to the punchline a bit is all of these things are features of sex chromosomes at the same time. So there's a number of things that point towards strong drift and a number of things that point towards adaptation. And it's not really clear which of these should matter more for the evolution of the chromosome. So just kind of orient everyone with the more familiar first, XX, XY systems like we have in mammals, some arthropods like beetles and flies. Here, biological females have 2 X chromosomes, The males have an X and a Y. To be honest, right now, we don't really care about the Y chromosome for the purposes of this talk. It's mainly degraded. There aren't many genes that are still functional on it. So we're going to focus on the X because the X still has a large number of genes that contribute to the phenotype of an organism. And if we just take a look at the X's here in this little tiniest possible population, one female and one male, we see, first of all, there's a smaller population size of X's than autosomes. So for every 4 gray autosomes here, we only have 3 yellow X chromosomes. And because of that, that's happening because there's only one X in the male. Any genes expressed on the X of a male are expressed in the haploid state. So right off the bat, we've got 2 opposing forces here. There should be more genetic drift because there's fewer X chromosomes than autosomes. No matter what the overall population size is of the species, this is true relative within the same genome. But on the other hand, this haploid expression leads to haploid selection, which is much more efficient. So depending on which of these 2 forces is more important here, the amount of adaptation, which I'm going to just shorthand as alpha here for the rest of the talk, is either going to be higher on the sex chromosomes or lower on the sex chromosomes. Funnily enough, theory predicts that either way, whichever of these is true, the rate of molecular change, the NDS, or scaled divergence between species, should be higher on the sex chromosomes than the autosomes. They should evolve faster. So in theory world, this is known as the faster X effect. And if that faster X is driven by adaptation, it's a more adaptive X as well. So from X world, this is a pretty commonly recovered result. We see a lot of faster X evolution. And this is a little bit less consistent. Sometimes adaptation wins, sometimes it doesn't. But to get a little bit closer to the heart of the talk, why should we care about this in salmon lice in particular? And the answer is simply put, because they're unstudied. And in fact, almost all of what we know, everything I was saying in that last slide, both the theory and the validation of that theory comes from these XX, XY systems. So it's not really clear how applicable all of our predictions, all of our observations are outside of XX, XY sex determination. And there are a lot of alternative sex determination systems, right? Especially across insects. There's a lot going on in this figure. We won't go through it all, but a couple of key highlights here. Red is XX, XY or XO. So like I said, a lot of Diptera, Coleoptera, Hemiptera, obviously us mammals, and some crustaceans. And there are a lot of alternatives, but the one that's kind of the easiest to extend our sex chromosome theory and validation to is ZZZW. So it's another chromosomal sex determination system. It's the one that all Lepidoptera use. It's the one that pretty much all birds use, some reptiles, and some crustaceans. And ZZZW is basically XXXY in reverse. So here males have 2 copies of the same chromosome, the Z, and then females have 1 Z and 1 W. Much like the Y in XY, the W chromosome doesn't recombine with the Z. There's usually very few genes maintained on it. It's pretty degraded. So we're going to ignore that going forward and really just focus on how the Z chromosome works. And we can pretty much port over that theory from the X chromosome to the Z. There's a lot of the same arguments, just sort of switched which sex we're talking about. So once again, the Z has a smaller population size than the autosomes because there's only 1 copy in females. And there's haploid expression of genes, again, expressed in females rather than males in this case. So just like the X, this could either lead to increased drift or more efficient selection. So either alpha decreasing relative to the autosomes or alpha increasing. Either way, theory predicts that this should increase the rate of evolutionary change relative to the autosomes. So we call this a faster Z effect. And of course, if adaptation is the culprit there, we'll call it a more adaptive Z as well. So we have a little bit of information about this, but it's not super generalizable. So this has mainly been studied in birds where genetic drift, it's a little hard to read that, but it's pretty consistent across the birds that genetic drift drives Z chromosome evolution. Butterflies and moths, on the other hand, we have more evidence for adaptation, stronger adaptation on the Z chromosome, thanks to expression of genes from females. So these two taxa don't agree with each other. And what's more, if we drill down into the Lepidoptera don't even agree with each other within established research. So there's sort of more consensus than not for an adaptive Z, but this faster Z process, which I kind of set up as non-controversial, we should expect it either way, is actually kind of hard to recover, and a lot of studies don't find it. If you really kind of stare at this too long, you'll note that there's something of a phylogenetic signal for this, where more closely related species tend to agree more in how their sex chromosomes are evolving. But if we zoom kind of the other direction, we're really only looking at 2 data points, because all birds share a single origin Z chromosome early on in that lineage. Lepidoptera and Trichoptera also share a single separate single origin Z chromosome. And so when we're trying to understand the general patterns of how and why different parts of the genome, especially the sex chromosomes, are evolving, all of our observations in birds and butterflies kind of amount to 2 independent data points that don't really agree with each other. So certainly we need to resolve this, especially what's happening in Lepidoptera. But another angle of attack we can do here is just to look across these independent evolutions of sex chromosomes. And so this is where we get to the sea louse. Here we go. I'm looking here at Lepidopterus salmonis, the salmon louse. It is another independently evolved ZZ-ZW system. It's a copepod, and it's importantly a pest of aquaculture. So you find this on fish farms, especially salmon farming is really kind of troubled by this. And for my purposes, that's convenient because it means there's a ton of data that have been generated to try and understand and mitigate this pest. I won't really bore you with bioinformatic methods here, but I did want to kind of briefly highlight first, this top row of boxes are all of our input data, and everything used to generate this study is already publicly available, which is kind of nice, and points to how we can step between applied and basic research and use data to answer different questions for different purposes. Just to sort of briefly orient you, what I'm about to show you, we will look at divergence data, DNDS, like I said, along with the linkage information, our genes on the Z chromosome or the autosomes, to ask, is the Z faster evolving than the autosomes? And then that alpha parameter for adaptation, is the Z more adaptively evolving than the autosomes? And then we can intersect this with the sex-specific data, our genes being expressed in females, males, or both, and use that to answer why do we see the patterns that we do for faster, more adaptive Z chromosomes. So not to belabor the point here then, just get to it. Is the Z chromosome faster evolving than the autosomes in the salmon louse? Yes, DNDS is higher for Z-linked genes than for autosomal genes. Pretty straightforward there. If we parse this a little bit, looking again at those sex bias genes, so things in blue are expressed mainly in males, gray is in both sexes here, and then red are the genes expressed by females. We don't really see any difference in how Z-linked female bias genes are evolving. So it doesn't really give us any evidence there for intensified selection from this angle. Although we do see that female bias genes on the autosomes are evolving quicker than other classes of genes, maybe potentially hinting at female bias drift. More on that in a couple of slides. Then on to the second question, is the Z chromosome more adaptively evolving? No, this is really crunched down for time, but the point estimates on here are practically the same between the Z chromosome and the autosomes. They're not statistically different. And if we kind of decompose this again into things expressed by males, females, or both, what you'll see is that it looks like everything's equal here. If anything, it looks like there's less adaptation of female biased genes on the Z. So that is more negative than the other alpha values here, although there's a lot of variance around that point estimate, so they're not statistically different, but certainly not more adaptation happening there. And just for completeness' sake, the only real difference we see is on the autosome, male biased genes are more adaptively evolving than other types of genes. It's kind of another story for another day. But it kind of leaves us with the question of why we don't see more adaptation through that female side in sea lice. And I think it really has to do with their life history. So these, like I said, are ectoparasites of fish. They attach to feed on them. And so this life history creates weird little mini populations on each individual fish. And depending on chance events, when the immature sea lice are looking for a host, you can get really skewed sex ratios on the fish. And so, you know, you need both males and females, hopefully in roughly equal ratios in order to get successful mating here. And if you have a case like this where you only have females on a fish, there's going to be no reproduction here. So there was a study a few years ago that looked at this more systematically and found that up to 30% of female sea lice actually fail to mate in a given year. And so what this does is this creates really strong drift on the female side. Kind of doesn't matter that you're having more intense opportunity for selection if you don't mate and pass those genes on. So wrapping this up for coffee hour then, we see drift, not adaptation, driving faster sex chromosomal evolution in sea lice, likely because of their parasitic lifestyle. If we zoom this out to the kind of greater context of Z and W chromosomes, we see that there's a more general pattern. There is a faster Z across different taxa more often than not, though adaptation seems to be less consistent. And really drilling into that life history ecology helps us understand the sea lice and maybe helps us understand why the butterflies are inconsistent with each other. So if you're interested in any of this, the preprint is up now. You can read more about it. Thanks to a bunch of people, especially my co-author, Rob there, Gustav Polly here at the museum for helping me parse through some sea louse systematics. I'll take questions if we have time. If not, just find me during the break.
Yes, we have time for questions.
Yeah.
How do the sea lice like travel from fish to fish? Is it like mating or like they close proximity or?
Yeah, that's a great question. As far as I understand it, it's dispersal at the juvenile stage, and then once they're on the fish, they stick pretty close.
They're found under the scales.
So they're sitting on the scales. They're blood feeders.
So there's nothing similar with Hemiptera. You have them as haplodiploidally different.
Yeah, well, so that's a kind of interesting philosophical question. In a sense, haplodiploid Hymenoptera, it's almost like the entire genome is a sex chromosome because it's in 2 copies in females and 1 copy in males. So it's like a full genome X chromosome. There's a lot of sort of similarity there. Yeah.
Okay, thank you. So now we have our coffee break and everyone is ready. So we have our presenter here, someone lead. His presentation is 2 exotic Hymenoptera Spieri and Sinara Watsoni, identified recently from Korea.
Hello, everyone. I'm Sung Han Lee from Korea. I'm especially working at Seoul National University. I'm also taxonomist, but topics of today is not taxonomy, but about the exotic species which has been discovered recently in our country. We have been experiencing, as like North America and US, we are also experiencing many exotic species, really problems in agriculture and forest. But these two, and one more about Asian longhorn beetle is just case discovered recently. So only 3 cases I will explain for you. Okay, let's get started. And then the 2 recent discoveries, the Asian, it's like a same of the same genera of the Asian longhorn beetles belong to the Anoplophila, but this species Anoplophila horsefield comes from Southeastern Asia. It's not native to Korea, it's invasive. And the other one is the giant pine aphids. Unfortunately, it's coming from maybe, I don't know which part of the North America, but coming from probably from Florida, or I don't know. Anyway, it arrived recently to Korea. That's the 2 cases. And let's talk about a little bit about the location of Korea, because many of you are not familiar with the location of Korea. And these, is it cursor working? Oh, yes, yes. Our country is located in the Far Eastern Asia between China here to the west and eastern Japanese islands, right? And then very much temperate climate we have, very good every 4 seasons we have. And then unfortunately, our territory is divided into the north and the south. And my place is here, Seoul, capital. And then there, the mainland is temperature, but only the things the southern islands belong to the larger subtropical climate, very similar to Florida in Gainesville. Not the southern part of Florida, but Gainesville, maybe the similar climate we have. And then 2 findings. The first one discovered here in Jeju Island, in the subtropical climate. That's why I'm presenting here. Probably in the future, don't get this species to here. Maybe we'll make a problem in Korea too. And then the Edessinae species found on the capital region, in central region, recently. And then this one, the Anoplophila hospitals are distributed naturally in as this region. It's like this region is the original distribution here. And then the host plant. The host plant is like Camellia recorded, and the Celtis trees, many of the Celtis trees recorded in the southern Southeastern Asian country, and neem tree and evergreen oak trees, but it's not in mainland, but in only Jeju Island. And one of the species of elm trees are recorded as host plant. And then this is quite big longhorn beetles, very colorful. So before arriving to Korea, already the Korean collectors, beetle collectors, have known this species because they want to have these specimens. And then the size is quite big, 4 centimeters long. So this is arrived. We found this. We didn't find, but the pictures suddenly appeared on the internet, and then the citizen reporters showed up to his photos on the internet. That was the first observation in 2019 here. Between, there is airport, international busy airport, because that island is tourist island. And then also there's a big seaport here. And in the between there is the first place we observed there. And after then, experiencing pandemic, the second report from this place, but that was the only picture. But here we have about 40 specimens collected and observed. And then also the emergence hole also observed. about 50. And then just 1 year after here, sorry. 1 year after 2023, the last summer, it's like an explosion of this species, just 1 year after, and then more than 1,000 specimens reported and collected and surveyed by our team, also the collectors. And then before we going to the place, already the collectors coming arrived and reported a lot. So that is the case. And then you think about the price is about $20 for one specimen. So this is the case of Danoprophala host building. And then after collecting the specimens, you tried to the preference of the oviposition. Even though there is a record of the different host plants, the result was simple. Only the Celtis trees is the host plant for the oviposition. And they do prefer not small trees, but the large trees they like. So that is a result. And also, this is the symptoms of damage, like this. And then they prefer the lower part of the trees, big trees. I don't know that Celtis grown in Florida, probably yes, because that is very much subtropical ornamental trees. And then the damages is like this. It will not kill that, that trees in 1 year, maybe 3-4 years accumulation of the larvas, they will kill at the end like this. The trees will die at the end. And then the adult feeding, natural feeding is looking like this. That is nowadays happening in Jeju Island. And then about the Cinara watsoni, as distributed originally from North America, I told you. And then 1993, it discovered in China first. And after 30 years, we have the species in our central region of South Korea. So they are basically oligopaus species, different Pilus species are recorded as host plant, and then quite a big IPs, and then again it was discovered on the ornamentally planted pine trees. It's on the rest area of the highway, main highway of the Korean Peninsula. And then it was together with that another North American origin, Cinara atlantica and watsoni together with the species. So Atlantica becomes problems in the natural forest. It feeds on the native species of pine trees. So Pyrus densiflora becomes a problem of that one. And we are worried about this species also in the future will make that kinds of problems. And then we confirmed that the species determination by the barcoding completed the same identical of the US population. And then those two cases, the invasive species recently discovered, but I want to talk a little bit about the ALB, Asian longhorn beetle in Korea. The Korea, that is the original native territory of the Anopropora gravipenis, but recently we discovered invasive population. It's not invasive species, invasive population we discovered. The original native population normally living on the northeastern part in deep forest. They feed on the native acer trees, but suddenly in 2014, two populations discovered in the metropolitan city of Seoul and near to Incheon, the harbor, the second largest harbor, and also in Busan, the biggest the seaport in the peninsula. Two populations are making rather high populations, not like the low population of native population. So that's why we have tried and made experiments compare the native population and to those invasive population. and characterize the northeastern native population. They are from the Acer trees, and they're very low density, not like in North America. And the invasive population due to the western seafood, they're not on the Acer trees. They're from the horse chestnut, the Asculus turbinata, and also the invasive introduced maple tree Acer saccharum. It's not a native species. And then the second invasive, possibly invasive population, they are not again from the Acer trees, they are from the Salix trees here. And then we compared that haplotypes and figured out that the native population is genetically diverse, which means that the native population and the Western harbor population is identical haplotype. And then this one again, the invasive populations, and then compared this part, that samples together with the Chinese, many of the Chinese population and some of the North American population and the European population invasive, and then figure out that the northeastern, the northwestern invasive population probably from the northeastern China, and then the other one comes from the west, southwestern population, southwestern China, independently invaded. So that is the case of the 3 invasive species and population in Korea. And I hope that you are not getting this species Anoploca hosted. Bring it to Florida will make a problem. That is the end of my presentation. These are the students who made that long-haul beetle investigation. This guy's working on the APS. So they are help a lot for this presentation and investigation. Thanks for your attention.
Any questions? Yes.
Yep.
I heard you mention that many of these were near seaports.
Yep.
Do you have thoughts about woodpacking material or other ways that perhaps that these were introduced?
Yeah, that is like a pallet, the packing, the importing goods, they're really the problems. That we guess that not only the species, but also, that some of the nematode vectors, monocoms, transported through that woodpacking, shipping materials. Yeah, that's true. Yes.
Is there any regulations on the control of the specimens from professional collectors?
If it is a case, that's like the same in the United States. If it is a case of the CITES, the regulated species, there is a legislation. But in this case, you mean that in this case, normal, not visit one, there's no regulation. Of course, in this case also, as an invasive quarantine species, the government control. Government control after the introduction. But normally, general species, we do not have control.
This sort of tie into what you were saying too. So once the specimen's found, you guys how do you guys control or try to contain the specimen from spreading? Do you guys use a pesticide?
Yeah, we did use that. I didn't treat the chemicals, but the local government, they treat once a time after just last summer, because of the explosion of these species, they did treat chemicals, but I don't know what kinds. But they didn't inject the chemicals into twigs, but treat sprayed.
Okay. I think the issues happening in Japan right now too. What I saw recently, in Tokyo, they're having a lot of the longhorn beetles eating the cherry blossom trees. They're trying to control. I think they're cutting the trees down.
Yeah.
They're destroying all the trees.
Yeah. That is the case of because of the fire of the nematode. Of course, in Korea, we do same things in the forest because Fine-wood nematode really problems. And also you know that Japan, they gave up the fine-wood nematode. They don't care. Just let it go everywhere. So, but in Korea, our country, the pine trees is very important, and culturally and also economically. So we cannot give up controlling the pine-wood nematode. That's why we are cutting. We are making the buffer zone, and if there is the pine-wood nematode there, we make a 4 kilometers barriers, buffer zones, all cutting the trees and then making like package and fumigant treatment. This is really, really bad and economically money consuming control. Yes.
Thank you.
Thank you very much. Our next speaker is Sebastian Palmieri.
I'm sorry, can I make a quick announcement before we do the next speaker? This announcement is only for people here at the McGuire Center, of course, not virtually, but at 5 o'clock, a reminder that the public exhibit areas will be closed. So since we'll be staying past 5 o'clock, when you leave McGuire at the end of the day, please make sure you leave through the side entrance in the stairwell. Do not try to go through the public exhibit. Best case scenario, the door will be locked. Worst case scenario, it's unlocked and you accidentally trigger an alarm. Again, please make sure you use the side entrance that heads from the stairwell that heads straight outdoors. And if you get lost or have any questions, me, Jim Hayden, plenty of other people here familiar with the building can help you find your way.
Thank you, David. Okay, so we have Sebastian. The tales from the vault, the Papilionidea found in Philip and Patricia Frost Museum of Science collection from the Little River neighborhood of Miami, Florida, 1920-1950.
All right, hello everybody. My name is Sebastian Palmieri. I am an undergraduate student at Broward College in Davie, Florida. Hoping to move on to my master's in entomology. Today I'm going to be presenting the Little River Leps project. I have to acknowledge my advisor, Dr. David Serrano, Roberto Fernandez, who did a lot of the history work on this manuscript, and Alexander Bliss, who did early organization of the specimens. So at the beginning, we're going to have a little bit of a history about this collection. It's a historical manuscript on a collection, and then we'll get more into specifics with the specimens. So our collector, we have Joseph Manson Valentine. He was born in 1902 and ended up receiving his BA and doctorate in zoology from Yale. After graduating, he partook on many expeditions. He went all around the country as well as internationally. His most notable publications were describing a few new genera and species of cave-dwelling carabidae. We'll see a picture of one later. And then in 1957, he ended up becoming the honorary curator at the Miami Museum of Science. He held this position for nearly 40 years until his departure over philosophical differences. So Valentine's extended career, Valentine was also an avid marine scientist and did a lot of scuba diving. In 1969, he took a trip with a few other men to the Bahamas and ended up finding the Bimini Road. And he reported these findings through the Miami Museum of Science. He never mentioned Atlantis in his findings, but due to the nature of his discovery, many people looked at him as the newest expert on the subject and finding the newest and greatest thing in Atlantis discovery. Many of the letters that were sent to him from these other scientists, he stored in the museum and in their vault. This was actually just beach rock that cracked very quickly due to drying. So about the location where this collection is from, we have the Little River neighborhood in Miami. You can see the exact border of it. It's about 2 kilometers west of the Biscayne Bay, and the name is derived from the river that runs from the east to the west, from the Everglades to the Biscayne Bay. And this is likely to be the historic easternmost point of the Florida Everglades. Up here you can see a picture of some people selling some native plants. They have some coontie and others. And in the back it says Little River on the sign up there. And then this is a picture of the Little River pre-dredging when they dredged it in 1935. And this is a little glimpse of the Sawgrass Prairie ecosystem. So a little bit more about the climate. Many people, we are all from, not all of us, sorry. Many people from Florida will know that we deal with a subtropical climate, characteristic humid and rainy season from June to September. And the area is also susceptible to tropical cyclones and hurricanes. And then just a little more info about the monthly mean temperature for the period of the study. In Miami, it was 75.1 degrees Fahrenheit, and then the mean rainfall year was 61.19 inches. This is actually a little advertisement of them advertising how beautiful the river was. So a little bit more about the vegetation in the area. In the historical manuscript, we have many accounts of early explorers and pioneers that express their sheer amazement of the denseness and the height of the mangrove forest that they encountered when first exploring the area. And then, as mentioned earlier, the ecosystem there was a combination of the sawgrass prairies as well as pine rocklands or flatlands. And so here's a picture of a one-lane road, maybe two. That's about all. And the other last weekend, we ended up going to this exact site to see how it has changed over the years. And this is what it has become. It's now a 5-lane road with sidewalks on the side. And taking a step back, you can see it is nothing but lined with businesses. It's just a little compare and contrast from the past. So where did this collection come from? So as mentioned before, Valentine was part of the Miami Museum of Science. He had a vault containing many different specimens of many different animals, insects, documents, many things. Some cool notable things were Carolina parakeets and ivory-billed woodpeckers, some cool extinct species that they had in the vault. And then in 2016, the museum ended up moving to the new modern building that you can see here. So this building was a wonderful architectural and technological feat, but the one downfall, they did not want to store any specimens there. So through connections, the Broward College insect collection ended up intaking all of the Frost Museum arthropod collection. This is just a display of some of the drawers. This is my collection, but we also have many more drawers of international, caught, and other groups of insects too, not just Lepidoptera. And then also with it being a very historical collection, 90, almost 100 years old, the specimens, a lot of them had not the best curation, data, all those types of things. So we had to really take a dive into it and see if there was any notable collections that we could do a study on. So the first collection that was noted from the Frost Museum was a series of cigar boxes that contained a bunch of carabid beetles that Valentine ended up collecting in Brazil. Due to the age as well as location of these specimens, they are very scientifically valuable. And then also I wanted to mention that Brazil, they had a fire in the National Museum of Brazil in 2018 and lost much of their entomology collection. The specimens are even more valuable at this point after that. And then we ended up sending these specimens to Terry Irwin at the Smithsonian to be further curated and studied. Sadly, we do not have any pictures of the carabids that Valentine had, but this is one of the species that he described, Darlingtonia kentuckensis. It is a cave-dwelling carabid found in the caves of Kentucky. All right, so then about my collection, we have the Little River Leps. This was the second notable collection out of the entire Frost Museum collection, and we have about 70 specimens in here. As you can see, a lot of these are in baggies, missing various wings and such, so it was definitely a project ahead of us. So at this portion, I'm going to go specimen by specimen. Some of these we have a single of each of them, such as these sphinges to start off with, but later on we'll have a bunch more numbers for some of the other species we have. And we have the color palettes, a reference, because these are very dated and old, faded specimens. So we have Xylophanes tersa to begin with, the Tersa sphinx moth. We have a Hyles lineata, the white-lined sphinx moth. And Enyo lugubris, the mournful sphinx moth. The polka dot wasp moth or oleander moth, we had a couple of these. The large orange sulfur, Phoebus agarith, we had nearly a dozen of these. I don't know if they smell like Doritos. Need to get to that soon. A couple dainty sulfurs, Nathalis aioli. We have a little more uncommon one, the sleepy orange, Eurema nasipi. We had 4 of these. And this guy is sadly missing his head. Another one sadly missing their head. We have the great southern white. For the Papilionidae, we had 2 or 3 specimens for each of the species that we're going to go through. Baptis polydamas, the polydamas swallowtail. We had the black swallowtail, and you can see just the body left on some of them with a bag containing the forewings and another bag containing the hindwings. The Papilio cresfontes, the biggest specimen in this, we had a couple of these as well. We have the red admiral, Vanessa Atalanta. I collected my first the other week, so it was very exciting. Danaeus glypis, the queen. You can see how their deep orange is quite faded. We had quite a few, 3 or so of these. The single variegated fritillary in the collection. The Junonia coena of Buckeye. We had 2 of these. This one was very wonderful condition, the other not so much. The single zebra longwing, Heliconius carathonia. We had about 3 of these, Physioides phaon, the phaon crescents. I like pointing out this monarch because you can very clearly tell how faded the specimen can get after 90 years of storage. And then this is a nice contrast of how bright and vibrant a specimen can stay. At the end, we got a little help section. Got a bunch of experts in the room and a bunch of things that were unable to be identified. Clearly a Hesperiid, but down to species level, some of these can be very difficult. So, and then for the final section of this presentation, I'm just going to do a little bit of analysis with iNaturalist. Many of you may know iNaturalist is a wonderful free source of citizen science where you can discuss your identification of species. So we use this as a source to compare the past locality data of our specimens to the current locality data that is found on the iNaturalist distribution. So this is one example of something you would expect to see. Valentine caught the zebra longwing in Miami about 90 years ago, and it is still distributed throughout the Miami area. And this is also an observation you could expect to see of the butterfly on its plant feeding. So for a couple other species, we were able to, while looking through these species, I was able to notice that I haven't seen these and they don't occur too often down in South Florida. So we looked further into them. And this was the first one we looked at. You can see it does not occur south of West Palm Beach anymore. We had 3 of these actually in wonderful condition of the Eurytides marcellus. This one with a little scarcer distribution. We have Olypia, the forester moss. We had 2 of these specimens. It was a toss-up between Olypia whitfeldii and Olypia octomaculata. I went with the Olypia whitfeldii because it was more common in Florida compared to the octomaculata. And for our final, most important specimen out of this collection, we have a eastern pine elfin, the Calliphorus nifus. As you can see, the distribution is completely scarce, almost non-existent in the state of Florida. The observations that do occur up in the north end of Florida, they were found on sand pine. And sand pine is no longer found south of the Fort Lauderdale area when looking at the iNaturalist distribution. And then last week, while we were doing our trip to look at the bridge to compare and contrast, we also wanted to see if we could find any pine trees in the Little River neighborhood because this specimen was so unique and just wanted to take a look for pine trees. We weren't able to find any sand pines, of course, like I said, but we were able to find a single South Florida slash pine while we did our search through the neighborhood. Also, if you look on iNaturalist, there is no other pines in the Little River neighborhood either. And this is the distribution of this pine. Although it is not a listed host plant of the Calophyllaceae fauna, it is just interesting to note that we still have a decent amount of pine, but it is not the listed host plant for this species that we're looking at. And this house is for sale, so hopefully the last pine in Little River does not go with it. And some cones we collected from it. And with that, I would like to thank my advisor, Dr. Serrano, and my photographer, Joey Sigmund, and all of you for being my audience.
Great job. Any question?
Sorry.
You're welcome.
Yes.
Since most of those specimens did not have date on them, are you sure that they were actually collected?
I'm sorry, I might have misspoken. The ones that we have data. They have dates, they have location of 1934 through 35 is most of the collections. The ones that didn't lack data didn't make it into the big collection that we assessed, the crambids or the leps. Those were separate and just stored as part of the Frost Museum collection, but studied in these total collections.
Thank you, Sebastian. Very good. Okay, we have our next speaker, Tracy Leisenthal. Genomic insights of recent invasion of Miridae in Florida.
Oops, I didn't do that. Hi, I'm Tracy Leisenthal. I'm a master's student at the University of Florida in Dr. Andrew Munn's lab. And today I'm here to talk about genomic insights of a recent invasion of Levick mealybug in Florida. So first and foremost, what is Levick mealybug? This thing you're seeing here is not quite the mealybug, but rather it's ovisac, a wax that it creates, producing some honeydew. The mealybug itself looks like this. So this is an example of two adults, the female as well as the winged male. So these are otherwise known as Nymphicoccus viridis, the scientific name. These are generalist hemipteran pests within the family Pseudococcidae. These are primarily sap feeders, and they have been reported to feed on a wide range of plants, 140 genera at least. You can also see in that image there within the leaf bud, there are tons of little juveniles. They're quite small. And as I've mentioned, the males and females are very sexually dimorphic. They typically live between 3 to 4 weeks, with the males typically living less time. As adults, they don't actually feed, so they have a shorter lifespan. And so, as you can see, they kind of congregate all together. The females also, when they make those ovisacs, have a tendency to aggregate together in their clusters. And when they create those ovisac clusters, they can produce between 400 to 1,000 eggs within that cluster themselves and within their lifetime. So moving on to those clusters, here's a pretty crazy invasion of one on a single citrus plant. And this here is the same tree just 3 months later. And as you can see how much damage that just these little tiny bugs can do to a single plant. I also have some examples here of what they may cause to fruits. So this is an example of a citrus fruit, and as they feed, they produce this saliva that causes almost gall-like outgrowths onto your fruits. So this is pretty important in terms of agriculture, because not only are you possibly losing young plants, but you're also having deformities in your fruit. And so you can't quite market that as fresh fruit and sell it. So now, to look into it in terms of invasion, I just wanted to also show here those clusters that I mentioned. This is what it looks like inside of it. All of those are little tiny crawlers. So just seeing one cluster there, it might look like maybe just one bug, but realistically, there are hundreds within that cluster. So originally, these are native to Asia, but they've been documented in at least 60 different countries. Not only that, but they've been reported to be a pest on a variety of economically important plants. This includes citrus, coffee, soybean, roses, just to name a few, but a wide variety. They have been reported to cause yield loss in Africa citrus. There's as high as 50% loss within some of their groves. And so this could be indicative of some of the danger that it poses to places like Florida, which have a wide range of citrus crops here. So moving into Florida. So originally it was documented as an invasion on dodder plants in 2009, but it didn't really see too much of a expansion across Florida until 2019, where it was recorded on commercial citrus. Since then, it's been documented in at least 23 different counties. And so I kind of have here in the orange, these are the different ones that I have record of from a variety of sources of where we've seen them so far. And yet, despite this, this is still pretty early in the invasion. You know, it only just got spreading in 2019. And this really poses a unique situation to be able to understand invasion in a different sense. And one of the ways we can do this is through genomics. So what's really cool is this genomic technology has continued to advance and move past just using model insects. We can expand these systems to non-models, such as these invasive pests. So here's my artist rendition of the mealybug we're working with. What's really cool is that we can now use, we can get a reference genome for this insect, which is not something we've been able to do in the past, and typically not something we've seen so much in scale insects. So it's really cool as we used a combination of PacBio hi-fi long reads in combination with Illumina high seed to be able to scaffold these reads together to get us to a genome level assembly, or sorry, chromosome level assembly. And this is what it's looking like. So this chart here, each of those 5 boxes, that represents the 5 chromosomes that we see within this mealybug. And we're at a genome size of 289 megabases with a sequence count of 552 and 53 million base pairs per N50. So to make sure that we're kind of getting the most complete genome and that we're really getting this assembled correctly, we use what's called BUSCOS. And so BUSCOS is looking for genome completeness. And within this, we're using specifically a Hemipteran BUSCOS. And so this is looking for the genes that we expect to see within Hemipterans. And so ours is at 89.9%. So we can be pretty confident that we're pretty much assembling what we would expect within this, and that we have a pretty good assembly when we compare it to other scale insects. So specifically, we also have Planococcus citri here, which is citrus mealybug. That one's at 94%. And Iceria persiani, which is the Cottony cushion scale, at 91%. So by comparison to those, we can have a pretty good feeling that we're assembling pretty good on this one. So what can we do with this? What's really cool is we can track population genetics and look at the invasions in the different counties. So I have here, you can see all those little black dots. These are the different counties I'm showing today. A lot of this is very preliminary data, like literally just got some of this last month. So it's very early stages. And so I just have a small representative sample so far. So in total, I had 20 samples. Within this, I had 10 different cities encompassing 9 different counties in Florida. So using these population samples, we used Illumina short read sequences to align to our reference genome. And in doing this, we first checked the alignment rate to make sure that we're actually sequencing what is also Nymphococcus viridis. And with good alignment rates, we can then move on to looking at just how genetically similar or different these populations are. So we moved on to creating a principal component analysis, or a PCA graph. And so this is using single nucleotide polymorphisms to check for genetic variation within these populations to see how genetically similar or different they may be. So here's just a more simple list of the samples. I have condensed it down to showing 10 samples since we have repeats for each of the cities, just for more clarity when showing the results. So you can see there's 9 different counties within 10 different cities. And I've also included a column that says agricultural practice. You know, where did these samples originate from? So I have 4 categories in total. There's some residential plots that they're from, some groves, campus gardens, as well as nurseries. So what do we get from this? So this is what the PCA graph is looking like. And I know this just looks like numbers on a graph. What it really looks like is we have 3 different subpopulations that are kind of clustering together, showing that they might be more genetically similar to one another. So now it's, what can we make sense of this? You know, we're seeing these groupings, but what could be contributing to these genetic similarities or differences? So first, let's look at it geographically. And what's really interesting is we're actually not seeing a lot of similarities when you look at your geography. You know, we have a sample, which is Pinellas County, which is this nice yellow one right up here, and Hillsborough County, which is this blue one here at 41. Those are right next to each other geographically, and yet they're pretty different in terms of our genetic variation. But then similarly, you have a population like Brevard, which is on the east in purple, and then a sample from Monroe County, specifically like Key West area. And these 2 are very, very similar to one another, which is really interesting when they're so far apart. So now we're looking at this and going, okay, so we don't really see clear connections by geography. There's like not a clear isolation by distance in these populations. What other features can we look at? So next I decided to look at the host plants that these samples were from. You know, maybe something with the host plants could contribute to how similar they are. So in total, I had 4 different host plants. Most of them came from citrus species. Others, I have Mariah, Procarpia, and Artocarpus. Please excuse my pronunciation on those. But as you can see, there's not really a clear trend on that either. You know, you have your citruses that are further apart, but you also have citrus that's in the middle here, as well as your ornamental plants. There's not really true clear connections just looking at your different plant species. So we're like, okay, well, what other thing can we look at? So the next thing we decide to look for is those different agricultural practices. Where did we take these from? And now we're starting to see a bit more of a pattern. Our campus gardens kind of secluded by itself, where for the most part, minus this one sample, the groves are kind of in their own little population. And then the nurseries and the residentials are more together. So again, this is very early data with just these cool, like small set of points, but it's kind of cool to start seeing a bit of these trends. So what does this mean exactly? So like I said, we're not really seeing anything isolated truly by distance, and there could be differences in the genetic variation as a result of the agricultural practices. And this might also suggest the means of human interactions and dispersal when it comes to contributing to this invasion. Maybe things are being moved from a nursery to a residential plot. It's suggesting there might be more human means, since, like I showed the pictures, our little females, wingless, they don't really move much. And our males, they only live a few days, so they're not going to be flying very far. So there might be a bit more of a human mediated means for this. So next steps, of course, adding more counties. As you can see, there's some blue dots on here. Those are just within the past 2 weeks, a couple more samples that I'm adding. There's a couple new counties, but you might also see some in counties that I've already recorded. And so another thing to look at is, if these agricultural practices are truly contributing to these different sources of variation, it's good to get more representatives within a county. So if I have some that are already groves, maybe look at ones that are in residential plots or vice versa. So that's another goal to add to this. And another goal is also getting more samples from prior to 2019 invasion, when they were initially recorded in 2009, past that. How different or similar could those populations be to what we're currently seeing? Is this a secondary invasion, or were they secretly invading throughout before we even noticed them? From here, another next step, as we fill out this counties and get more samples, we can look at K values to truly look at those different populations and kind of see how many populations we may truly have. Because it may show 3 clusterings, but there could be more individual populations within that. And then similarly, once we fill out more samples, we can use a fixation index or an FST. And this allows us to look at the differences in allelic frequencies within our populations. It's a bit early to do that now, as one of our population clusters only has 1 sample. So we're hoping to fill that out more so then we can truly look at the different frequencies. And then just even beyond the scope of this project, something that's really cool is like having a reference genome opens a whole new door of opportunities for this subject area. So one thing that's really cool is mealybugs have endosymbiotic bacteria that contribute to their success as a generalist pest and being able to go on a wide range of host plants. So what's really cool is using this genome, we can actually look further and try to analyze what bacteria they contain and analyze it further and compare it to other mealybug species. Similarly, we can also look at difference in the gene expression between your males and your females. Without derailing things too much, they actually are part of a really unique clade that has, for their sex determination system, they undergo a process called parental genome elimination. So they don't actually have sex chromosomes, and the males are functionally haploid despite being diploid. And so this is really cool because it gives us another opportunity to use this reference genome as a means to expand other areas. And then furthermore, it also can just even add into understanding the phylogenetics of scale insects beyond the morphological characteristics. Cool. And so my work science. And with that, thank you to the Among Lab, old and new and current, because there's a lot of troubleshooting that goes into creating a reference genome, especially when you're working with something so small. Similarly, thank you to the Deep and Brock Lab at UF IFAS Citrus Research Education Center for providing us lab-reared mealybugs, so that way we can use them for our reference genome. And then finally, thank you for FDAX, for the Department of Plant Industry, for providing us our different county samples within this research. And with that, thank you all for listening, and I'm open to any questions.
Questions? I have a question.
Yes.
How many samples will you need to have like a more?
Ideally, I would like to try to get like multiple per county, just so I can try to get those different variations in the agricultural practice. Like currently there's like 9 and there's been documented 23 counties. So branching out beyond that, hopefully to be able to encompass all the counties.
Thank you, Tracy.
Thank you.
Okay, our next speaker is Bert Coleman. New work done beetle colonization of a new Miocene insular territory. And we have the presentation on Zoom.
Hello, good afternoon. Thank you very much for inviting me to this presentation. I would like to present the team who is working on this project. We are 5 people, and these are the different persons working on the project. So, regarding the origin of Costa Rica, well, at the beginning, there was nothing. And then Costa Rica slowly emerged from the seas, first as volcanic cones, then as the volcanoes, then as volcanic islands, and finally it started to develop an archipelago system. Using previous work by Lucia Valerio, who is a specialist in fossils, and Guillermo Alvarado, who is a geologist, we have been able to reconstruct in a better way the situation of Lower Central America from the Miocene to the Pliocene. And one of the interesting things we have been able to determine is when Costa Rica actually connected to Nicaragua, which was around 5.8 million years ago. This is based on the fossil remains we have been finding here in San Gerardo, Limoncito, which are all mastodons, camels, horses, as well northern fauna, more than anything else. So this means that the northern fauna had a little bit of an advantage at colonizing that archipelago in relation to South America. 2.8 million years ago, the Panama Portal closes, and then Costa Rica becomes completely embedded into this continental matrix. Well, in order to study this Miocene process, we have used 2 different dung beetle genera. One is Canthidium, which is of South American non-Gondwanan origin, and the other one is Ornithophagus, which comes from North America. Actually, it invaded via Beringia from East Asia. Why are we comparing all these 2 genera? Well, it all began with a study by Edgar Baron von Harold, who was the first to notice that Canthidium is actually the tropical equivalent of Ornithophagus. So we wanted to see how these 2 very similar genera would behave while invading this new Miocenic archipelago. And we actually studied 3 different areas, 3 different geographic regions in Costa Rica. One is the Caribbean, another one is South Pacific, and the third one is the North Pacific. And as you can see here in black, we have all the endemic species. And as you can see, endemic species in all the areas and at different altitudes are incredibly numerous. We have an enormous amount of endemic species here, and this is usually typical of what is called an island syndrome. We can see here the percentages of endemics for Canthidium and for Ornithophagus for Costa Rica, and the nearest comparison is with Oaxaca in continental Mexico. You can see here the percentages for Oaxaca. So we could say that Canthidium and Ornithophagus are behaving in a very island-like style. There are other island syndromes that are present in Costa Rica. One, of course, is brachyptery, which is the reduction of the wings so the beetles cannot fly. And we can see here a map. And what this map tells us is that this area is the area with the highest amount of brachypterous, dung beetle species for the whole American continent. And at the same time, is the area with the highest amount of different brachypterous genera as well. So there's nothing similar in any part of the continent. At the same time, all these species and other endemic species are very micro-endemic, which is again another typical island syndrome. Well, finally, we have another island syndrome, which is Foster's rule, which says that in islands, animals will either become very small or very large. Very small because there is a lack of resource, or very large because there is a suppression or a reduction in predation. The first persons that noticed this for Central America were Howard and Young in 1981, and they did a statistical analysis comparing the endemic Panamanian dung beetle fauna versus the non-endemic, and they found actually quite a nice value for P. So we now concentrated just on Canthidium, and we can see that Canthidium has become, or the endemic species have become very, very small, whereas Tophagus species have taken the other route, which is becoming very large. We can see here several species of Canthidium. These are here the endemic ones. You can see they are really and clearly very, very small in relation to the non-endemic species. Well, and now we have something really new. In 1855, a Prussian doctor called Karl Hoffmann was living in Costa Rica, and he made an ascent of the Irazu volcano in central Costa Rica. He went up and visited the upland forest, which is this one here, and he gave very precise descriptions of the vegetation, like this one here, Heteroptera. And this tells us that he was somewhere around 2,600 to 2,800 meters when he wrote this. He said that he could find very few beetles, just a few carabids and a few lampreyts, and that the cowboy who was guiding the expedition and had been living there for all his life had never in his whole life seen a snake or a lizard or a frog or a toad. So this is quite an interesting observation. It's the first time that such a thing had been recorded. So if we check again the dung beetles, but now the totality of the dung beetles, 199 species, for the different biogeographic regions, we can see that after a certain altitude, biodiversity just and simply drops terribly. You can see how it drops. And this is not the only phenomenon we have seen. For example, fishes, local fishes will just go up to 1,500 meters and then they just and simply disappear. Or we can also compare these sunflowers. This is the species Tithonia diversifolia, which exists in Costa Rica. And if you check what sort of insects visit this flower, basically almost nothing. A few syrphid flies will arrive here. And of course, the main visitor is the European honeybee. Now, if you go to Mexico and check the equivalent Tithonia tubiformis in central Mexico, you will actually find an enormous amount of fauna of all types and sorts living here. Like, for example, this beetle here, Euphoria basalis, which is the famous jicotillo of the Buena Blanca children's song in Mexico. Now, if we check, for example, for the 3 biogeographic regions, how many species do we have at different elevations? And this is based on 32 years collecting 1,017 localities and more than 150,000 specimens. So we are quite sure that these amounts are quite exact, quite correct. Now we can compare these numbers with a marvelous piece of work that José Lobo from Spain has made. He developed 2 equations determining the amount of dung beetle species for North America and for South America. So using these equations, we can see that at the beginning, at the lower altitudes, the amount of species in Costa Rica follow more or less South American behavior. It's incredibly rich, even richer than what you would be able to find in South America. At middle altitudes, things start to change, and it starts becoming more North American in behavior. And when you go high up, there is definitely an upland faunal deficiency. So we are thinking and proposing that most probably this could be a New Island syndrome. And this can be explained by the fact that, as we saw before, most of the species are tropical lowland species, and they just and simply haven't had enough time to develop, let's say, an upland fauna adapted to cold situations, to cold regions. And with this, I would like to finish presenting a thought by Alexander von Humboldt. We are all entomologists and we all work on small things, so he has a very nice thought about working and the importance of small things. And as you can see in this biometric photograph, Humboldt was not very tall physically. And with this, I finish my presentation.
We have plenty of time for questions. Yes, we have.
May there have been recent volcanic activity on this mountain that has contributed to the lack of fauna that you see higher up?
I'm sorry, the connection is not very good, so I couldn't hear your questions.
Try to repeat the question.
Has there been any recent volcanic activity on this particular mountain? And could that explain why we see kind of a drop at higher altitudes in the number of fauna?
Okay, in the northern part, there is still volcanic activity, so that could probably explain some drops. But the majority of the upland area is the Talamanca Cordillera. It's a non-volcanic cordillera, and there hasn't been any volcanic activity in the past 15 million years. So it's very clear that that area is very poor in the upper part.
Okay, thank you.
Okay, thank you very much.
Thank you.
Okay, we have our last speaker, Erin.
Please stop sharing your screen.
Erin Powell. A trash talk, defensive behaviors of debris-carrying lacewick larvae and their mealybug prey.
All right. Good afternoon, everybody. Thanks for sticking with us. So my name is Erin Powell. I am the curator of Coccoidea and Alrodidae at FDAX DPI here in Gainesville. So I work on scale insects, mealybugs, and whiteflies. And every day I get bags of leaves, basically, bags of plant material for identification. And each year I make about 1,000 regulatory identifications of these groups. And I love getting these bags of plant material because it lets me do things like rear out adult males of scale insects, which are poorly known. The estimate is maybe about 5%, at least of mealybug species, where the adult male is actually described. So I've got this nice collection of males going. I'm also interested in rearing out parasitoid wasps out of the material that comes in and giving that to people that's interested in it. So I had this fortuitous find back in December 2022, where a sample came in, a bag of sugar apple leaves, and this little guy was on there, this lacewing larva with a heavy infestation of coconut mealybug. So today I'm just going to talk about this fun little kind of natural history observation, the videos that I got of this little guy behaving, and just talk a little bit about how I'm lucky to have a job where on the side of doing taxonomy projects and identifications, I can write up silly little natural history papers like this in my free time. So I'm just going to talk a bit about this. So insects wearing backpacks is not uncommon. So across insects, we have things like caddisflies that pick up little bits of debris and make a case. We have Cassidine tortoise beetles that either put poop fetal shields on their backs, or they might take their previous exuviae and stack them on top of each other. And generally, this is to an anti-predation strategy to avoid predators, to trick predators. So even within my own groups, the Armic scales, they incorporate their past exuvia, the first instar, the second instar, into their cover, and then the adult bodies underneath. Some whiteflies actually do this as well, and we think that it could be an anti-parasitoid strategy to either confuse them that, they're tapping and might not know if it's already been parasitized already, or it might, actually physically obstruct them from ovipositing. So within the Neuroptera, I didn't know a ton about lacewings going into this. My background is mostly arachnids and scale insects, but I fell into this little Neuroptera project. So within the Neuroptera, they have been using backpacks to deter predators for millions of years. So there's these Cretaceous fossils where we have owl flies and their relatives and green lacewings and their relatives that have been putting various debris on their backs. So this debris might be things like exuvia from other insects, little pieces of wood, aphid bodies that they've consumed, lichen, trichomes and lichen, or even ants that they've eaten. So I'm going to kind of sum up the literature that I could find, doing a very broad review of using other insects' wax to make the dorsal packet, as it's called, the backpack of a lacewing. So in the 1920s or so, Smith said, oh yeah, lacewing larvae are using the flocculence or the wax of other insects, but he didn't say where they got it. So it wasn't until 1978 that Tom Eisner publishes this study called A Wolf in Sheep's Clothing, and he shows that this lacewing larva of Chrysopus lawsoni is using the wax of the woolly alder aphid to make its dorsal packet. And he also very nicely shows in this neat study that this packet is protecting the lacewings from ants. Because as we know, a lot of Sternorinkin bugs and other true bugs that produce a lot of honeydew are tended and protected by ants. So this whole wolf in sheep's clothing, if you're the wolf that's going in to eat the sheep, you better look like a sheep to deceive the shepherd, the ant. So since then, Eisner again found this in the 80s where these lacewing larvae were actually using mealybug wax here in Florida. So here's a lacewing larva with mealybug wax packet, and these are the mealybugs that they're taking that wax from. And again, they didn't show that this is probably going to deceive the ants that tend those mealybugs. And since then, there hasn't really been much. This nice review by Catherine Tauber kind of looked at, everything that these debris-carrying lacewing larvae might be using in their packets. And basically, it was summed up, this is a really ubiquitous predator around the world. They're really important for biological control, but we still don't know a lot about what they use to make their packets and how they do it. So in this review, they have this kind of observation of this lacewing from Asia using male pupal cocoons of Drosicia. So that was pretty neat, but they couldn't confirm the identification of the lacewing in that paper for sure. And then Wheeler and Stocks showed that this lacewing larva in, I think, South Carolina was actually using the wax of flatted nymphs and the trichomes of the plants that they were on to make their packets. But there's a lot of just photographs of lacewing larvae on the internet that, they have wax, they're definitely using wax, but where's the wax from? So I basically summed up everywhere we know where they might be getting the wax from so far in the literature, which is pretty crazy how little is actually known about them. So then you jump over to iNaturalist, and these are all lacewing larvae that not only are they using wax to make their packet, but they were misidentified as scale insects on iNaturalist by the artificial intelligence or by humans that don't know any better. So this is just a small subset. I've found over 100 now of chrysopid larvae with wax on their back that have been misidentified as either mealybugs or woolly aphids or even like Cryptolemus mealybug destroyers. Like the artificial intelligence and humans just don't know what's going on when something is waxy. So in this particular study, this bag of leaves comes in, my technician Trudy says, come over here, it's cool. And I always have my camera, and here we have an infestation of coconut mealybug, Nippicoccus nippi. So this is the same genus that Tracy was talking about, but it's a different species of mealybug. They're really common on things like avocado, sugar apple, mango. And here in this bag was this third instar Syrphidae claveri larva that is just going around this sample. And, it's been in the mail, it's been in this baggie, and it's just wrecking these mealybugs. It's going through and eating them, it's going through and making its dorsal packet. So I sit down with my camera and just start watching it for a few minutes. So it had come from Palm Beach County. And the first thing that I was watching was it just feeding on the mealybugs. So lacewings have these long piercing sucking mouth parts, and I apologize for the depth of field. This lens is not really made for videography, but it does the job. So as you know, lacewing larvae and their mealybug nymphs are very tiny, so videography gets tricky the tinier it gets, and I'm no Nat Geo videographer. But so basically I found that this is the first instar mealybug nymph, and they're sucking them dry within 45 seconds. And what was also interesting is that they were not incorporating the exoskeleton, the discards into their packet. So sometimes you see the lacewing larvae that are, putting the discards into their packet, but these were just scraping them off and leaving them behind. And what you also notice is that we have a first instar mealybug nymph that's alive, that's walking around on its back. So I will talk a little bit more about that in a second. So the next thing that I started observing was that in between voraciously feeding on these mealybugs, it was also building its packet. So it was looking a little naked in that last video, and it started to construct the packet with mostly wax from the male pupal cocoons on the leaves, but it was also grabbing wax off of the adult females and some of the second instars that had some wax. So this followed, other descriptions of how they make their packet. They use their mandibles to kind of gather up the material. They flip their head back and they scrape it off. It's not particularly delicate, or I don't know how the ones that stack to ants so well are doing that, because this one did not seem like it could stack anything well. So here is just a video of that. So it's plucking, and actually the first thing it gets is a live mealybug. So it's trying to put wax on its back, but these leaves were just so infested. It was teeming with mealybugs. So it was having a really hard time finding any clean wax to put on its back. So I started to wonder, mealybugs, they're thought of as being kind of pitiful, right? The adult females, they don't have wings. How are they even moving around? And we know from, a lot of regulatory science with scale insects that the first instar crawler is the most mobile stage. They're either dispersing via wind or via foracy. So they're grabbing onto other more mobile insects. But for the first time, here we have the lacewings actually sticking them onto their back. So could these nymphs that it's stuck into its packet actually ride on it to a new host? And how far do lacewing larvae even travel? I mean, it had plenty of food right here. Maybe it would never even leave a single plant in its lifetime. So, and we know that Nyphicoccus nipi and other mealybugs have plenty of ant associates. So we can also, predict that in this case as well, as Eisner so beautifully showed in his paper, that the dorsal packet on these lacewing larvae are probably protecting them from the ant associates. So there are no ant associates formally described for Nyphicoccus nipi in Florida, but this is a single tree I just photographed in West Palm Beach last week. And you can see the ant diversity. There are many species that are tending them for their honeydew, and many things for the predators and parasitoids to contend with if they're trying to eat mealybugs. So there's a second part to this story. Mealybugs are not entirely defenseless. So they're often tended by ants. And there's actually a nice study that just came out that shows that when there's an obligate association between ants and mealybugs, the mealybugs start to lose their defenses, called the osteoles. And these are these pairs of slit-like openings on the dorsum. They have an anterior pair and a posterior pair in most species, but not all. And here's an SEM image that I have of a mealybug osteole. And these osteoles can reflex bleed, essentially. So when a mealybug is disturbed, a little globule of liquid comes out. And what happens is that apparently, this hasn't really been looked into for many decades, but as the globule of liquid contacts the mandibles of the lacewing, it gums them up. And it gums them up so much that some studies have shown that if the osteole is contacted by an early instar lacewing that's particularly small, it can gum up their mouth parts so bad that they can't feed or ever develop into a proper adult. Or they might eclose, malformed because of this damage. So this also goes for other potential predators that kind of get gummed up by this osteolar fluid. And what they think is that, mealybugs are making all this wax, and they think that the fluid that's coming out is just hemolymph, basically, but there's a lot of liquid-free wax in that hemolymph. So when it contacts the air, it dries very suddenly and will gum up these mouth parts. And to my knowledge, this is the only video that I can find of the osteolar fluid actually in action. So, and the lacewing had this really aversive reaction to the fluid. It immediately kind of retreated and started scraping the mouth parts off onto the leaf to try to get that off. And you'll also notice that we still have some live mealybug nymphs in the packet. So I went ahead and reared this out to adulthood. From the cocoon spinning to eclosion as an adult, it took about 22 days. You can see that the mealybug wax and other debris from the substrate has been included in the cocoon. Here's the cocoon after the adult has emerged. So we have this beautiful lacewing that emerged. Just a couple shots of very pretty. I think it's like on auto to come up, so it's coming up slowly. So this is the first record of Syrphidae claveri even eating Nyppicoccus nyppi, but more exciting, it's the first time that we've seen a lacewing larva actively putting live insects into its packet, and what could be the implications of that. And despite lacewing larvae being such a ubiquitous, important biological control agent, I can't find any videos on the internet of them making their dorsal packet. So there might be some locked away on a floppy disk somewhere from one of these old naturalists that did beautiful work, but right now I can't find much on the internet for that. And also the first videos, to my knowledge, on the internet of reflex bleeding in action and a predator contacting that reflex bleeding, the osseolar fluid, and having a aversive reaction to that. So this work was just recently published in Insecta Mundi in December, and all these videos are on YouTube and linked in the Insecta Mundi article if you would like to read more. So thank you to my technician Trudy for pointing out the lacewing larva going about its business in the baggie, Sally Simmons, who is the DPI inspector that sent in this material, and Katie Tauber at Cornell for identifying the lacewing for me. Also, I'd love your scale insect bycatch for Berlesi funnels, if anybody has Berlesi samples. It's not really a group that we think of going in Berlesis very often, and a lot of times it's just the first insect crawlers. But Doug Miller and I are working on a lot of grass infesting genera, and we have this really cool thing that we think could show up in a Berlesi funnel from the roots of grass. And I'll take any questions. Thank you. Questions? Go ahead. Yeah, Andrew.
Yeah, so, did you ever see them, the lacewing larvae messing with the ovisex, trying to incorporate that being flexed?
Yeah, Nyppicoccus nyppi doesn't really make an ovisac like Miridae does. So they just kind of have the egg babies like a little bit underneath their bodies, but they don't make that huge ovisac like Miridae. Yeah. Yeah.
You expect to get them in like litter siftings, like in winter?
Quite possibly. I mean, I see them all over the place, walking on my house, on fences, I think that they're sort of everywhere. But whether they would keep the packet intact once they fall into fluid, kind of also another question. Would it be preserved? Probably not. Might just dissolve. Yeah.
Do you happen to know if they only go for mealybugs only, or do you go for?
So it depends on the species. I think that some lacewing species are more host specific. This one seems to be a generalist. So this lacewing is like a really popular biocontrol in Brazil. And when I was doing the literature review, they try to kill it with neem oil constantly to see if it's gonna, survive in an agricultural setting where they're using neem oil to try to control things. And they can rear them in the lab just on moth eggs. And what was interesting is they never talk about packet construction or anything, but they have a picture of a cocoon of one of these in the lab that had been fed exclusively on moth eggs. And the cocoon is just naked because it didn't have anything to camouflage it with. So, but I would say this species is a generalist. Thank you.
Thank you. So these was our last talk. We will have posters, right, showing up on the screen.
I don't know. I didn't plan for that. We have, did people send you PDFs of posters?
Yes, they're in our emails, but if you don't have them, it's okay. We'll put them on the CSE website.
Okay, that sounds good.
Okay.
I'll see if I can find them the next 15 minutes or so I can.
And we have time left for questions to the presenters, for those that are present, or for the posters.
Yeah. The whole group? That'll be nice. Where do we do it? Wait a minute.
There.
Oh, everybody to come as well?
Yeah.
Or how about just me taking a picture of it? I haven't done it. I've done it.
So do you wanna take the group picture now?
How do we do it now? People standing up together or just sitting whatever? It's a lot. It's a lot.
Yeah,
Before we leave, I'd like to say something. I think this is Julieta's 5th year of organizing. And I think she does a great job. I think she almost did all that.
Restaurants? Do they want to come to the restaurant?
Okay, anyone interested to come to the restaurant, just raise your hand so we know how many people.
1, 2, 3, 5, 6, 7, 8. Oh, more than 15. Okay, I'll call the restaurant.
Okay,
Yes.
okay, thank you.
How do you want to do it?
That's okay. Take a picture
A few of them, yeah.
Yeah, that's okay.
Maybe next time at lunchtime, we should go at the stairs.
Okay. So also, before we close up everything, please. Julieta said that she doesn't want to do it next year, again, the organization. We don't believe it. Okay, so we're gonna push it for her to do it again. But she did an amazing job, I think, this year. So please again. We have the annual business meeting at 6 right here. For anyone who wanna stick around, please do it. We're gonna have a summary of what we discussed at last meeting, and we have a few new issues coming up in recent days. So please stick around, and again, thank you all the presenters here live, online, all the attendees. This has been an amazing meeting. David planning to bear with us, right? And the McGuire Center for hosting us again. And
The moderators, thank you.
thank you to Andre as well, right? Our host, and thank you all for being here again. Thank you,
Understand the panel.
Should have a committee that does, then you can divide up.
So when I do.
It's not working.
I am. Sometimes I send messages, David, to help me